This is symposium season for us. This is just 2 out of 3 that we have going on.
I think we're gonna.
Awesome, really. Thank you. This is symposium season for us. This just 2 out of 3.
So I just sent the link.
Ah, yes, there it is. Okay, cool.
I think the Alana symposium we're going to do as a webcast.
Okay.
Just FYI.
Thanks. DD. All right.
Oh, damn.
All right. The link is on the YouTube page.
Wunderbar.
Yeah.
Okay, so I'm going, if we want to mute and turn off the video, I will put the slides up and make Tom host and then start the webinar.
Okay.
Okay, bye.
Hello.
Hello, how are you?
Who am I speaking with?
Oh, David Ornstein. I'm a member of the staff at Pick Hour.
Okay, so I just wanted to check in with you guys to show you I'm here. And do you guys want to check sharing my screen?
We certainly could, yeah. Brittany, I think, is kind of running our show for us for the most part. I'm just the greeter, I guess. So, Brittany, why don't you?
I'm ready.
I think this is already broadcasting, so I think we're all set
Okay,
because I want to keep sharing these slides.
okay.
Okay, thank you.
Okay, so we're going to assume everything is going to work just fine.
Yes.
Good morning. Can everyone hear me?
Yes, we can.
Thank you. I am Li Wei Cai, Picard Professor of Neuroscience here at MIT and Director of the Picard Institute for Learning and Memory. Welcome to our 2020 Fall Symposium, Internal States of the Brain. Today, we will learn from fantastic lineup of speakers about an emerging area of neuroscience with tremendous implications for understanding how the brain produces and transitions among sustained representations of feelings, needs, and desires. These internal states influence how sensory information is processed and how behaviors are generated. Yet, we know relatively little about them. Advances in neural recording technology, imaging, and data analysis, including machine learning and AI methods, are giving us new opportunities to make discoveries in this exciting area. I would like to thank our speakers for joining us today. This is an extraordinary group of experts who are using a wide variety of approaches to blaze new trails in the study of internal states. We will hear about how internal states arising from neural activity and neuromodulatory systems in specific regions influence the brain in formulating behaviors ranging from sleep to fear reactions to socialization. I would particularly like to thank one of our speakers, my Picard colleague, Stephen Flavel, for putting this wonderful program together. I also want to acknowledge the work of Picard staff, especially Brittany Greenall, for producing this event online. After each speaker's remarks, we will have time for questions and answers. You can enter questions in the Q&A box at the bottom of your Zoom screen. You can vote for questions you like, which will increase their prominence. The moderator would choose questions for the speakers to answer. So let's begin. The first session will be moderated by Steve Flavel. Steve.
All right, thank you, Li Wei. And thanks to everybody, the speakers and all of the participants who are logging in. As a reminder to echo what Li Wei just said before I introduce the speakers, if you have a question, just click on the Q&A tab at the bottom of your screen and type it in there. And you can also vote up other people's questions if you think that they're interesting and you'd like to hear them answered. Okay, so this morning for the first session, we have 3 speakers followed by a break. And the first of those speakers is Eve Marder from Brandeis University. And so Eve, take it away.
Thank you very much. And I'd like to, okay, so just 1 second while I get my pointer. There. I'd like to thank the organizers for the invitation and the opportunity to speak with you all today. And it's a great privilege to be with such an illustrious cast. Before starting, I would also like to give a special thanks to my lab, who are shown here, as they've been remarkably heroic in continuing to work and produce data and ideas throughout the last 7 or 8 months. And then I would just like to acknowledge all the people with whom I've worked over the many years. And I would just like to say that I'm equally proud of all the people who've gone through my lab, who've gone into all the different disparate things they've gone on to do. And again, this is my present lab. So what I'm going to be talking with you today about is a problem that I think is near and dear to all of our hearts and particularly relevant to today's world, which is how is individual variability related to resilience to challenge. And I'm not going to be talking about resilience to challenge by COVID, but I think this is a general problem as we face a world with large numbers of challenges. Now, most of you probably know that for my entire scientific career, I've worked using the crustacean stomatogastric nervous system. And I'm just showing you this introductory slide just to point out the two kinds of recordings from the physiology that you'll be seeing. Most of these data come from the crab Cancer borealis. It's the crab that lives out in the Boston Harbor, right on the other side of MIT. And we get them from local fishermen. Actually, we get them from a local distributor who gets them from local fishermen, and they come from somewhere in the ocean. The pyloric rhythm, which I'll be showing you in a minute, controls the back end of the animal's stomach, but crustacean stomachs are completely different from your stomachs, and they are controlled by a nervous system with motor neurons that innervate a whole set of striated muscles. So it's much more like your shoulder or your mouth than it is like your gut. And because it's rhythmically active, just like your respiratory system, it's been a remarkably good system with which to evaluate the role of individual neurons, individual synapses, modulatory neurons, etc., in the production of an ongoing rhythmic activity. And the circuit mechanisms. So the way we do the experiments is remove the stomach from the animal. We dissect the nervous system from the surface of the stomach. We lay it out in a Silgard lined Petri dish, and it's then superfused with physiological saline with the composition more or less of the seawater and at whatever temperature we decide, and that will become relevant. We desheath the ganglion. The stomatogastric ganglion in crabs has about 26, 27 neurons, and then we can record intracellular with sharp microelectrodes from the somata of these neurons. These are quite large. And when we do that, we get these beautiful intracellular recordings, such as you see here. If we look at the neuron called PD, it depolarizes, fires aversive reaction potentials, and hyperpolarizes, and this is ongoing. And this blue neuron is shown here, down on the extracellular trace. Now, it's very important for what you hear in the next few minutes that you realize that these neurons are unambiguously identified on the basis of their projections to muscles. As I said, there are 40 sets of striated muscles, and what we do is we put extracellular recording electrodes on the motor nerves, and therefore we use the innervation pattern to identify the neurons. This is a typical extracellular recording from a motor nerve up here before it's branched to go to the different muscles, and here you can see that triphasic rhythm, LP, PY, PD, LP, PY, PD. Now, LP, there's one LP per ganglion. It's a constrictor motor neuron. The PYs in crabs, there are about 5 PYs, and that's a second constrictor set of neurons. And the PD, it's a dilator motor neurons. And so we have a constriction 1, constriction 2, dilation, constrictor 1, constrictor 2, dilation. And this rhythm is ongoing in the animal, like your respiratory rhythm, as long as the animal is alive. Now, like your respiratory rhythm, it can vary in frequency and phase relationships to some degree, but basically it's always on. And because it's always on, it's particularly useful for understanding how circuit dynamics arise from interactions of the component properties. Now, on the right, I'm showing you a connectome. This is what we used to call it as a connectivity diagram. Today it would be called a connectome. And in all of my pictures, all of these black circles are chemical inhibitory synapses, and the resistor symbol means electrical coupling. So the first thing you'll notice is that there are no excitatory synapses within this network. Everything is inhibitory, and the follower cells fire on rebound from inhibition. So rebound from inhibition is a very important timing cue in the way the stomatogastric ganglion works and in the way many rhythmic systems work. So just to give you the 1980 version word model of how this connectome gives rise to these dynamics. The AB neuron is a neuron, it's a bursting pacemaker. It's electrocoupled to the 2 PD neurons, and therefore the PD neurons fire in a burst. And then when these 2 neurons are coactive, as they are, they rhythmically inhibit the LP and the PY. The LP pops up, recovers from its inhibition first. It holds the PY down, and then PY pops up, and it turns off LP, and then the cycle continues. So As I said, that's the 1980 version. That word model is still true. But if you really think about trying to understand how all of the voltage and time-dependent conductances in this circuit give rise to these dynamics, and then what gives rise to changes in these dynamics, you realize you have to characterize all the voltage and time-dependent conductances in all the cells, the strength and time course of all the synapses, and then if you use biophysics to do so, you need some way of putting it all back together again. And that's why starting in the late 1980s, I started collaborating with FIRST and then doing a fair bit of computational theoretical work, because if you want to put back together again, follow the reductionist path, describe the properties of the components, and then really try and understand how the system depends on those properties, you need some way of putting all that information back together again. So I'm going to show you a mixture of experimental and computational work. The computational work will largely be done with models of this form. So I'd like to spend a moment showing it to you. These are conductance-based models. There's mostly single compartments. And in most of the models, there'll be an inward sodium current, 2 inward calcium currents with different properties, a hyperpolarization activated inward current or IH, 3 potassium currents, including an A current, that's the fast transient outward current, calcium activated potassium current, and a delayed rectifier. And then the challenge is to understand for a model, and then there's a leak current. The challenge is to understand what role each of these conductances plays in producing the dynamics of the cell. Now, as an electrophysiologist, as a biophysicist, we don't really have a way of evaluating what each of those currents is doing at any moment in time. But with models, you can. And I'd just like to spend 1 minute showing you this method that Leandro Alonso developed to be able to visualize the contributions of all these currents to dynamics. So this is now a model. On top, you have a bursting neuron. It's hyperpolarizing, depolarizing, finding a burst of action potentials and then turning off. And this would continue over and over again. And now, here we have the 0 current. We're using biophysical sort of nomenclature, or where outward currents are plotted upwards and inward currents are plotted downwards. The total outward current is seen here in black on a very compressed log scale. So there's a lot more current up here than there is down here. And now the contribution of the different outward currents to these dynamics is shown here. So for example, in purple, we see the A current, that's transient outward current, the A current. And you can see how it turns on and off over time. The calcium activated potassium current is seen here in orange. The delayed rectifier is shown here in yellow. The leak is shown there in pink. And now in the inward currents, you can see the sodium current in red and the 2 calcium currents and the H current. Okay, so this allows one to actually see what's going on in these dynamics. Now, on the next slide, this slide is actually simpler than it looks, so I'll talk you through it because this slide has the sort of the guts of everything I'm going to be talking to you about today. So for a lot of years, we've been talking about there being multiple solutions or degenerate solutions to producing very similar behavior. And I'm showing you that here in this way of visualizing it, and then we'll show it to you again and again in different ways. So what Leandro's done is he's created 6 models, A, B, C, D, E, F. Notice these models have very similar behavior if you just look at their voltage waveforms. But now if we use Leandro's way of visualizing what's going on with currents, let's just first look at the A current. Here it's very small, it's doing almost nothing, it's contributing very little. Whereas down here, the A current is contributing an enormous amount to the dynamics of the cell. And you can see that each one of these models has an A current, but they go from being very, very unimportant or seemingly insignificant to being very important. If we look at the calcium activated K, the orange one, you can again see the relative contribution varies. And likewise, that's true of all the others. So now up here, we actually just see the values of the conductance densities, the maximal conductances. I'd just like to point one thing out. The A current here is is doing more than it is doing here, although the maximal conductance, the number of channels in the membrane, is higher here than it is here. And that's because of all the other currents and the way in which the dynamics evolve. Okay, so we have 6 models with very similar behavior, but quite different underlying mechanisms. And I think for those of you who worry about mechanisms, it's very, very important to realize that you can't just look at what the behavior of something is and make inferences about the importance of a particular property or component. Now, this follows on a paper that most of you probably have seen before. This was the very influential paper that Astrid Prince did, where she created 20 million models of the pyloric rhythm. And this is where our real multiple solutions work started. This is model network 1, model network 2. And here you can see one set of parameters that give rise to model network 1. So now this is on the network level, the same message I just showed you. And this is model network 2. And I'd just like to show you that here in model network 2, the PY to LP synapse is big here and is smaller here. The AB to LP synapse is big here and small there. The H current here is big, here it's much smaller. So this set of parameters and this set of parameters both can produce very similar behaviors. Now, when we first started talking about this, most biologists were very relieved, as was I, because it said that some of the variability we were seeing in our biological measurements was likely real. That creates a whole lot of other issues about how to think about variability, and that's what we've been doing. But theorists pointed out, and rightfully so, that just because this set of component parameters and this set of component parameters could give rise to similar behavior under control conditions doesn't mean that there wouldn't be perturbations that would separate the two. And that's certainly true. In the universe of all possible perturbations, there's bound to be a perturbation that will separate the behavior of these two networks. And so then as a biologist, I started thinking about if my crabs were really well described by this kind of differences in component properties, then the question is, what would happen to the perturbations that the crab would normally see in his life? So this slide, or this work, gave rise to these questions. How variable are the parameters in real biological networks? Do perturbations reveal cryptic animal-to-animal differences? How reliably can animals with intrinsic variability respond to perturbations? Are some sets of parameters associated with higher resilience? And I think this is the question that we don't really understand yet, but is very, very important as we start thinking about internal states and the role of internal states or parameters in then configuring the network to respond to perturbation of all kinds. So I'm going to very briefly answer the first question for you. And maybe what I'll do, because we don't have much time, is I'll skip through the raw data, but only show you, just point out one. These are recordings from the LP neuron in one animal. The second animal, notice in the network, they're doing very similar things. Nonetheless, in voltage clamp, the calcium activated K current is much, much bigger in LP2 than it is in LP1. And as we measure all three of the conductances, we see a 2 to 6-fold range in conductances across cells. That's the same identified neuron in different animals. And the same is true for mRNA copy number when the single cells were dissected and individually measured in this early study in the early days of single cell real-time PCR. And then in more recent data, this is 2020, Dave Schultz and our lab. Now here are measurements, and we've done the same thing in both single cell real-time PCR and with RNA-seq. You see about the same 2 to 6, 2 to fold 8 range of mRNA copy number. On the other hand, and this is work that Adrian Topolik did, just to make a slide for talks, you can see these are recordings now, extracellular recordings from NL1, NL2, 3, 4, 5. The pyloric rhythms that you see are very reliable and very, very similar, despite the fact that there is this range in their parameters. And so here are now intracellular recordings of 3 animals, PD cell, the LP, the PY. Again, you can see how similar the waveforms are, even though all the currents that give rise to those waveforms are quite different across preparations. So individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when they have similar behavior. And I think this is a general feature. You see this kind of variability anytime people have looked in ways that they can measure it. And that means you have to unpack your data and not just plot means and standard errors, but really look at ranges and then start looking at correlations. And so there are a whole set of really interesting correlations that I won't have time to talk about in these properties. Okay, so what we wanted to do then, after we established that there was this variability, then the question is we wanted to start looking for perturbations and biologically realistic perturbations that would allow us to evaluate whether crabs with this 2 to 6-fold range of variability could nonetheless respond reliably to the perturbations that they see. And so temperature is a logical perturbation to study because it's a global perturbation that influences all biological processes, but does so differentially so that the relationships among processes will be altered. And this is really critical. If you think about it, you realize that if temperature affected every biological process exactly the same, then there'd be no problem in maintaining function as you change temperature. But if the dynamics are affected differentially, then you might see all sorts of problems. We've also studied pH, and we've also studied high potassium, but I won't have time to talk about these. Okay, so just to remind you, we often express the effects of temperature as a Q10, which has an exponential in it. A Q10 of 1 is a process which is temperature invariant. I was taught when I was an undergraduate that biological processes had Q10s of 2. That means to say they approximately doubled for a 10-fold degree change in temperature. Q10 of 3, notice the effects of the exponential. Now, most ion channels have Q10s between about, for their Maxwell conductance and the rate constants for all the activation and inactivation of their channel proteins, have Q10s in the 1.5 to about 4 range. However, obviously the channels that are important for temperature sensing can have Q10s that go at 10, 20, 50, or even 100. So there are specialized ion channels with very, very high Q10s, but most of the Q10s we're going to be talking about are in that range of sort of 1.5 to 4. So now why is this a problem? And this is a model that Tim O'Leary built a number of years ago just to point out what the problem is. Here we have a reference temperature of 10 degrees. This is a model, a bursting neuron, 3 different versions of it, and at 10 degrees, they all look the same. But then Tim used one set of Q10s for the first model, a different set for the second, and a different set for the third. And you can see that as he changes temperature now, this model goes from bursting to being a single spike burster and then into a depolarization block in the heat. This model here, when you cool it, goes into a single spike burster, similar to that. But when you depolarize it, it goes into a hyperpolarized block. And this one shows different behaviors. And that's because the dynamics of all those currents that are important for this bursting, if those currents have different relationships to temperature, you can easily imagine you would get what we call crashes. So, but what about the animal? So here's the pyloric rhythm. These are data from Lamont Tang at cold temperatures, 7, and at 19. So you hear PD, LP, PY, a beautiful pyloric rhythm. 19, beautiful pyloric rhythm. Notice that the phase relationships, that is to say the relative timing of the activation or the turning on and turning off of the different elements is maintained, although the frequency is increased. And this was what I found most stunning from Lamont's early work, which is now we're plotting the phase relationships, the relative time that PD turns on, turns off, LP turns on, turns off. And notice they're completely temperature invariant, even though the frequency is going up. So this tells you that the crab manages to find solutions that allow it to maintain its canonical function. It's 1, 2, 3, 1, 2, 3, 1, 2, 3, as the frequency is changing, as temperature is changing. Okay, and this is just another slide that shows you that with intracellular recordings, 7, 11, 15, 19, 23, this beautiful phase maintenance, although, and here you can see that with superimposed traces, which says that these animals, and they all do this, they all do really well up to about 23 degrees, and they all have these waveforms that are virtually superimposable. That said, if you go higher in temperature, and Lamont did this in this early work, and I apologize for this typo that I keep noticing on the slide. And these are now data from 4 crabs, A, B, C, D. Notice as he went higher in temperature, you get what he called intermittent crashes, and they all look different. And the fact that they all look different is exactly what you would expect if each one of them had a different set of underlying conductances. So if the burst is evolving by slightly different cellular mechanisms, when you perturb it past those permissive temperatures, you'd expect it to start crashing, and start crashing, in this case, differentially. And so this was the fundamental finding I wanted you to see. Now, that said, because we're talking about differential resilience, I'd just like to show you some recordings that we made in 2012 after the winter that never was. The water never got cold. And here you see 11, 20, 25, 30, 36. These are now 6 or 7 hertz rhythms, which we had never ever seen before. And we realized that when the animals see very long-term higher temperatures, either the animals that would have died are all dead, and we're only seeing those animals with solutions that allow them to be temperature resistant, or all of the animals we usually studied went to Maine, along with the squid and the dogfish, which also went to Maine that year, etc. But you can see this kind of long-term adaptation gives us pause, because every year we check to see where our crash points are, and we're seeing, in a sense, we're tracking the water temperature and climate change, and it's very upsetting. Okay, but now, Leandro, not only did he build and develop that beautiful way of visualizing what's going on with currents, he also used a genetic algorithm to find models that would be able to respond to temperature with increased frequency and constant phase relationships between 10, 15, 20, and 25 degrees, and then to crash. And so now we're going to use these models to get a little bit more insight into those different mechanisms. So these are 5 versions of the model with different values of Q10s for different currents and different conductance densities. And this is A, B, C, D, E. And just like those 4 traces from the biology I showed you, these models are all at high temperature, are all crashing differently. And see, these are slow time based recordings. So this is the same recording spread out, and you can see this, these are all disordered rhythms, but they're disordered differently. And that's precisely because they have these different underlying structures. So now this will be disorder, even though they're all fine up to 25 degrees. Okay, so the conclusions from crashes were that at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation. And this is really important because at low temperatures, they all are well behaved. So they all have a solution which allows them to deal with the normal temperatures that they see. Now I should say these crabs see temperatures from about 3 degrees to about 24, 25 degrees in their normal life. And that's both seasonally, and they can see as much as 10-degree temperature change within a day. So they have to be able to deal in the north, around the Boston area, they have to be able to deal with those temperatures, but they don't routinely see temperatures up at 35. Each individual animal crashes with individual dynamics as predicted by their underlying parameter differences. And then I didn't show you the data, but I'll tell you, some neuromodulators can expand the stable range of temperatures while others make the preparations more temperature sensitive. Now I'm going to show you here my most favorite slide of 2020. And this is another slide from Leandro's work. Now what he's showing you here is the last spike of a burst, just the last spike of a burst. And he's showing it with looking at these currentscapes. And notice at 10 to 25 degrees, I want you to just see what's happening to some of the currents. At 10 degrees, the A current is big. And then as we go warmer and warmer, notice that the A current is getting smaller and smaller. At 10 degrees, the calcium activated K is small, and it's getting bigger and bigger and bigger and bigger. And likewise, the relative proportion or the relative contribution of all these currents is changing over this temperature range. But the important thing is, and the reason I like this slide so much, is the system is smoothly transitioning from a spike repolarization, burst repolarization, which is quite dependent on the A current and the delayed rectifier, to one here, which is much more dependent on the calcium activated K current. And the fact that there's a smooth transition means that the animal or the neuron in the network can respond absolutely smoothly as it's moving through these different underlying mechanisms. And to my mind, the hypothesis I take from this is that one of the reasons why we have so many potassium currents and so many calcium currents is because they have overlapping voltage and time dependencies that allow you to smoothly transition between dynamics dominated by different currents. And it's precisely that that gives us our robustness to perturbation. Okay, now I only have a minute or two left, so I'll show you one or two more slides and then I'll sort of end with a summary of what else we know. So these are two neurons. This is neuron 1, neuron 2, and this is a burster. Both of them are bursters at 10 degrees, 25 degrees. And now in this one, we can look at the sodium current, and at 10 degrees it's small, at 25 it's gotten much bigger. However, in this model, the sodium current is bigger at 10 degrees and smaller at 25 degrees. Here you see again, one of the calcium currents is small at 10 degrees, much, much bigger at 25 degrees. Whereas here, it's bigger at 10 degrees and smaller at 25 degrees. So these are two perfectly good bursting neurons that respond to temperature in a very, very different way as we move through this conductive space. And then I would just like to show you one more example of this. This is important for people who think about pharmacology. Here we're looking at 10 degrees, a network with the A current at 100%. And now we're turning off the A current. That's equivalent to doing a knockout or pharmacological blockade. And you can see the network crashes. However, that same network now looking at 25 degrees, notice it's going faster as you expect. You turn off the A current and it's doing just fine. So in this case, the A current is much more salient at 10 degrees than it is at 25. And now in a different network, we'll look at the calcium activated K. Here you have it at 10 and you turn it off and it's going slower, but it's perfectly fine. However, at 25 degrees, here you have it, and now you turn it off and it's completely crashed. So that tells you that the effects of a pharmacological agent or something else can be very, very different. Now, I'd just like to summarize individual similar starting circuit performance with relatively similar circuit mechanisms in the control can nonetheless show entirely different crash mechanisms. And this is a cautionary tale for thinking about disease. Now, I won't have time to go through this, but I want to make the point that randomly sampling parameter space shows that neurons that are temperature compensated are not common in the population. That is to say, not all multiple solutions will do this. Most of them won't. So the question is, we had to use fairly complicated genetic algorithms, other algorithms to find the sets of parameters that can be temperature compensated. But obviously in biology, crabs can't do that. So how did they find them? And I'll just summarize that and tell you the way we think, the way we think that occurs is that homeostatic self-tuning models, which Tim O'Leary has built, can find they generate correlations, they generate multiple solutions, and the correlations that occur in those models allow you to find solutions that can be temperature invariant. And then I'll just summarize at the end: differential resilience in the population is a necessary feature that comes from the inherent degeneracy or multiple solutions to producing similar behavior in healthy nervous systems. Some mechanisms for differential resilience are automatic and arise from the properties of ion channels, receptors, and signaling. And some mechanisms for resilience will depend on homeostatic mechanisms, behavioral adaptation, and learning. And I'm going to stop there, and thank you.
All right, thank you for an amazing talk. We have a bunch of questions here, and so I'll dive right in. Caroline Schmidt asked, have you seen differences or perturbation trends based on gender, age, etc., like other properties of the animals that you're recording?
All the animals we study are male, and that's because the fishermen throw the females back. So if we wanted to study females, we would have to make deals with fishermen, and they're very difficult to make deals with. We've tried. All of these animals are adult. They're 5 to 7 years old, which means they have all lived through the North Atlantic Ocean for a number of years. They've seen changes in temperature. They've seen changes in their environment. So they are the robust ones. And I think it's important to say that we're seeing them as well. And actually, we're having trouble right now with our crabs because they're molting too late, and we had this really weird too warm water. The water was much warmer this summer than it was supposed to be. And so we're become very, and you don't notice this until you challenge them. At 11 degrees, they're fine. It's only when you start perturbing them that you reveal these cryptic changes.
All right, cool. And then let's see, Eric Melanakos asks, are there popular combinations of conductance parameters? For example, if you plotted the maximum conductance values of different neurons on a multi-dimensional plot, would you expect there to be clusters representing cells with similar combinations?
Right, so I didn't have time to talk to you about all the correlations that exist, but there's a very, very interesting set of two-dimensional and probably some higher dimensional correlations, which are cell type specific. So for example, there's a very strong correlation between the expression of IA and IH that we see in most cell types, but there are other correlations which we see only in some cell types. And so the set of correlations that we see seems to be cell type specific, but many of the correlations you see in multiple cell types.
Interesting. Cool. And then Harris Kaplan wrote in, are the individualized crashes at higher temperatures repeatable within individual animals? In other words, are those true lifelong individual differences, or could they be based on the animal's individual internal state?
Well, I mean, that turns out to be a very hard problem. So when Lamontagne was first doing this work, he would crash the same animal repeatedly. And he wanted to ask the question, does an animal repeatedly crash more similarly to itself than animals crashes across animals? Well, it turns out that when he did this originally, we didn't have a good enough way to characterize the crashes. It's very easy to characterize the normal state because it's very regular. And the crash dynamics are so interesting and unusual that it's only now that we have a good enough way of actually visualizing and measuring so that we can actually quantify, put on some, we've tried over 10 years, we've tried, so that we can put on a really good quantitative basis, a way of characterizing the crash dynamics to make that comparison. So I think we're getting there. Anecdotally, we think that animals crash more like themselves than they do like others. But there's also a lot of lack of stationarity, and then there's hysteresis, and that also depends. So there's a lot of sliding in this. It's a hard problem, and I think that's really interesting that it's a hard problem.
Great. Yeah, good question. And then maybe time for one more. So the question is, really fascinating work. How likely is it that the relative expression levels of the different potassium channels is genetically hard-coded versus the case where each neuron learns to adjust its relative expression levels to achieve smoothness across different temperatures?
Well, it's not smoothness across temperatures, it's smoothness across lots of different perturbations. Remember, the same mechanism will hold for many other perturbations. I think there probably are some genetically hardwired sets of correlations so that the IHIA correlation may be hardwired, they may be co-trans, who knows what molecular mechanism there is. However, the actual values of IH and IA vary across the population. And remember, these are 5-year-old animals, so they have replaced every single ion channel in their membrane many, many thousands of times. And so the homeostatic mechanisms, which would be the subject of another talk, that allow them to constantly replace their ion channels, means that the conductance densities they have today were not the conductance densities they had 6 months ago or even in 2 weeks. So they're constantly retuning and self-tuning using rules.
And maybe one question I had related to all that is, as you looked at the mRNA measurements in real animals, have you sampled enough animals at this point, or enough cells that that you've been able to kind of ask the question of whether there are subsets of solutions that maybe work well in the models that you aren't seeing in kind of the biological case?
I think the models are conceptually useful, but not useful to talk about real neurons. I say we have the same currents in the models that we have in the cells, and we've actually tried to use dynamics that are pretty similar. But remember, these are single compartment models. We don't have single intracellular signal transduction pathways in there. So I think they're not to be believed. That is to say, I think they're conceptually to be believed, but not to match one-to-one with the biology.
All right. Thank you so much for your talk. And we'll move on to the next speaker. And the next speaker is Mario De Bono from IST Austria, who will be telling us about a talk titled Regulating the Regulator.
Hello. Can people see my slides?
Yes.
Yep.
Good. Thank you. So it's fantastic. It's fantastic to be here. I just noticed there's more than 450 people in the audience, which is, despite all the bad things about COVID, it's one of the good things that people can come together by Zoom. And the story I'm going to tell you about today, it's unpublished, and it's the work of 2 people in the lab who I'm showing you here, a postdoc called Tan Vuong Brenda, and a student called Sean Flynn. And the regulator that I'm referring to is calcium signaling, which is so central to so many processes in neurons, including things like homeostatic plasticity, which Eve has studied so extensively, and also learning and memory. So calcium signaling is a key regulator, and much of calcium signaling happens through calcium binding calmodulin. And the question, the way we came into this study is by studying a switch in state of C. elegans that's evoked by changes in the environmental levels of oxygen. And when you look at the behavior of C. elegans at 21% oxygen or at 7% oxygen, the behavior is actually quite dramatically different. So at 21% oxygen, which is the oxygen levels at the surface of the Earth, these animals are hyperactive. They don't like that environment. They're trying to escape it. Whereas if animals are kept at slightly lower oxygen levels, here I'm showing you 7%, then the animals are relatively quiescent and feed. Now, if you keep the animals at these different oxygen levels for long periods of time, they will reprogram their behavior and their physiology. They change their preferences for various sensory cues like pheromones, for example. The animals also use this escape behavior to aggregate together into groups. And we have used this as a way to get at the mechanisms, at the neural mechanisms that underpin this switch in state. So we've done extensive forward genetic screens where we begin with animals that aggregate strongly, and then select mutants that fail to do so following mutagenesis. And we've identified more than 500 mutants of this kind. These mutants, I should add, overtly look fine. They can move fairly normally. And then we phenotype these mutants and look for a particular leitmotif. And I'm showing you in here how many of these mutants respond to a stimulus strain. So they have defects in their escape from 21% oxygen, but they respond normally to hypoxia, which evokes a very similar behavioral escape response. But they also have sensitized responses to CO2. And when we see this sort of fingerprint, we know that these mutants are very likely to be defective in this compact neural circuit that's been studied by a number of labs, including Cory Bargmann's lab and my lab. And so these mutants have led us into a number of protein complexes and mechanisms. And today I'd like to talk to you about just one of these. And the work focuses on this transcriptional activator called CAMTA. So this transcriptional factor, so it's a DNA binding protein that also has calmodulin binding domains towards its C-terminus. And it's been studied in the context of human populations because humans that have are heterozygous for mutations in one of the two orthologs of CAMTA, CAMTA1 in humans, have episodic memory defects. They also have intellectual disability, ataxia, and some amount of neuronal degeneration, particularly in cerebellum. However, the mechanisms, the detailed mechanisms by which these phenotypes emerge was not clear. And we thought we'd perhaps get insights into this by studying this protein in the worm. This is what C. elegans CAMTA1 mutants look like. So the control on the left, these are animals aggregating together on a lawn of food. And these are CAMTA1 mutants which don't show this behavior. Now, when we characterize other phenotypes in these mutants, we show that they have quite a pleiotropic behavioral defects. So for example, they show hyperactivity. They're defective in their response to salt. So instead of being attracted to salt, which wild type animals would be, these have a defect in their attraction to salt. And they also have defects in response to odors like benzaldehyde and diacetyl. And these defects can be rescued by adding a transgene for a wild type version of this transcription factor. And this protein is expressed very broadly in the nervous system. In fact, probably most neurons in C. elegans express this transcription factor, and also work in the fly has also shown from the Yamamoto lab has also shown that in the fly the ortholog of CAMTA is also expressed very broadly. And work in the mouse and in the human brain have also shown that this transcription factor is quite broadly expressed. And I thought this was quite interesting, actually. Why would you have a transcription factor that's expressed so broadly in the brain, a calmodulin binding transcription factor? So we started doing some imaging work using calcium sensors, in this case YC260, to see how defects in this protein alters neural signaling. And I'm showing you here the calcium response in this oxygen sensor called URX. And what you can see is that compared to wild type, these CAMTA1 mutants show an increased calcium response across a whole range of oxygen stimuli. So they always seem to be responding much more strongly than wild type animals. Now we've extended the analysis to other neurons. So for example, if we look at carbon dioxide sensors like this BAG neuron, again, we see that these BAG neurons respond by elevated, with increased calcium responses to a CO2 stimulus. So here we're giving a 5% CO2 stimulus or a 1% CO2 stimulus and so on. And same when we look at another neuron called AFD. This is a temperature sensor and it also responds to CO2. And again, if we disrupt CAMTA1, we get elevated sensory responses to the stimulus. Now, not all neurons show increased excitability. for example, this interneuron, RMG, which is actually connected to URX by synapses and also by gap junctions, shows the converse phenotype. RMG neurons are actually less active when you're defective in the counter transcription factor. We've also done experiments where we have overexpressed the transcription factor in a set of neurons to see what happens, again, to the calcium responses. And here what we find is that you can toggle the transcription factor and toggle calcium signaling. So in the oxygen sensors, whereas loss of the CAMTA causes increased sensitivity to stimuli, if we overexpress the transcription factor, specifically in these neurons, we get decreased calcium responses. And similarly, if we look at these other neurons, these BAG neurons, again, we can see that if we overexpress the transcription factor selectively in these BAG sensors, we now get reduced calcium responses. So it looks like this transcription factor can somehow regulate the excitability of neurons, pardon me. So what's going on? To try and figure out what might be underpinning this change in property, we profiled each of these 4 groups of neurons by using FACS sorting. So we labeled them with GFP, we then FACS sorted them from associated animals, made libraries and sequenced them and compared the expression in wild type and mutant animals. And what we find was something quite striking. So in each individual animal, we find many genes changing expression pattern. But when we overlap all of the different neurons, so AFD, RMG, BAG, and so on, we find that there's only 2 genes that are always being regulated and regulated actually in the same direction. And one of these is calmodulin. And we thought this was quite interesting. And so this is here what I'm showing you is the fold change in calmodulin levels in each of these neurons in wild type and CAMT1 mutants. And you can see that in the absence of the transcription factor, there's between a 4-fold and a 2.5-fold changes in the level of calmodulin. And this is actually quite significant because it's known that the levels of calmodulin in cells and in neurons is actually lower than the level of the expression levels of its many targets. It's actually hundreds of targets. And so if you change the levels of calmodulin in a neuron, you will change the ability of calmodulin to modify it. You'll change the properties of the calcium evoked changes mediated by calmodulin. As you know, calmodulin regulates a whole variety of proteins, including the CAM kinases, calcineurin, phosphodiesterase, and so on. This is just really a slide to highlight the centrality of calmodulin as a calcium receptor, regulating many aspects of neural function. So the question we then wanted to ask is, can we actually rescue these phenotypes of CAMT1 mutants by simply changing the levels of neuronal calmodulin? And to do this, we put a cDNA for calmodulin under the control of a pan-neuronal promoter called RAB3 and made transgenic animals and then assayed behavioral phenotypes. So this is what I showed you earlier. This is wild type and CAMT1 mutants, and you can see the effects in these responses to salt and odors. And now if we transgenically express calmodulin in neurons, we can rescue these phenotypes. So sodium chloride, odors, and so on. So it seems that adding calmodulin then, increasing the levels of calmodulin in neurons can actually rescue these behavioral defects in these transcriptional factor mutants. We've also looked at how the calcium imaging defects, whether the calcium imaging defects can be rescued in the same way. And again, we can see here is URX. You can see that in the CAMT1 mutant, URX neurons respond more strongly to an oxygen stimulus. And again, we can rescue this by expressing just calmodulin from a pan-neuronal promoter. So we think this is quite interesting. We think that what we've discovered here is a transcription factor that seems to be regulating levels of calmodulin in neurons. One obvious question is this actually conserved? This is just a peculiarity of C. elegans, or is this actually more broadly conserved? So to address this, we turned to the fly in the first instance, and we collected fly heads, and then made Western blots to find in wild type and 2 alleles that have defects in the Drosophila ortholog of CAMTA1, and we find that we find a twofold decrease in calmodulin expression in both alleles. We've also done qPCR, and we find exactly the same thing, that in these Drosophila CAMTA mutants, the level of mRNA is decreased by about twofold. And interestingly, there's a study published a number of years ago in Arabidopsis, where they showed in vitro that an ortholog of CAMTA from Arabidopsis can bind upstream of the calmodulin two calmodulin promoters in plants. This is shown in a gel retardation assay. So here you can see that if you have a wild type promoter element, it will in vitro bind the protein and be retarded. So here's the free probe and here's the retarded probe. And if you have mutations in the binding site, then you don't get this retardation. This work did not actually go on to study whether this in vitro binding was relevant in vivo, but we wonder if this is actually actually much more broadly conserved. So an obvious question is, how is this transcription factor regulating calmodulin expression? And so we did a chip experiment, chromatin immunoprecipitation, using a knock-in, a functional GFP knock-in into CAMTA, and then using an anti-GFP nanobody to pull down chromatin and sequence it. And we find 3 peaks that are upstream of the calmodulin promoter, calmodulin gene, shown here, A, B, and C. And so we then went ahead and used CRISPR-Cas9 to delete these binding sites. And what you see is that if we delete 2 of these binding sites, B and C, the animals appear wild type. If we delete just A, they are also wild type. But if we delete all 3 binding sites, then we have a mutant phenotype. The animals no longer aggregate. And moreover, they also exhibit defects in their responses to salt, benzaldehyde, and diacetyl. So in other words, deleting the binding sites for this transcription factor upstream of the calmodulin promoter is sufficient to phenocopy loss of the transcription factor itself. So calmodulin seems to be the principal target which generates these behavioral pleiotropic phenotypes. And we can again rescue these phenotypes by expressing calmodulin specifically in neurons. So then we having done all this work and shown that this transcription factor regulates the expression of calmodulin in neurons, we wondered whether actually calmodulin, which is such a central regulator, might be able to regulate its own expression. So to test this, we set up the following set of reporters. We made a reporter for the calmodulin promoter itself, hooking it up to GFP. So that's reporting expression of calmodulin. And then we made 2 other constructs, one a control and one an experimental, in which we expressed from a pan-neuronal promoter calmodulin in a polycistronic message with mCherry, or just mCherry. And we needed to have the mCherry there because calmodulin is of course expressed, calmodulin is of course expressed everywhere, not just in neurons, and we needed to identify neurons by using the mCherry. And so then what we can do is use the mCherry to recognize the neurons and then look at how the GFP levels from the calmodulin promoter changes according to the presence or absence of exogenously added calmodulin. And what we find is that calmodulin actually negatively regulates its own expression. So if we look at relative GFP fluorescence in the control, where we just have mCherry, there's a substantially higher levels of GFP compared to when we overexpress calmodulin paneuronally. The obvious next question then is, given that this CAMTA transcription factor can bind calmodulin through both IQ motifs and also calcium calmodulin, a calcium calmodulin binding domain, whether this negative feedback might be mediated by the CAMTA transcription factor. So here I'm showing you again the structure of CAMTA with these IQ binding domains at the C terminus. There's also actually, although I'm not showing it here, overlapping with these IQ domains, a calcium calmodulin, a predicted, I should say, calcium calmodulin binding domain. And so what we did is we used CRISPR-Cas9 to disrupt all of these 4 IQ domains by mutating this isoleucine to an asparagine, and then asked what happens in our experimental scenario. And what you see is that if we knock out these IQ domains in this allele here called the SYB1919, calmodulin loses its ability to feed back, to control its own expression to control its own expression. So we think then that one of the principal ways in which calmodulin can feed back to negatively regulate its own expression is by binding these IQ motifs in this CAMTA transcription factor. And so the model that we have is that when calcium levels enter and bind calmodulin, of course, it goes and regulates all the calmodulin binding proteins, but the levels of calmodulin are being regulated by CAMTA binding to the calmodulin promoter and promoting its expression, unless there is too much calmodulin, and that can feed back and inhibit the expression of calmodulin. So in other words, this transcription factor can both promote the expression of calmodulin, as I showed you earlier in the talk, and also inhibit it. So 2 other questions that one might ask are, do the CAMTA neural phenotypes reflect dysregulated calmodulin levels in mammals? Does this also extend to vertebrates? And we have a knockout now for CAMTA. We don't have the results yet. We're just waiting for the mice to breed. But there are a couple of experiments that suggest that this might be the case. Oh dear, what's going on here? Okay. So firstly, in flies, work in flies by the Lee lab has shown that in fly photoreceptors, mutants in Drosophila CAMTA results in a defect in the termination of the photoresponse. So this is the wild type photoresponse. Here's the light stimulus. When the light goes off, the photoresponse terminates quite quickly. In this allele of Drosophila CAMTA, the response fails to terminate. And interestingly, some years before this work, there had been a study looking at a mutant of calmodulin, promoting mutation of calmodulin, which significantly reduces calmodulin levels. And these mutants show very similar phenotypes actually in the photoreceptors. And so we wonder actually whether this reflects the ability of CAMTA to actually directly regulate calmodulin in the fly. What we've shown so far is just looking at the entire fly head. We haven't actually looked specifically in photoreceptors. There's also work looking at the other ortholog of CAMTA in mice, CAMTA2. So CAMTA2 in mice regulates cardiac growth. So if you overexpress CAMTA2, you actually cause hypertrophy of the heart. And again, interestingly, there's a parallel study looking at overexpression of calmodulin, specifically in cardiomyocytes, and you get the same phenotype. You get a hypertrophy of the heart. So we wonder, and we want to now test these hypotheses, whether in fact this ability of this transcription factor to regulate calmodulin levels is actually also relevant in vertebrates. And we think this is interesting because, as I said, toggling this transcription factor up or down in worms seems to be able to toggle the excitability of neurons. And the obvious question that comes from that is whether this transcription factor's function is itself regulated, because you could imagine if its function is regulated that you could tune calmodulin mediated processes by regulating the function of the transcription factor in all sorts of interesting ways, mediated by feedback based on calmodulin binding. So we don't know whether this is the case. We do have some insights into it, because we know that at least in C. elegans, this transcription factor is predominantly cytoplasmic. So this is a GFP tagged version of calmodulin expressed from its own promoter. And you can see here nuclei highlighted with a tag RFP targeted to nuclei. And you can see that most of the transcription factor, of the CAMT1 transcription factor, is actually not in nuclei. And it's only by disrupting a nuclear localization signal and also the ANC motifs in this protein that we can get it to go into the nucleus. So this may mean that that nuclear entry of this protein is actually regulated, and this is something that we'd like to study in more detail. So that's my last slide. I'd like to again thank the people who've done the work, Than and Sean, and I'll stop there and I'll take questions.
All right, thank you for a fantastic talk. So there are questions coming in here. First question from Augustin Almaril-Poros is, what was the other gene that had a differential expression common in all the neuronal cell types?
Yes, so it's actually a gene that is not conserved. So we don't know what it's doing. So we will do some work to try and find out what it might be doing. But at this moment in time, it's sort of an orphan. It has no homology. But it is interesting that it's there.
All right. Cool. And then this next one from Alison Hansen. If CAMTA is required to regulate calmodulin expression, why would simply overexpressing calmodulin in an animal without CAMTA rescue the mutant phenotype?
Could you say that again? Because I'm not sure yeah.
Sure, I'll read it one more time. If CAMTA is required to regulate calmodulin expression,
yeah,
why would overexpressing calmodulin in an animal without CAMTA rescue the mutant phenotype?
So, well, what we're overexpressing is calmodulin itself. So the concept is, the CAMTA is promoting the expression of calmodulin. That's one of its functions. So in the absence of CAMTA, you have lower levels of calmodulin. And as a result, we think of the lower levels of calmodulin, the neurons have altered properties. They either have these elevated sensory responses. They have defects. They have defective responses. But if the main function of this transcription factor is to promote calmodulin expression, then by adding more calmodulin, we can rescue it. And that's in fact what we can do. So we can just add more and compensate for its absence. But there may be some confusion also, because of course it also acts not only positively, but also negatively. It also acts but those are two different functions, we think, of the same transcription factor, to both promote and also inhibit calmodulin levels, depending on the levels of calmodulin in the animal.
All right. And then Josh Kaplan is asking, do you think that CAMTA explains the behavioral deficits caused by GCAMP or chameleon expression, kind of referring to anecdotal but reliable evidence?
Yeah. This is a good question, and we don't know. I think we have to really test this. I think what's interesting about this is the notion that perhaps this transcription factor can act as an integrator. I mean, obviously, when calcium comes in and calmodulin goes to all sorts of places, and so the levels of free calmodulin will change depending on neural activity. And you could imagine all sorts of interesting integration events happening to regulate calmodulin levels over the long term, based on levels of free calmodulin in neurons. But we don't know, the answer is we don't know.
Gotcha. Maybe just a couple more quick ones here. One question, is, does NLS, this is Zhao Zhuan, does NLS CAMTA rescue the CAMT1 mutants? In other words, is cytoplasmic CAMTA required for its function?
We don't know. I think we haven't done those experiments carefully enough to know, but it's a good question.
Great, and then maybe the last one, Rafa Yusta wrote in, exciting results, Mario. Do you know if CAMTA2 knockout affects the function of mammalian neurons?
So there's been one work looking at hippocampus where CAMTA1 does affect hippocampal function, but there was no evidence of CAMTA2 function in hippocampus at least. And the work that looked at it focused most, there are rather few papers on these transcription factors actually in any organism apart from plants. But what's published on CAMTA2 hasn't really found defects. But I think, one has to look at double mutants. I think there's lots of work to be done here.
All right, thank you for a fantastic talk again.
Thank you very much.
We'll move on to the last speaker of this session, who is Susan Demecki from Harvard Medical School. And the title of her talk is Embracing Differences, Heterogeneity in the Brain Serotonergic System.
Okay, good morning. Can you see my slides and hear me well?
Yep.
Okay, great. Okay, great. So thank you, Steve, for the introduction, for organizing this tremendous symposium, and especially for the invitation to participate. It's a real privilege to be able to share with the audience today, everyone here, some of the work going on in the lab. Okay, so keeping in with the theme of the symposium, internal states of the brain, such states tune behavior to causal environmental changes. This tuning of behavior is adaptive, beneficial to the organism, indeed even life-sustaining. And there's a growing body of evidence for the role of serotonin in modulating behavioral and physiological flexibility. So the focus of the presentation this morning will be on the work from my lab, where we're looking at the organization of the mammalian brain serotonergic system and on how we're identifying molecular and functional subsystems that comprise the brain serotonergic system. So first in the talk, I'm going to go through work that we've done on mapping developmental lineages of serotonin neurons. And on this sort of roadmap of the talk slide, I've put lineages in quotes because here we're really going to be fate mapping populations of neurons. And this is in distinction to the strict lineage definition in terms of clonal analysis of neurons, such as in C. elegans. And so in this first part of mapping developmental lineages, I'm going to briefly give some fundamentals around serotonergic neurons, very short anecdote about what sort of got us into a certain fate mapping question here, and then the enabling genetic platform that we're using. I'll step through how we're resolving subtypes of serotonin neurons using transcriptomics, so RNA-seq work, and then how we're delineating behaviors and physiological processes that are modulated by these specific serotonergic neuron subtypes in the mouse. So essentially asking how labor is divided across this system. So just very briefly, an introduction, serotonin neurons, the bodies of serotonin neurons reside in the brainstem in the serotonergic raphe. I'm showing a field of serotonin cell bodies here. Serotonergic neurons are relatively few in the brain, and just some numbers here, about 350,000 serotonin neurons in the human brain, contrasting that to the 100 billion neurons in the brain. So relatively few, yet their axons arborize extensively in terminal fields throughout the brain. And I hope you can see that here in this very classic schematic by Efrain Azmisha, showing the dense innervation in different brain regions. Now, commensurate with these extensive terminal fields throughout the brain, serotonin has been implicated in modulating numerous biological processes and implicated in human pathology, perhaps most famous for its role in modulation of mood and anxiety, but also involved in very fundamental aspects of regulating body temperature and breathing, so vital homeostatic processes. So our lab has been interested in trying to understand how the serotonergic neuronal system is organized in mammals. How is it organized to accomplish such a multiplicity of tasks? And part of our inspiration to start looking into this really came from looking at a different brainstem system, which is cranial motor neurons. And I'm just showing a cartoon here on the right. Each cranial motor nerve arises at a specific axial level of the brainstem and innervates distinct target muscles. And learning about cranial motor nuclei really led me to the beautiful work on hindbrain patterning of investigators like Andrew Lumsden, Roger Keynes, Rob Krumloff, William McGinnis, Sarah Guthrie, and their identification of the hindbrain as an axial segmented structure comprised of rhombomeres, each with a distinct transcription factor code, such as the Cox code shown here on the right. So that at each rhombomeric level, there are distinct differentiation programs enacted that lead to the development of specific cranial motor nerves that then innervate very distinct muscles and serve very distinct functions. So what about the serotonergic system, again, that arises from the hindbrain and is located in the brainstem? By looking developmentally at the origin of serotonin neurons, can it teach us something about functional subdivisions within the system? So we study this in the mouse, and I've just put a cartoon here on the right. The organization is very similar to the human brainstem, with a set of serotonin producing neurons clustered in nuclei in the brainstem. I'll use terms like dorsal raphe, median raphe, raphe magnus to describe these clusters or nuclei. And in some of the slides, there will be the B1 through B9 nomenclature of Dahlstrom and Fuchs, but please don't confuse that with the rhombomere origin that I'll be talking about, the R1 through R8 nomenclature. Now on this slide, I'm just showing you these beautiful serotonin neurons in sagittal view, dorsal raphe, median raphe in the medullary system. And if we take cross sections, again, I want to just show you a field of what the dorsal raphe looks like in coronal section, median raphe, moving rostral on the top to caudal on the bottom. In the medulla, we have the raphe magnus, pallidus obscurus. So this is the anatomy that you'll see in slides moving forward. So the genetic platform then that we're using to study the development and function of the serotonergic system is shown here. And central is a way that we're gaining genetic access to subsets of serotonin neurons. And I'll step through that in a minute. But the key thing is that we're gaining access to subsets of serotonin neurons, and then we go down one of each of these arms of the platform. If we go down the platform on the right, we can drive specific effector molecules like the DREADS or the light chain from tetanus toxin or channel rhodopsin in the specific subset of serotonin neuron, functionally manipulate those neurons and see if we can map specific functions to specific subsets. We can then go down the left arm of the pathway and platform and label cells and study their lineage, which I set the stage for, and I'll tell you more on in a minute. We can look at their transcriptome, we can do slice electrophysiology, begin to map circuitry, and then link really all of these cellular and molecular properties to the functional properties we've discerned. Okay, so how do we access the subsets of serotonin neurons genetically? Well, there are two kinds of genes that are expressed in serotonin neurons. So grossly speaking, we have one group that are expressed in all serotonin neurons, such as TPH2 and PET1. TPH2 encodes the rate-limiting enzyme in serotonin synthesis, as shown here. And PET1 is a transcription factor discovered by Evan Daneris back in 1999. It really is a master regulator of serotonergic differentiation and regulates expression of TPH2 as well as VMAT2 for loading serotonin in vesicles and for the reuptake transporter SERT. Oops. Okay, so in addition to genes expressed exclusively in serotonin neurons, but in all serotonin neurons, other genes are expressed in some serotonin neurons, but also in other regions of the brain. And it's really in this sweet spot that we want to be able to study and manipulate and map serotonin neurons. So to do that, we've generated a PET1 flip driver. So we're going to express flip recombinase in all serotonin neurons, and we iteratively use that with a number of different Cree drivers and a variety of intersectional reporters. And I'm showing that here on this next slide, where we generate triple transgenic mice. And then in the PET1 flip cells, I stop cassette is excised, shown here on these left transgene diagrams, so that mCherry is expressed, as shown here on the right, labeling dorsal raphe neurons. But then in the specific Cree driver that we use, we'll excise mCherry and turn on GFP, and then we can see that specific subset. We call that intersectional subset of serotonin neurons. And of course, in this strategy, one can express not just GFP in this intersectional position, but a variety of different reporters, as just listed here on the right, as well as effector molecules, allowing us to manipulate the activity of these cells. And again, many of these effector molecules generated by investigators at MIT. Okay, so with this platform then, we've been able to ask a number of questions. And really the first question that we asked was the one I set the stage for already, which is from where in the embryo do distinct serotonin neurons arise that are populating the adult serotonergic system. So as I alluded to earlier, serotonin neurons arise from the brainstem. They, here in the hindbrain, the progenitor cells sit ventrally here in this coronal cartoon of the hindbrain, and essentially derive from a progenitor zone shown here in gray that's subdivided by rhombomeres, and I've color-coded them here. So we can use rhombomer-specific Cree drivers. I've just listed some here on the right. Coupled with our PET1 flip driver, PET1 comes on early in development as serotonergic precursor cells take on their terminal cell division. So we can use rhombomer-specific Cree drivers along with our serotonergic-specific PET1 flip driver to fate map cells coming from distinct rhombomeres. Again, so we're going to use this intersectional strategy to turn on mCherry in serotonergic precursors, as I'm showing here on the right. We use our rhombomer-specific Cree drivers to delete mCherry and turn on GFP just in the rhombomer-specific rhombomer-derived serotonergic neurons, and track the whole system then into the adult. And I'm just summarizing now on this slide work done over 10 years ago, showing the results of that fate mapping. So we have very distinct distributions of serotonin neurons coming from a given rhombomeric origin, as well as nuclei that have an intermingling are comprised of multiple lineages. And I'm just showing that here, giving the example of the median raphe really is comprised of 3 different rhombomer-derived lineages. And you can see a convergence of lineages here on the right in the medulla. Okay, so we have this lineage map now, but we really wanted to know whether is this a good way to classify subtypes of serotonin neurons in the adult, or are, when the neurons actually get to their final location in the adult, perhaps they really all look the same molecularly and functionally. So we could ask that question then. Are mature 5-HT neurons the same transcriptomically despite their different origins, or do they retain their rhombomer identity? So Ben O'Cady took this work on really starting back in 2013. So using technologies from 2013, where he dissociated fluorescently labeled serotonin neurons from the adult mouse brainstem. He had these manually sorted pools of cells that then we sequenced and kept track really of lineage and anatomy as we went. And so now on this slide, I'm showing you the unsupervised hierarchical clustering dendrogram that resulted from that work. And if we overlay the lineage tracking information, what we indeed found was in the adult, the transcriptome groupings align with the developmental rhombomeric lineage. And I'll show that again, just highlighting this median raphe structure that's comprised of 3 lineages. And indeed, if we look at the transcriptome dendrogram on the left, these yellow R3-derived serotonin neurons are clustering separately from the R2-derived serotonin neurons in purple here, while they're more similar transcriptomically to the R1-derived median raphe serotonin neuron. So even though these 3 lineages are sitting in the same anatomical location, they have a distinct molecular profile of gene expression. So their lineage of origin and maintaining molecular identity is maintained in the adult. Now, it's not just rhombomereic origin. Anatomy is also playing a role. If we look at the rhombomere that gives rise to these blue serotonin neurons, they're spread across the dorsal raphe and median raphe, and the ones in the median raphe transcriptomically here on the left look more like the other R2 median raphe cells. So the anatomical location is giving cues too, and we think this combination is really shaping mature molecular identity. Okay, so now what's different transcriptomically between these gross classifications of serotonin neurons? Okay, hundreds of differentially expressed transcripts across these subtypes. So I'm just showing this in replicate of biological samples here, and on this heat map of different genes expressed, you can see a very different pattern with each of these clusters. And I'll just quickly just make the point that we see differences in cell adhesion genes, transcriptional regulators, G protein coupled receptors, synaptic transmission, and ion channel genes. So really from here, we have this combination of lineage and anatomical location that we believe is providing a mechanistically relevant definition of serotonin neuron subtypes. And these subtypes differ across numerous categories. So next then, we wanted to look at, well, how much variation really is there within these subtypes? And we went on to single cell RNA-seq work. And initially this work involved really, again, back in 2015, technologies, low throughput single cell profiling, fluorescently labeled cells, keeping track of lineage, isolating single cells. And importantly here, what Ben O'Cady, spearheading this work, found was that we had a very similar clustering structure, again by rhombomereic lineage. So both single cell and the pooled cells revealed this rhombomereic structure. And even with the low cell numbers here for this initial single cell RNA-seq experiment, substructure began to emerge within these clusters. So we could see subclusters here in rhombomere 2 derived cells, for example, and in rhombomere 1 derived cells and elsewhere. Okay, so summarizing the 2015 transcriptomics, we have this rhombomere lineage-based classification scheme. It's robust across scales, whether single cell or pooled cells. There's variation at the single cell level that suggests that we have some of these subdivisions within these lineage clusters, and that these subdivisions are really bounded by the rhombomere of origin. And this data then really prompted us to want to pursue high throughput single cell RNA-seq work. And we've started with the dorsal raphe, the largest structure in the raphe system, harboring more than 50% of the serotonin neurons in the mammalian brain. And so here, Ben O'Cady, along with Nikita Sturak in the lab, took a high throughput approach. They isolated fluorescently labeled serotonin neurons dissected from the raphe. They dissociated and sorted these cells using a microfluidic sorting, using this on-chip sort, and then performed a droplet-based RNA-seq library prep using 10x genomics chromium single-cell protocols, ultimately sequencing over 2,000 single-cell libraries with roughly 7,500 genes per cell. And the results are shown here. So on the left, I'm just showing you an unsupervised hierarchical clustering dendrogram of the predicted 14 PET1 neuron subtypes in the dorsal raphe. On the right, I'm showing the UMAP visualization of these single cell transcriptomics. And in first sort of analyzing the structure what Ben and Nikita realized was that characterizing the structure using 5 particular genes really helped frame how the system was organized. And I've just listed those 5 genes here, showing the violin plot of their expression levels across these 14 different subtypes. So as I mentioned earlier, TPH2 is the rate limiting enzyme in serotonin synthesis, and not surprising, it's expressed at high levels across most of the subtypes of clusters here of serotonin neurons. GAD1 and GAD2, these are genes that encode for GABA synthetic enzymes. Then VGLUT3. VGLUT3 is a vesicular glutamate transporter that packages glutamate into synaptic vesicles, and MET, or receptor tyrosine kinase. So these were very helpful at defining the gross structure, cluster structure of the dorsal raphe. And I'll just briefly go through this. This cluster 1, high levels of TPH2, and it was really a principally serotonergic cell type. Clusters 2 through 6, so 2, 3, 4 over here on the right on the UMAP plot, 5 and 6, expressed high levels of GAD2 and GAD1. So potentially both GABAergic as well as serotonergic. And this is really about 50% of the cells in the dorsal raphe. Then we have clusters 7 through 11. So this group here, the blue purplish clusters on the right in the UMAP plot, express high levels of VGLUT3, so serotonergic and glutamatergic. And then we have a cluster that serotonergic, glutamatergic, and uniquely expressing this receptor tyrosine kinase MET. And then a group that has sort of variable levels of TPH2 and VGLUT3. So this is a gross sort of structure looking at some other neurotransmitter synthetic genes. And this cluster structure at this gross level aligns with work of others. So recent work by Kiwi Huang in Bernardo Sabatini's lab and Jing Ren in Li-Chin Liu's lab. The ability to resolve this to 14 clusters here largely stems from sequencing more cells as well as read depth. Now here I'm just showing you a heat map of the 5 top expressed genes for each of these 14 clusters. And just to make the point again that these enriched genes span multiple categories of molecular and biological functions, and that this supports then the idea that these 14 dorsal raphe subtypes reflect functionally meaningful diversity. Now, this approach that we used here of dissecting, pooling, sorting, and sequencing, we lose all anatomy in this. So how can we actually link these 14 subtypes to the anatomical structure of the dorsal raphe? And we could do that by using these gene expression information to identify Cre drivers and then use them in our intersectional genetic platform and actually label these cells and look at the anatomy. And I'm just going to quickly step through a few examples here. And if I pull up our violin plot and UMAP plot again to help step through this top row. So here we're using a VGLUT3 Cre driver with our PET1 flip. So this is going to label clusters 7 through 14, so cells here. And anatomically, we're finding them in the ventral medial part of the dorsal raphe as we move rostral to caudal in this top row, and they move more dorsal here as we end up more caudal. Okay, so we then by subtraction, the more GABAergic clusters of serotonin neurons are sitting more dorsal medial and then in these lateral wings. If we look at another gene that's expressed in clusters 6, 7, and 8, we can define the subtype expression of a receptor for neuropeptide Y. And indeed, we see that they're a subset within this VGLUT3 medial domain. And then an example of a very specific clustered subtype is defined by expression of a purinergic receptor P2RY1 and is tightly clustered in the very caudal part of the dorsal raphe. Ben and Nikita went on to manually sort each of these different molecularly defined subtypes by single-cell RNA-seq and mapped them back onto the 10x genomics data, again, confirming their transcript identity. And that led them to this summary of the 14 subtypes and where they anatomically distribute and are quite intermingled within the dorsal raphe. I'll just put a quick plug in. Ben and Nikita have also put together an interactive companion web app, which you can link to from our lab website or from the eLife paper in which this is published. And you can put in your genes of interest and really search for their differential expression across the subtypes in the dorsal raphe. Okay, so we have 22 now subtypes of serotonin neurons in the mouse brainstem, 14 identified in the dorsal raphe and 8 in the medullary and median raphe. We have this emerging transcription factor code related to each subtype. Neurotransmitter co-expression may be the norm. The subtypes are intermingled as shown here, and we can use Cree and flip lines to begin to study their function. So we can use our platform here then to now begin to manipulate these neurons. And I just want to very quickly in the last few minutes share 2 vignettes of subtype specific serotonergic neurons in the dorsal raphe. And the first is this subtype here in the caudal dorsal raphe that we define by expression of this purinergic receptor. And it has a really unique profile. So it's this population here, population 12, this island, very unique profile and shown in these violin plots of GPCRs like the mu opioid receptor, the relaxin receptor, and absent of expression of other GPCRs that are expressed throughout the serotonin system. So very unique profile of expressed genes. Electrophysiologically, much less excitable in orange here in this current injection versus spike frequency plot. And you can see it takes much more injection of significant current, substantial current to get these neurons to fire compared to the typical dorsal raphe serotonergic neuron. And when they do fire, we have this very long latency to their first action potential. So electrophysiologically, they're as well quite different than other serotonin neurons. And what I'm showing in this brain cartoon here is that they actually, if this orange blob here is to be the dorsal raphe and this intense orange here, this caudal portion, these neurons innervate the ventricular system and rostral migratory stream. So in gray here on this slide, I'm showing you the brain ventricular cerebrospinal fluid system, and these cells innervate the walls of the lateral ventricle. And I'm showing you that here in a flat mount of the lateral ventricle. And I hope you can see these intense serotonin fibers coming from this population of caudal dorsal raphe neurons. These neurons also innervate this rostral migratory stream in the subventricular zone, so this neurogenic region. And so you can see these fibers here really wrapping around in apposition to neuroblasts in the rostral migratory stream. So we have a subtype of serotonin neuron that is involved in regulating the ventricular cerebral spinal fluid system and this, the neurogenic subventricular zone. And very quickly, I just want to touch on one other subtype. I realize I'm tight on time here, the DRD2 PET1 subtype that sits here in the lateral wings of the dorsal raphe, cartooned here in the middle, and just showing you that image here. So these are cells that express the type 2 dopamine receptor, indeed the principal sole dopamine receptor expressed by serotonin neurons in the adult. When we track their innervation using synaptophysin GFP in our intersectional strategy, I show one example here and summarize that data here. Intense innervation of some descending structures from this region, so cochlear nucleus superior olive, rostral periolivary region, lateral lemniscus, inferior colliculus, so auditory processing centers, and then as well the VTA, lateral hypothalamus, medial preoptic area, and septum. So areas involved in exploration, spatial learning, appetitive reward centers. So then we took these mice and subjected these mice to a broad behavioral screen when we manipulated neuron activity by expressing the light chain of tetanus toxins. So we're silencing them or blocking neurotransmitter release selectively from these neurons. And we found that they had increased exploration, persistent active coping, so less time immobile in a forced swim test, and some aspects of impaired spatial learning and impulsivity, and an increase in aggressive behaviors in a resident intruder assay. So biasing their behaviors to exploration, approach, offensive, attack, active coping if we silence these neurons. Next, Chrissy Lyon in the lab asked, what is the phenotype if we actually delete this receptor, this inhibitory receptor? And she found two really interesting phenotypes, and I'll quickly mention them. One is in this acoustic startle protective defensive reflex. Specifically, female mice had a significant blunting of the startle response to a wide range of auditory stimulus intensities, where we saw no effect in males. And this is somewhat consistent, at least the female response, with this intense innervation in the auditory centers, and in particular, the caudal pontine reticular formation that's critical for this defensive startle reflex. In males, she found a phenotype of altered social dominance. So in a social dominance tube test assay, she found that mice where we had deleted the DRD2 receptor, they backed out of this confrontation assay less, which is an expression of social dominance. Okay, so we have these phenotypes that involve shaping social and sensory alertness, protective defensive behaviors around this dopamine responsive serotonin neuron. Again, so functionally distinct subsystems that I've shown here. I'll just quickly state, but not show any of the data. We've mapped features to this median RAFA population that are involved in facilitating extinction learning, so unlearning conditioned behaviors such as cocaine conditioned place preference. These cells are also involved in sensory motor gating. And then down here in the medulla, we've mapped specific functions, the EGR2, the green serotonin neurons here in this image. These EGR2 PET1 neurons are intrinsically chemosensitive. So they are reminding me of the BAG neurons in C. elegans that we just heard about. These neurons increase their firing in response to elevated conditions of elevated CO2 and a decrease in pH, so conditions of hypercapnic acidosis, and are critical for actually increasing respiratory drive under such life-threatening conditions. These red neurons, these TAC1, PET1 neurons here in the medulla also increase respiratory drive, but they do it through motor output, motor gain. So we really have a sensory motor divide in this caudal aspect of the serotonin system. So here I've given you a flavor of just a few of the functionally distinct serotonin systems. Let me summarize now what I've shown you today. I've shown how we've used transcriptomic RNA-seq as well as fate mapping analysis to reveal up to 22 different subtypes of serotonin neurons in the mouse brain, with the first level of organization really deriving from rhombomereic lineage, and that this is providing a mechanistically relevant way of thinking about serotonin neurons, that there's substructure within the rhombomer-derived lineages, but largely bounded by these lineages. We have distinct profiles of many different gene categories across these subtypes. That neurotransmitter co-expression seems to be the norm. So these neurons are not just serotonergic, but also glutamatergic or also potentially GABAergic, as well as expressing a range of different neuropeptides. A structure function map is beginning to emerge. This is really a work from many in the field, of which our lab is just one small piece of the contribution. But collectively, the data is really suggesting that the serotonergic system is really a conglomeration of functional subsystems that are deployed to modulate different brain regions and behaviors. So for example, to tune alertness to environmental context, to speed unlearning, to avoid perseverative actions, for example, or to match breathing to physiological demands. And then importantly, these subsystems appear to be separable pharmacologically. And so our hope is that would mean also possibly therapeutically. Okay, and I'll just close finally and acknowledging the folks in my lab who have done this work, and it's really a privilege. I've had such a privilege to work with everyone in the lab. I'm showing you, I believe I mentioned most of their names throughout the talk. We've had an enormous set of collaborators in all of this work, and then many, many people, lab alumni on this serotonin project. Thank you, and I'd be happy to take questions.
All right, thanks for a wonderful talk. There's a bunch of questions coming in. So first question from Richard Bourne. If you could visualize the axonal arbor of a single cortically projecting serotonin neuron, over how large a region would its arbor ramify? Is there any regional selectivity, or are single axonal arbors promiscuous?
So here the question is if we could look at a single, for example, dorsal raphe neuron projecting to the cortex, how much of the cortical field will it cover? How extensive is that arborization? How extensive are the collaterals? And I have to say, we have not actually looked yet at individual neurons. We've looked at very small populations, and within those small populations, it's remarkable the extent of collateralization that we do see, innervation of, on the order of 10 different regions of the brain. So we think extensive collateralization and arborization for even individual neurons. Now in Jing Ren's work in Li-Chin Lu's lab recently, they have published some really extraordinary images where they have been able to label individual cells. And you really do see this extensive collateralization and then a single fiber arriving at a particular cortical area and then really arborizing within that local area. So I think there's a lot more to be done, and it's something that we're really interested in and comparing across the different subtypes of serotonin neurons that we've identified. Thank you.
All right, very cool. And then another question from Mark Anderman. Beautiful talk. You emphasize diversity of serotonin subtypes. Could you please discuss signals that might regulate all serotonin neurons in a coherent manner, for example, during certain forms of arousal?
So maybe one example I could point out. So in one of the heat maps that I showed, violin plots, you could see that orexin receptors, histamine receptors were really a set of GPCRs that are expressed at varying levels, but expressed in almost every subtype of serotonin neuron. The exception being that cluster 12, that P2R-Y1 defined PET1 neuron population. So that was unique in that it was sort of protected or sequestered and did not respond to secreted molecules like orexin or histamine. But those would be two examples of ligands where we see receptor expression really throughout most subtypes of serotonin neurons.
All right. And let's see, a few more questions here. Stephanie Rudolph asked, some of your early slides showed serotonin axons that were localized to the surface of the brain or brainstem. Any idea when and where these neurons originate, what subtype they are, what their functions might be?
Okay, so the question relates to serotonin neurons, and if I'm understanding the question, that extend fibers to the surface, the ventral surface of the brainstem. And we think that many of those serotonin neurons in the medulla, as the example, come from that EGR2 PET1 population that is chemosensitive, and those neurons are sending fibers to other chemosensory centers in the brain stem, such as the RTN nucleus. They're innervating more dorsally locus ceruleus. They're innervating the Pre-Botzinger complex, as well as even showing some apposition around ventral surface vasculature. So these are the neurons that are responding by increasing their firing rate to conditions of hypercapnic acidosis and are important for mediating an increase in respiratory drive under those conditions. So I think that's an example of one such population that's having fibers to more ventral nuclei in the brainstem or ventral surface of the brainstem.
All right. And then let's see here. So Christina Wright is asking, it seems like the P2RY1 PET, in other words, cluster 12 island, is well positioned to interact with many brain areas given its access to the ventricular system. Do you have thoughts about this subsystem maybe setting emotional tone or backdrop for behavior? What types of behavioral assays have you run with this system in mind?
Okay, so that's a great question, because here we have a population of serotonin neurons that may be able to influence the ventricular cerebral spinal fluid system. And we know from some beautiful work from Maria Leitonen's lab, along with Mark Anderman, showing that the 5-HT2C receptor is expressed on the choroid plexus epithelium, and serotonin, and triggering that receptor can lead to the release by the choroid plexus epithelium of various content into the CSF. So we have this population of P2RY1 PET1 cells that may both be releasing serotonin themselves into the ventricle system, but may also be, could potentially be manipulating release by the choroid plexus epithelium and changing content of the cerebral spinal fluid, which would then have a much broader capacity to influence cells in the brain. So it's a really good question in terms of understanding sort of what is the behavioral and state different consequences of triggering these P2RY1 cells. And Nikita Sturrock on the lab is working very hard to really address that question. I'll tell you, technically, we've had to have an additional layer of technology to try to address that question for the following reason. So usually in our intersectional strategy, by combining PET1 FLIPI with a Cree driver, that intersection gets us very exclusively to a brain subset of neurons. But in this case, PET1 is actually expressed in the beta cells of the pancreas, and in those same cells is expressed P2RY1. So if we use our dual recombinase strategy to activate or inhibit these cells, we're not just manipulating these caudal dorsal raphe cells, but we're manipulating beta cells in the pancreas. So we have elicited some phenotypes, but right now we haven't been able to distinguish those that are derived from the brain modulation by these P2RY1 cells versus pancreatic beta cell function. So the way we're getting around that is to essentially deliver Cree via injecting a Cree virus into the ventricles, which is taken up and beautifully labels this population of cells, these P2RY1 PET cells in the brain, and then repeating those behavioral and functional assays. So we don't know the answer to that yet, but it's a very exciting question. Thank you.
All right, very cool. And then maybe the last question from Sishrati Ramaswamy. What's the role of serotonergic neurons in the gut? I think referring to kind of the enteric nervous system and other serotonin classes in the periphery.
So talking about release of serotonin by cells in the gut and their role in gut motility. So that's an area that's being actively pursued by many, and I think there's going to be a very exciting connection between serotonin release in the gut, as well as its role in the vagal nerve and influencing brainstem centers through the vagus and the nucleus of the solitary tract in the brainstem. So I don't have answers there for you other than sort of the very broad ones around serotonin and gut motility, but it's a really, again, exciting direction that other labs are taking. Thank you.
All right, fantastic. So I'd like to thank all three of the speakers from this first session. And we're going to take a break now. We will resume with the second session at 11:20 Eastern time. So about a half-hour break, 25 minutes or so. All right.
Hi, everyone. Welcome back. I'm Li Wei Tai, and I'm going to moderate this session after the break. And I'm very pleased to welcome Mala Murphy, who is from Princeton University. And she will tell us about adjusting behavior on the fly, internal states, and dynamic social interactions. Mala, please.
Hi, Li Wei, thank you. And I want to thank the organizers for inviting me. Can you hear me okay?
Yes.
Okay, and then let me share my screen. Hopefully you can see that. Okay. Great, all right, well, thanks again for including me. It's a stellar list of speakers today, and I'm excited about this symposium. Okay, so my lab, as many of you know, is interested in neural mechanisms of communication. And in putting this talk together, it wasn't lost on me how ironic it is to talk about communication in this virtual seminar where we can't directly communicate with each other. And in fact, over the course of the pandemic, I have thought a lot about how aspects of communication we take for granted and that my lab studies in flies are in fact now quite rare. So I just wanted to say that I certainly look forward to when we can gather in person again. I hope you're all doing well, hanging in there, and finding ways to still communicate. So the aspects of communication that interest my lab the most are how social feedback cues are used in real time to shape ongoing behavior, as happens when we have a conversation. So when I'm talking to somebody, for example, their gestures or their facial expressions affect what I say in real time, and my speech in turn affects their behavior. And we want to understand how that happens in the nervous system. And the approach we've taken to solving these problems in my lab is really a neuroethological one. It's one pioneered by neuroscientists studying acoustic communication for some time now in a variety of really powerful model systems. We've, of course, applied this approach to a genetic model system, Drosophila, that has a much smaller brain, really unparalleled genetic and neural circuit tools. And I wouldn't be telling you about all of this if flies were not capable of acoustic communication. Males produce songs by extending and vibrating a wing, and they direct this information at the female. Songs carry sexual and social information, and as I'll tell you in a minute, my lab has shown that acoustic communication in flies is quite dynamic and is shaped by ongoing feedback. Sorry. Okay. So over many years now, what we've shown is that cues from the female, like how far away she is and how fast she's moving, shape what the male sings. And he processes this information from her using his visual system and by reading out his own self-motion. And that information affects the song choices he makes. So he can choose between these modes, the one shown in orange, red, or blue. He also patterns how loudly he sings relative to the female. She listens to the song, she adjusts her pattern of locomotion in response, and her behavior then in turn affects the male. So there's this nice feedback loop between them. And we've used a mix of quantitative behavioral measurements, computational modeling, neural recordings, neural manipulations in my lab to address some of the mechanisms underlying this behavior. So for example, we've used generalized linear models to predict song behavior, and song bouts are basically alternations between signs and pulses. And the models revealed that these two parameters in particular, how close the male is to the female and how fast he's moving, can predict song mode choices. So when he's close to the female, he tends to bias towards starting song bouts in sign mode. And when he's when he's moving fast, he tends to bias towards starting song bouts in pulse mode. And the models reveal the timescales over which these features, these feedback cues, are predictive. So together, this work has established the system, I think, as one that's really powerful for understanding sensory motor transformation, how sensory cues affect ongoing behaviors. And over the last few years, we've become quite interested in how the internal states of the brain affect the sensory motor transformation. And that's, of course, what I'm going to focus on today. So there's two things that I've been thinking about with regards to internal states, and that I find quite interesting. That is that their timescales and how obvious they may be from outward behavior. So I just want to give you two examples to drive this home. Sorry. Okay. So imagine that we were gathered together and I was at the lectern and you were listening to me in person. Your internal state as an audience member might be changing over rapid timescales, fluctuating in and out of attention, but it wouldn't be obvious to an outward observer necessarily how your internal state was changing. On the other hand, sometimes our internal states can change on slower timescales, from a pensive, restive mood to a very overtly excited one. And we usually don't switch back and forth between these states so quickly. And it's often obvious from our outward behavior what state we're in. And these differences have been influencing the way I've been thinking about internal state. And with that in mind, I want to address three questions in my talk today. So the first is, how do we identify changing internal states of the brain? How do internal states shape animal communication? And what neural mechanisms underlie internal states? I'll first briefly tell you about some work from my lab that was published last year, in which we developed new models to identify dynamically changing internal states. And this is the work of a super talented postdoc in my lab, Adam Calhoun. I'll then focus on a newer story on persistent neural activity and internal states. This work appeared on bioRxiv earlier this year, but we've added some new data, and so I'll tell you about the latest. This is the work of another talented postdoc in my lab, Judy Deutsch, and I just want to mention that both postdocs are on the job market this year, so I would highly recommend snatching them up. So this is 30 minutes of courtship. Males sing a lot during courtship. And at the bottom left here is the history of a male chasing a female over a short period of time. You can see how they interact. At the right is a single song bout. So over time, he's composing his song into bouts. And he can choose between fast pulses, slow pulses, or sine mode. I'll play you a little bit of the song so you can get a sense for what it sounds like. Okay, so hopefully you could hear the purring and the hum. As I mentioned already, my lab has been using models like GLMs to predict song from fly movements and interactions. And when Adam began this work, he was interested in applying this modeling strategy in a generative format. So trying to predict the moment-to-moment changes in song, over time. So we knew on average that these feedback cues were predictive of song mode choices, but we didn't know how predictive they were on a moment-to-moment basis. And so what Adam found, looking over large data sets of courtship interactions, is that sometimes the GLM gets it right. For example, when the male is singing sine song, the GLM predicts sine mode. But there are many instances where the GLM gets it wrong. And you can see that here where the predictions are off from what's actually happening. And so if we look over all time, the GLM certainly is more predictive than a chance model, but it only captures about 14% of the remaining information beyond what a chance model can do. And so that prompted Adam to think about other models that might be more predictive of the behavior. And so Adam decided to collaborate with my colleague, Jonathan Pillow, and inspired by really nice work from Sean Escola, Adam built this GLM HMM, which incorporated states, internal hidden states in the model, in addition to these feedback cues. And so what Adam found was really quite remarkable. He could predict the moment-to-moment changes in song patterning with very high accuracy. And so you can see now that the GLM HMM performs much better. It predicts sign mode when it happens precisely. It can predict switches between these modes when they happen, and is even good at predicting the difference between slow and fast pulses, which is quite challenging given how similar these modes of song are. And so now when we look at performance, a 3-state GLM HMM is a major improvement over a chance model or the GLM alone. It now accounts for about 84% of the remaining information. So it's not perfect, but it's approaching that level. So what are the 3 states of the model? Adam actually named them close, chasing, and whatever. And they basically describe the context the male is in. So in the close state, the male tends to be close to the female and moving slowly. In the chasing state, he tends to be close to the female and moving fast. And in the whatever state, he tends to be far from the female and often angled away from her. Now, there are different song modes that tend to occur in these states. So he mostly sings sign song in the close state, but there's not a one-to-one mapping. You can see that other song modes are still produced in the close state, and that's true across states. In addition, these states are not overt action phenotypes. That is, even though males tend to be closer to females in the close and chasing states, they still are close to females in the whatever state, and these distributions overlap quite a bit. Adam showed that the state residencies are longer than the durations of individual modes of song, again, suggesting they're not a one-to-one mapping with song mode, and that males continually switch through these states throughout courtship. So if you look over courtship time, there's a steady distribution of these states. Here you can see for a little stretch of song, the predictions from the model. So the model predicts that he's continually switching between these states. You can see how good the performance of the 3-state GLM HMM is. And if we only use GLM filters from just the close state, just the chasing state, or just the whatever state, you can see that the model doesn't do as well. You really need the filters from all 3 states to generate the actual song pattern. And so what this tells us is that the states are really about responding to sensory cues or feedback cues in different ways at different times. And to really drive that point home, if you look outside the model, so if you just plot the relationship between these feedback cues and song, you can see these differences. So for example, for male velocity and the probability of singing pulse song, you can see that as males move faster, they're more likely to sing pulses in the close state. But in the chasing state, the faster they move, the less likely they are to sing pulses. And the same is true for male-female distance. The further the male gets from the female in the close state, the more likely he is to sing pulses. But the further he gets from her in the chasing state, the less likely he is to sing pulses. All right, so with this model in hand, we could now ask, what neurons actually control state? That's something, those of us interested in internal states really want to understand. And we were aided in Drosophila by driver lines that could allow us to activate or silence individual neurons. And Adam had the idea to target neurons that drive singing. So driver lines for these neurons had been identified from really beautiful work from Barry Dixon and also David Stern. This particular image is from David Stern's lab. Lines like this, when optogenetically activated, can produce song in males. And the idea was neurons that are involved in state have to be linked to motor behaviors ultimately. And if you don't know anything about the states and you activate these neurons, you'll drive the behavior. But now with this model, we could uncover a relationship with these states. And so that's what Adam did. And just to summarize a bunch of data, he found that when he intermittently activates this particular neuron, PIP10, he can induce singing in males. You see that here. Males are more likely to sing fast pulses when the optogenetic stimulus is on. But using the model, he could show that activation of this neuron in particular and not other song pathway neurons pushes males into the closed state. So he's more likely to use the set of sensory motor transformation filters that are aligned with the closed state. And so, I just want to emphasize that this kind of discovery would not have been possible without the model. And we are interested if this applies more generally. Often we think of neurons, higher order neurons in the brain detached from motor behaviors that control state. Perhaps we should be looking closer to areas of motor control. So what we're doing with these results now is we have a way now to assess state by activating the PIP10 neuron, imaging from neural activity panneuronally in males that are walking on a treadmill, viewing movies on a screen that mimic female motion, and vibrating their wings to produce song. And so we're hoping in this fictive courtship setup, and by activating the PIP10 neuron, we can start to uncover the neural mechanisms that underlie state. And this is ongoing work I'm quite excited about. Okay, so there we go. All right, so oops, let's go back. All right, so I wanted to go back to the slide and think again about timescales of internal states and how visible they might be from outward behavior. And I'm going to switch gears now and talk about female behavior. And I'm going to tell you about some new work about internal state in the female that changes on much longer timescales of minutes. So before I do that, I need to give you a little bit of background on sexual dimorphisms in the Drosophila brain. They're controlled by two transcription factors, double sex and fruitless. There are different isoforms in males and females, and they control all of the sexual dimorphisms in neuron number and neuron morphology between male and female brains. So if you look at neurons that express double sex, there are different numbers in males and females, and they are identifiable, so they have names. And in this figure from one of Stephen Goodwin's papers, you can see the different subsets that express double sex in the male and female brain. This subset, PC1, in very nice work from Bruce Baker's lab, if it's silenced, females are less receptive. So they're less receptive to males when these specific neurons are silenced. And that was interesting to us because it suggested that PC1 neurons might be involved in controlling how females respond to sensory cues in particular from a male. Now, males, as I said, also have PC1 neurons, and they have a male-specific subset called P1. In really beautiful work from David Anderson's lab that we might hear about soon, they've shown that activating P1 neurons produces persistent effects on behavior. So it can drive, for several minutes following activation, either courtship-like behavior in males or aggressive behavior, depending on the sensory context. And David's work has shown that there's a circuit downstream of P1 that seems to be very important for this persistence. Okay, so given this work, we decided to test something similar in females. And Judy created a protocol in which we activated PC1 neurons in females, followed by a variable delay period before we introduced a male. And the idea was that we would drive these neurons into a persistent state, and then we would allow this activity to decay over variable periods of time and examine the persistent effects on behavior. So when we introduce a male, we can assay behavior and quantify it in the context of courtship. So what we found is that these females are hyper receptive. So they mate very quickly with males once introduced. And this effect is strongest at the 0-minute delay and decays with longer delays. And we also found that females accelerate more in response to male song. So normally females slow down in response to song. The more song, the more they slow. And in this case, they're speeding up more, okay, which is not a receptive behavior. So these two behaviors may be, you know, it's not totally intuitive why they're coupled, but both are heightened at the 0-minute delay. When Duty went on to analyze the behavior in more detail, so he used these parameters that describe interactions between male and female to use unsupervised methods to then cluster behavior. He found there were two behaviors that were really elevated in females, what he calls shoving, where females push males, and chasing, where females stand behind males and sort of seem to court males. And both of these behaviors were actually elevated with a 3-minute delay. And I'll show you that here. So you can see that at the 0-minute delay, there's persistent shoving and chasing, but the amount of this behavior really elevates following a delay period before males are introduced. So just to sum up these behavioral effects, what we find is that with a 0-minute delay, there's fast copulation and females tend to run away from male song. So those behaviors are heightened. And then if we wait a little bit longer before we introduce males, we see that the behaviors that are heightened are female shoving and behavior that looks like courtship, females chasing males, okay? So this suggests to us that PC1 may be inducing a kind of arousal state in females. And I'll just play you a movie of this interaction. So the male is painted with a little white dot. You can see the female is shoving him. Then she'll chase him. It looks like she's singing to him, but she doesn't produce any audible sounds with wing vibration, which we can tell because they're recording on this or interacting on this array of microphones. And then the male will turn right around and quickly copulate with her in this particular example. Okay, so we were interested in the underlying mechanisms, and I, over the past couple of years, have been involved in a really wonderful collaboration with Sebastian Sung here at Princeton to generate a whole brain connectome for Drosophila. So the data set that we've been using comes from beautiful work from Janelia, from Davi Bach's lab, an entire EM volume of an adult female brain. And using AI methods developed in the Sung lab, we've been able to align this volume and automatically segment all of the neurons in this EM volume. So using this resource called Flywire, and I should mention it's completely open to the community. If you're interested in using this resource, please go to the website and sign up. We identified PC1 neurons in the volume, and they come in 5 flavors, I guess. A, B, C, D, and E. These cell types were actually named at Janelia. I should point you to the fact that both Barry Dixon and Jerry Rubin's lab have also identified these PC1 neurons in EM data sets, and they were the ones who named these cell types. Okay, so now having identified the PC1 cell types, oops, let's go this way. We could find driver lines that targeted specific cell types. And so we found one that targets the A and B cell types, and a different one that targets the D and E cell types. This driver line we call PC1A. And in fact, work again from Barry Dixon and Jerry Rubin shows that these 2 cell types, D and E, are female specific neurons. So they're PC1 subtypes that are in females, not in males. Okay, so now if we activate these DE neurons specifically and we repeat this protocol, what happens? Well, we still see persistent chasing and shoving, suggesting that it's these 2 cell types that are important for these persistent effects on behavior. I'm going to show you the data for shoving here. You can see it's elevated following 5 minutes of activation. And if we shorten the amount of optoactivation, so if we reduce that time period, we still see persistent effects on shoving, but they're somewhat reduced compared to 5 minutes of activation. And so this suggests to us that there may be some scalability of this behavior. I'll tell you that other PC1 neurons, we still have to identify specifically which ones drive the persistent effects on receptivity. So it's not these DE neurons, but there's a very interesting interaction. So females that are receptive to males, that mate quickly with males, tend to chase males less, but shove males more. And so we're very interested in how these PC1 cell types modulate each other. Okay, so now that we had mapped the persistent effects on shoving and chasing to the DE neurons, we could go back to Flywire and start to examine the connectome. And so here I'm showing you a PC1D neuron and all of the input and output synapses in red and green. And I just want to mention that Janelia has created a really beautiful annotated connectome resource called the HemiBrain. The HemiBrain volume is shown here in red. It's a portion of the whole brain, but you can see that these neurons, PC1D, lie mostly within this volume. There's a few synapses that leave the volume, but it affords us the opportunity to actually compare connectivity results in 2 EM volumes. Okay, so from Flywire, these are all of the top inputs and outputs for PC1D and E. There's a lot of text on this slide. You don't need to pay attention to all this. In light blue here, I've indicated the HemiBrain neurons that are most similar to the ones we find in Flywire. But I want to point out something in particular, which is that 3 of the top inputs for PC1D and E are all a single cell type called AIPG. It comes in 3 varieties, A, B, and C. And this particular type of neuron, AIPG, is also one of the top inputs for PC1D and E. So there's a reciprocal connection between these neurons, forming a recurrent neural network. And I should mention that work from Jerry Rubin's lab has shown that both PC1 and AIPG are excitatory cell types. Okay, so what are AIPG? They are fruitless positive neurons, first identified in this study. Here they are in Flywire, and here I'm showing you the number of back and forth connections between the AIPG cell types and PC1D. This recent paper from Jerry Rubin, in fact, identified drivers for AIPGB, and their work has nicely shown that when you activate this cell type, you drive female aggression. And in fact, if you notice in this data, there's a bit of persistent aggression beyond the optogenetic activation period. And so we think the model that's most consistent with our data is that strong activation of PC1D and E recruits AIPGB to drive persistent aggression, for example. Okay, so we wanted to get at the mechanisms, and to do that, Duty collaborated with a graduate student in my lab, Diego Pacheco, who had developed methods for whole brain imaging and registration to catalog sensory activity or neural activity throughout the entire brain. And so these methods allow us to image from the brain in volumes, stitch those volumes back together again, register activity to an in vivo atlas, and automatically segment ROIs that are active. So this method then allows us to link an in vivo atlas that Diego developed to a fixed brain atlas, of which there are many for Drosophila, and then to bring into our in vivo atlas all of the maps of neuropils and axon tracks. These resources were developed by Greg Jeffries and collaborators and colleagues, and are a wonderful resource in Drosophila to be able to map neural activity. So here I'm showing you data from 2 volumes imaged in the Drosophila brain, and you can see what automated ROI segmentation looks like. These are the centers of ROIs that are active in each of these volumes. And this is Diego's data showing that you can cluster these ROIs and identify those ROIs that have stimulus modulated responses. Here I'm showing you data in response to auditory stimuli, but we can do the same for optogenetic stimuli. So doing that, and activating PC1-DE neurons, Diego and Duty find that while much of the activity is highest during the stimulus, there is persistent activity that persists beyond the stimulation period, as you can see here in these two clusters. And so if we look at where that activity is, you can see first that it's strongest when PC1-DE neurons specifically are activated, and it's in the central part of the brain. There are two different types of persistent responses we see, some that decay very quickly and others that decay to a steady state and stay high. And we think this might correspond to the different effects we see on behavior. I'd be happy to come back to that. So to summarize our results, we find that this persistent activity decays over time. It scales with activation duration. It overlaps with the processes of double sex and Fru positive neurons. And if we image just from these double sex and fruitless positive neurons, we find persistent activity specifically in neural clusters, in the cell bodies of neurons, where PC1 and AIPG are. And I'll show you some of that data. So here we're imaging only double sex neurons. We've focused our ROI analysis only on PC1 somas, and we find strong persistent activity that outlasts the stimulus for minutes. And so this nicely links minutes-long persistent activity in the brain, specifically in regions containing PC1 and AIPG, to this recurrent neural network. And so I'm basically at the end of my talk now. I see I'm right at time. So I just wanted to mention that in the second part of the talk, I talked about persistence on timescales of minutes. And in general, we tend to think about these longer timescales of internal states as being mediated by peptides and hormones. We talked about that already this morning. What I told you about was a recurrent neural network in the Drosophila brain that seems important for generating these long-term effects. And recurrent neural networks are best known for seconds-long timescales of persistence, maybe underlying working memory. And so I think it's interesting to think about how these two are linked. And I want to point you to really beautiful work from David Anderson that came out recently, showing that both recurrent neural networks and peptides contribute to arousal states in mice. And maybe he'll tell us about that this afternoon. So to summarize, what I showed you today is that there are three dynamically changing internal states that shape song patterning decisions in Drosophila males, that a GLM-HMM can identify these invisible internal states from behavioral data and can help us find the neurons that control state, that females have an arousal state that persists for minutes, that the PC1-DE neurons drive persistent neural activity, particularly aggressive and courtship-like behaviors in females, and long-term changes in behavior, and that PC1-D and E are a part of a strongly recurrent neural network with fruitless positive AIPG neurons. And so with that, I'd like to conclude, and I want to, in particular, thank the members of my lab. They've been really remarkable over the past 7 months. It's been a hard time for everyone, but it's been a privilege to work with them. And I want to thank you for attending today.
Thank you very much, Mala. I think we have time for maybe a couple of questions. One question is posted by Augustine Amarillo Porras. How did you discriminate between pushing into the closed state and just activation of the fast pulse execution motor circuit in the pulsatile optogenetic experiment?
Oh, that's an interesting question. So I think the question is about kind of dissociating the effects of optogenetic activation on the motor behavior and on the state. And it's because we have the GLM-HMM model that we could make that dissociation. So when you activate a neuron, you see an effect on behavior. In this case, the male starts to sing these fast pulses. But if we fit the model to the data, we can look at the change in state behavior, and we can see it's more likely for the male to be entering the close state. What does that mean? It means that the sensory motor transformation he's using is aligned with the one he uses during the close state. So he's using those filters to take sensory cue and turn it into pulses. And it's really interesting that pulses don't dominate in the close state. Actually, sine song dominates in the close state. But you do see some pulses in the close state. And for those pulses, it's the close state filters that you need to predict those pulses. So that's what we see here.
Maybe we should go for another question. So can you tell us a little more about this PC1 ABCDE neurons? Are they excitatory neurons? Just what kind of properties these neurons are?
Yeah, yeah, they're really interesting neurons. So this is not our work. So there's some previous work on this. I'm sorry, I've forgotten the citations, but most recently, Jerry Rubin has shown that in fact, these neurons are excitatory, as are the AIPG neurons. So that recurrent neural network I talked about consists of cholinergic neurons. Acetylcholine is a major excitatory neurotransmitter in the Drosophila brain. And so we think this is an excitatory loop. Of course, we need to do physiology within that circuit to confirm that. And we're very excited to, really kind of analyze the dynamics of activity in this network related to these long timescales of persistence. So we haven't done that yet. It's future work that we need to do. But these PC1 neurons control a variety of behaviors. And in fact, there are some recent papers from Barry Dixon showing that they modulate how females respond to courtship song, and they modulate how females respond to other sensory cues that drive their egg-laying behavior. So they seem to be modulators of a wide variety of behaviors beyond the ones I talked about here.
Very cool. Well, thank you. So such a stimulating talk. Thank you. So now we're gonna move to the next speaker. And it is my pleasure to introduce our dearest colleague here at MIT Brain and Cognitive Sciences Department, Ela Fee. Ela is going to tell us about mixed modular codes and remapping for highly generalizable learning and inference. Ela.
Thank you so much, Li Wei and Steve. It's really a pleasure for me to be here. And it's also an honor to be here among this august group of speakers. So also, given the circumstances of all of us working alone and separately in our offices and only interacting remotely, I guess I take special pleasure in the chance to have some interactions through these kind of symposia, even though it's not the same as being there in person. But thanks very much for attending. And I would like to tell you about one particular project that we've been doing in my group, and it's related to how we flexibly reuse mental representations. So let me share my screen and make sure that you can all see it first. Li Wei, is this visible?
Yes, perfect.
Okay, fantastic. Okay, so thanks again. And so I know that the title is a bit of a handful, but I hope that I can explain some of these terms and what I mean. So I'd like to start with setting out a fundamental tension, I think, that exists when we try to think about how the brain flexibly learns many things and how it represents many things using the same structures over and over again. So on the one hand, we hypothesize widely that the brain constructs neural representations of the world based on how the world factorizes. If the world has different pieces, the brain's representations reflect some kind of decomposed or decomposition of those pieces based on the structure of that problem or that data. But at the same time, general intelligence or the ability to make sense of lots of different kinds of data requires that we should be able to solve many different problems, but we have to use the same neural structures. And we have to do so without unlearning what has been learned before. So the fundamental tension here between these two objectives is how can these structured representations that may be formed based on factorizing certain problems be reused across problems in a way that allows this flexibility of learning. And what I want to talk about is both some insights from the brain, so some data motivated ideas for how to solve this problem, together with some theory on how this problem could be solved in the brain and also potentially in machine learning. So the outline, I want to sort of make my case in 3 pieces. So the first is to show you examples of how the brain is constructing factorized representations of metric spaces. The second one is to show you that the brain seems to reuse these very structured representations for disparate types of inputs. And then the third part is the theoretical part, which is a hypothesis that a specific way to reuse these structured representations, which I'll call mixed modular representations, and that involve random mappings from arbitrary input spaces to these structures, can allow for both high capacity and flexibility in representation and learning. So let's go to the first piece. So the first piece is some evidence that the brain constructs abstract factorized internal representations of metric spaces. So what I'm going to focus on for this part is the spatial circuit, the circuit in the hippocampus and entorhinal cortex that is involved in spatial processing and spatial representations as animals move around in the world. So I just want to show you, using the famous Van Essen and Feldman diagram, that these two structures, hippocampus and entorhinal cortex, sit atop this vast sensory processing hierarchy. So it's really at the pinnacle of this big sensory hierarchy. And in fact, this is just the visual system hierarchy, but hippocampus and entorhinal cortex actually funnel in all the different sensory hierarchies they project into the entorhinal cortex and into the hippocampus. So they're, they are the recipients of vast funnels of cortical processing of the sensory data, and then they construct representations of the world based on the sensory inputs and perform functions like memory and inference on those inputs. So here is a piece of that circuit that I want to talk about, and I want to just assign some names and roles to some of these pieces, specifically in the spatial context. So here I just show you some of the connectivity within that circuit. So there's thalamus and subicular inputs that come into entorhinal cortex. Entorhinal cortex in turn projects to hippocampus, and then hippocampus in turn, in a loop, projects back out to entorhinal cortex. So entorhinal cortex is the cortical gateway for cortical inputs to hippocampus, and then in turn, hippocampus sends various outputs out to the rest of the brain, including to entorhinal cortex. So now within the spatial context, the thalamus contains head direction cells that maintain an internal compass estimate of where an animal is facing in the world with respect to the external with respect to some external cues. So it is really truly a compass in the sense of some externally referenced sense of orientation. I'll show you a little bit about these head direction cells. And in turn, the entorhinal cortex contains many different cell types, including famously grid cells, which seem to represent an agent's position as it's moving around in the world. So X and Y, 2D Euclidean coordinates as it moves around in the world, or potentially 2D Euclidean displacements relative to a starting position in the world. And then place cells, of course, have tuning to locations, but they also represent other things which are non-spatial. So they represent context. So they represent non-spatial contextual cues, like, for example, the color of an environment, the smells in an environment, reward locations, et cetera, and also things like environment identity. So they not only encode positions within an environment, but also environment identity. So you can see that there are many different variables that are involved in spatial navigation, including one's orientation, one's position in the world, contextual cues, non-spatial cues, as well as things like landmark-based information about features in the world, like external landmarks, environmental boundaries, etc. And what's interesting is that these different areas, although they're intimately connected and interacting, they do have some kind of specialized representations for specialized subparts or subvariables related to localization in the world and related to mapping and related to navigation. So this is an example of a kind of factorized representation of different variables necessary for navigation in the world. And that was an anatomical diagram, but I would like to give you some of the data and show you an example of what neural responses as a population look like in some of these areas. So this is data from the thalamic structure, the ADN, that sends projections downstream to the entorhinal cortex and contains head direction cells. So what's very interesting about this system is not only does it encode this compass direction, but if you look at the whole population responses, and here each dot represents, it's a vector, and it represents the instantaneous state of the circuit of all these neurons in this area, ADN. In this case, we have a sample of about 30 to 50 cells simultaneously recorded from one animal in that circuit. And so this is a 30 to 50 dimensional vector, and it's a snapshot of activity at one moment in time in that network. And the different dots here are snapshots taken at different moments in time as an animal is running around in a two-dimensional space. And the animal's behavior is not constrained in that space. It does rich things like moves around in 2D at different speeds and different directions. It pauses to eat bits of scattered food. It rears up on its hind legs to attempt to look over the walls. It can pause and groom itself. And so it is engaging freely in behaviors in this environment. And during this whole time, all of the data are being recorded from ADN. And what I'm showing you here now is a plot of the set of states recorded across the ADN in a 20 to 30 minute window. And what you can see, I think, is that this structure, the set of all states that are visited by this 2,000 strong neural circuit, lie on a very structured manifold, this distorted and bent, but still one-dimensional ring. And this is fitting because these neurons are encoding head direction, but what this one-dimensional ring is telling us is that really they're not encoding much else during the behaviors that the animal is performing here. It's not really encoding any other variable but head direction. In fact, we can show that the variable being encoded is head direction because we can perform a nonlinear manifold fitting to this manifold. So we've got this one-dimensional manifold. We can characterize its dimensionality and topological structure, and then fit a spline to it that matches its topology and dimension, then parametrize points along that spline over here. That's the color coding. And now this color coding is and completely unsupervised estimate of whatever variable this internal set of states, these circuits, are encoding. So we can, without any reference to the measured direction of the head of the animal. So of course, this fit is arbitrary in terms of which direction corresponds to clockwise, and also it's arbitrary in terms of which dimension, which location we assign the 0 coordinate to, or 0 degrees. But up to those two unknowns, we get a full decoding of this angular variable, which is plotted in blue, and then modulo that arbitrary rotation and choice of origin, you can see that the actual measured physical head direction of the animal as it's moving through this two-dimensional world is a close match to this latent space representation on this one-dimensional ring structure inside the brain of the animal. So it's remarkable that although in the mammalian head direction circuit, cells may not lie next to each other physically, like shown in this beautiful head direction work in Drosophila by Vivek Jayaraman's group and Gabby Maimon's group. Nevertheless, internally, the dynamics fall in this one-dimensional ring structure, and you can decode all of these one-dimensional states. So that's one example of a low-dimensional factorized encoding in the brain. I'd like to give you another example. This is, of course, grid cells. These grid cells, again, are recorded in similar conditions as in the previous experiment. Black is the trajectory of the animal as it runs around, and red are the spikes, individual spikes emitted by one neuron. All the spikes in this plot are emitted by a single cell, and you can see why it's called a grid cell because of its periodic and regular arrangement of fields. Okay, so these grid cells, theoretically, based on dynamical models of these grid cells, the circuit is hypothesized to lie, the states of the circuit, just like in the previous head direction case, the states of the circuit lay on this one-dimensional ring, even though there were 2,000 neurons and the state space is in principle 2,000 dimensional. Similarly here, the thousands of neuron strong circuit is hypothesized by the models to lie on just a two-dimensional torus. So all the states, if recorded, should lie on the surface of just a mere two-dimensional torus in the state space. In fact, some experimental results support this hypothesis. So this is two cells simultaneously recorded in two different environments, a familiar environment and a novel environment. And that's true for cell 1 and cell 2, and they were simultaneously recorded. And it turns out that their cell-cell relationships are strongly constrained. So although the response in the novel environment is significantly different in period and in orientation from the familiar environment, nevertheless, the cell-cell relationships are completely preserved. So if the period is different in a certain way between two environments in cell 1, it's different in the same way for cell 2, and thereby the cell-cell ratios for all the parameters described in the response are completely rigidly preserved relative to each other across time and environments. Moreover, the cell-cell relationships, like the correlation structure seen between different grid cells in a module that share a common period, are preserved across different behavioral states, including running, REM sleep, and non-REM sleep. So that means that this is a circuit that's really internally structured and stabilized by internal recurrent connections, and it's rigid. It doesn't change. The cell-cell relationships do not change. The dynamics do not change across environments and across behavioral states. So these I give you as pieces of evidence that the brain really contains very low dimensional, rigid, structured representations that factorize the variables that are being represented in the world. Now, a few things to note about the grid cell representation are the following, which is one is that the grid cells perform, so not only are the spatial variables like two-dimensional Euclidean position, angle, and then external cues like landmarks, all of these are decomposed and factorized and represented by different cells in different brain areas. But what's strange and surprising about some of these representations is that even variables that don't factorize seem to be represented in a factorized way. So, so let me explain what I mean here. So grid cells perform a modular or factorized decomposition of a quantity that's unfactorized itself. So grid cells are believed to encode 2D spatial locations. So it's just a Euclidean 2D coordinate about where the animal is in the world on the floor of a 2D space. But nevertheless, the representation by grid cells is broken up into the representation by many different modules that have different spatial periodicities. So here, as you move along this anatomical direction from dorsal to ventral in the entorhinal cortex, the periodicity of the spatial response of the grid cells is changing. And in fact, there are only a few discrete periods. So this is showing that there's a gradient, but subsequent work from the Moser lab has shown beautiful work showing that the grid periods are really discretized. So in fact, there are just a few different networks, grid networks, maybe 3 to 7, that have a few different periods that range from 1/3 of a meter to about 1.5 meters. And that's the range of spatial periods by these cells. Now, these cells have now formed these discrete modules that are encoding this one common quantity, which is 2D position. And so, and the way that they're encoding position is by these periodic responses. So if the animal were here, or if the animal were one period away over here, this group of neurons, this module, would have no difference in its firing response to that location. So in other words, this is a periodic representation of a non-periodic variable, which is position. And so it's a set of phases, periodic phases, two-dimensional periodic phases, which is x modulo the lattice period lambda divided by lambda. So we can think about these as 0, 1 circular variables in each of the 2 dimensions. And then we have a set for n different modules. We have a set of n of these different 2D phases. So that's strange. And I should also note that, you know, the spatial distances animals, these rats run through in the course of a day looking for food goes from 100 to 1,000 meters per linear dimension, much, much bigger than the spatial scales. And in fact, this is a nice paper showing that animals are prodigious explorers and can move not only across land, but across large expanses of open water to explore their world. So then why do you have this factorized representation of a fundamentally non-factorized variable, which is just 2D spatial location? So the idea and the analogy I like to give is, you walk into a room and instead of seeing a clock that indicates time in the, over the course of a 12-hour day, instead you're faced by a wall of clocks, all of which are telling time, but their periods are extremely small. So this is a 9-minute clock, a 10-minute period clock, 11 minutes, 17 minutes, and so on. And for some reason, then this is the way to represent time. And the goal is to then extract an estimate of time in the 12-hour day using many of these short period clocks. So it seems a bizarre representation, and it seems bizarre to factorize or modularize a representation for a variable that isn't modular. Now, it turns out there's at least in principle, there's a very good reason why this kind of modular representation might be useful. So this is properties of this modular code for space. And the idea is the following. So this is just a really simple example. Let's just consider that positions are just one-dimensional and integer valued. So you have position 0, 1, 2, 3, etc. And suppose that we've got responses that are periodic with a period 3 and a period 4. So then if you start at position 0, then maybe the phases associated with that position 0 are 0, 0. You move 1 unit, so the phases go up by 1. You move 2 units, the phases go up to 2, 2. And then once you've gone a distance of 3, then the first period wraps back around and you go back to a phase of 0, 0, 3, and so on. So you can easily see that actually it's possible to generate unique codes for a range of positions X that actually can be described by the product of these two periods, in this case, 3 times 4. So now the range of unique representations is given by the product of the components, these modular components periods. And I've given you an example of co-prime integer periods, but in general, this whole idea generalizes. So we can talk about real valued positions and real valued phases. And the idea is if you've got one grid network that's representing position in a periodic way along one pair of dimensions. So then, so according to one period in 2D, so then its phase wraps around, every time position goes around some distance. So I represent that by one circle here. This is one module. A second circle is the second dimension of the torus over here, and so on. So if you have, say, 4 different modules, then you would have a 4-dimensional torus. Here I'm only showing you a 2-dimensional one. And what we're saying is let's start at some phase. So let's say this point 0, 0. And as you move in space, this is position X, and position increments, then the phase wraps around the torus in this coding line. And as you go through, this line keeps wrapping around. In fact, the coding range of this code is scaling exponentially in the number of distinct periods, even if the periods are similar in size. Okay, so that means that breaking down even a non-periodic or non-modular or non-factorized variable into some modular representations can have some big efficiencies because you get these exponential codes even using linear neurons. So this is using order n neurons, but you get this exponential scaling. So that's the evidence that the brain constructs these low-dimensional structured representations based on how a problem or some inputs factorize. So now the question is, the brain has to use the same structures to be able to do many things, not just one. And here's some very interesting recent evidence that has been coming into focus over the last 4 or 5 years from many different labs showing that the brain seems to reuse these specific rigid structures that we previously studied in the context of space, also to represent non-spatial cognitive spaces. So, so this is a body of work, and I've just compiled, and there's actually some other beautiful results that have been put out over the last several years. I'm just highlighting a few to give you the idea. So here, this is one experiment from Beth Buffalo's group showing that as primates, these are macaques, are free viewing a visual scene. So they're looking at just images projected in front of them. Then if they record neurons from entorhinal cortex where the grid cells are found and plot their activation as a function of gaze location on that screen. And what they find is they find a multi-peaked response pattern of single cells again here. And I think you'll see that this image is reminiscent of grid cell activity as a function of 2-dimensional space. it looks like these grid cells it looks like these cells in entorhinal cortex are expressing grid-like firing patterns as a function of gaze location. Similarly, these are two now different human fMRI experiments. And in fMRI, there are some established hallmarks of grid-like activity in an fMRI signal. And what they find is a very interesting result, which is that they construct in this experiment a 2D, just an image, which they show to a subject. So here's an image, it's a cartoon bird. And this cartoon bird can be varied parametrically in 2 dimensions. Its neck can be stretched or its legs can be stretched. So this is now a 2-dimensional conceptual space. Subjects are able to move a joystick and actually move through this 2-dimensional space of neck and leg length parametrically and smoothly. And as they do that, then they solve some kind of spatial task because certain landmarks are co-located with, at some point in that 2-dimensional parametric space. And as the subjects navigate through this 2-dimensional abstract space, they find some of the characteristic signatures of modulation of neural activity at every 60 degrees, which they interpret as consistent with the grid cell activity during navigation through this abstract space. And this is also true for a similar experiment repeated for gaze location in humans. So the bottom line is that it looks like these circuits that are involved in representing spatial location as you navigate in an embodied way through the world is also being repurposed to represent location through some more abstract cognitive spaces. So now those examples that I showed you were just done in 2 dimensions. So although we move through the world in 2 spatial dimensions, those particular non-spatial examples were also 2 dimensions. But the question, really the bigger question, is how do we reuse the same networks when the inputs may be differently structured? So they may not be 2-dimensional, they may have different structure to them. So my hypothesis, which I want to talk about now, is that random input mappings to these structured modules can give rise to both high capacity, like we saw already, and give you flexibility of representation. So let's dive in. So I want to give you 2 examples of that hypothesis. So the first one is the use of the 2D grid cell spatial navigation circuit to represent non-spatial variables, but of higher dimensions, so not just 2 dimensions. And the question is, how can a set of grid cells or grid modules that represent 2-dimensional space with these 2-dimensional geometric patterns, how could they be used to represent variables of a different dimension? So the idea is really simple. So let's just first consider just conceptually over here. So all of us have studied the visual system at various points, even if we're not vision neuroscientists, but we know that the retina constructs 2D projections of a 3D world, right? So here is a 3D bust of Beethoven, and here are a series of 2-dimensional snapshots or projections of this 3-dimensional bust of Beethoven. And given enough of these 2D projections or samples, it's possible to reconstruct the full 3D structure from these 2D projections. So that's the very high level idea. And so given that, and it turns out that if you have enough of these, you don't need to make these very specific evenly spaced projections. You can actually just make random projections and then reconstruct the higher dimensional information. So that's the simple idea right here. So the idea is, given an arbitrary variable in arbitrary dimensions that's metric, construct a random projection matrix which takes velocities in that abstract space and then maps them into velocities that drive the different grid modules, and do so with different amplitudes and different random projections onto each of the modules. So if you have some k-dimensional variable, the k-dimensional variable is projected to this module's 2D representational space with some projection, and this one with a different projection, and so on. So the idea is that then now you have 2 different projections of, say, an animal's location in a 3D space, and by using these 2 different projections, you can then reconstruct uniquely the representation of the location of that animal in 3D. In fact, we find that with just a few number of modules, so this is 1 module, represents with this projection method, the representation is both it's periodic, so it's ambiguous even in 1 plane about the location of the animal, and this is a second possible module. So, but if you have 2 different modules, then 2 different modules with different projections, angular projections, both help to reduce a bit of that periodic ambiguity, but also some of that dimensional ambiguity, which comes from this elongated dimension in the 3rd dimension, because these cells only represent 2 dimensions each. So by combining multiple modules, you get a reduction in ambiguity from both the periodic nature of the representation and from the fact that the representation representations confined to 2D. And so by the time we get to 4 modules combining together, you can get unique representations of 3D space from multiple modules. In fact, what's interesting too about this representation, specifically with this random projections of higher dimensional data onto 2-dimensional representations, is that you get, first of all, an exponential capacity in the representation, just like we did before for grid cells representing 2D space. And there's an on-the-fly range dimension trade-off that involves no plasticity. So let me explain that result. So here we're plotting the range over which the code has a unique representation for different locations per dimension. So this is the per dimension unique representational range as a function of the number of dimensions for a fixed number of modules. So here we're considering 9 modules, and then we're just varying the dimensions. So as the number of dimensions increases, say it's 6, it's large, then the range per dimension gets lower. And if the number of dimensions that needs to be represented is smaller, then the unique coding range per dimension is bigger. And this is true just flexibly. It's a property of just packing some high dimensional variable into a lower dimensional space through these random modular, these mixed modular random projections. And the nature of that packing means that if you pack fewer dimensions in, the manifold that you've packed in is just naturally more spaced out, better spaced out, and then you can get a bigger range per dimension. And if you pack more dimensions in, then naturally the way the packing happens is you get smaller range per dimension. This requires no plasticity. And it's a smooth and continuous trade-off of encoding dimension with range per dimensions. So let me just take a moment to just contrast or just talk about what different possibilities could be. So given that you have modules to represent information, what are different ways that those same modules could be used? And I want to contrast a few different possibilities. So what we have here, this is a highly abstract schematic, so just bear with me. So let's just consider that we have 2 distinct variables, x and z, right? And for simplicity, we can just think of x as 1-dimensional and z as 1-dimensional. So combined, you can think of this as a 2-dimensional variable or 2 1-dimensional variables, it doesn't matter. So now the traditional grid cell code that I described to you right at the beginning for how it might encode 2-dimensional space with 2-dimensional modules is that, you just have x, you just have positions, and we just think of this as x as positions in 2-dimensional space. And as you go from x equals 0 to, bigger and bigger values of x, then we have this coding space, which consists of the phase phi1 of module 1, phase phi2 of module 2. And so we start at some phase, which is say 0, 0. And then as the position increments, the coding line is this wrapping thing on the torus, like we talked about here. I've just unfolded the torus into a square handkerchief. So I've just cut the 2 ends of the torus. And so this coding line comes out from, it exits here and emerges here, and it continues wrapping around and so on. So in this scheme, we're using the full coding space, phi1, phi2, these 2 modules to encode just the variable x. And as a result, like we already saw, you get an exponential coding range of x because you've broken the representation into this modularized or factorized representation. So you get, a product of the number of neurons in this times the number of neurons in this as your total coding range, rather than something that's just linear in the total number of neurons. So you get this exponential gain in coding range. But the disadvantage of this code is that you can't now, you've used these multiple modules to encode 1 variable, so you have no space to code the second variable z. So this is an efficient coding scheme because it has a high capacity, big range, but it doesn't code different variables. The second thing that you can do is you can say, well, look, if I've got 2 modules, I'm going to specialize 1 module to code for x, and I'm going to use changes along the other module, which is the other phase, to code for z. So you can do that. So now you've got 2 different variables, 2 different coding dimensions, use 1 each. And this is also efficient because if you had now 4 total modules, you could devote 2 to coding this x and 2 different modules to coding z. And now you would have exponential capacity in each one, but it's not flexible because you have to pre-factorize, you have to pre-allocate specific modules to specific variables. Now, the third version, which is what I'm proposing here, is a mixed modular coding, which is to say that we take both x and z and encode x using both modules. So small changes in x map to different changes in both modules, and changes in z also map to changes in both modules. So another way to say it is that each module encodes both x and z. So any change in x pushes you along in both module 1 and 2, and any change in Z pushes you along also modules 1 and modules 2, but they do so at different angles. And so both of these excursions in X and Z can access all the same coding points in coding space. And therefore, if you only have excursions in X, then X can use the whole coding space without interference from Z. And if you encode only Z, it's the same. And if you encode both, then each of them is using some subset of the coding space, and then the range for each one goes down. So this is the idea of modular codes, but factorized modular codes. And this is the idea of modular codes, but mixed modular coding. So this is the thing that I want to contrast. So fantastic. So now I want to give you just very briefly, this is a bit more of an involved example, and I have less time on it. So I'm just going to give you just a very high level idea, but it's a very different use of these mixed modular representations to generate now high capacity and robust content addressable associative memory. So here the idea is to construct a network that's a bipartite network. So you've got some constraint nodes and you've got some input nodes. These input nodes are just neurons. These constraint nodes here are actually modules. So each constraint node is a small network of a few neurons that are connected to each other through winner-take-all dynamics, so lateral inhibition, and then they receive inputs that are pooled. They're okay. So moreover, these hidden layer neurons. Okay, I said here that they're connected to produce winner-take-all dynamics within them, and they're called constraint nodes. Now, the connectivity between the inputs and the constraints, or this hidden layer, are follow a random graph. So it's a random expander graph connectivity, and expander graphs don't mean that the network, the hidden network up here, so this is the inputs, these are the hidden layers. Expansion doesn't mean that the network is big. Actually, these two networks are the same size. Rather, expansion is a property about how a set of random edges is distributed between networks. So if I have a neuron that can have 3 outputs, and I consider some group of 4 neurons, the set of 4 neurons can together project to 12 output neurons, and that's considered a good expander graph. But if I have the same 4 neurons with the same 3 outputs each projecting to the same 3 outgoing neurons, the neurons in the output, then this is called poor expansion. So this kind of graph structure with good expansion and very sparse connectivity can give rise to, and these are set randomly, these connections and these weights. So this is actually a form of mixed modular coding. These kinds of structures can give rise to exponential capacity in memory. So they can give rise to a number of, so if the number of neurons in the network is plotted on this axis, then the number of stable states that this network has, so the number of fixed points, which can be used for persistent activity states or memory states, grows. As you can see on this axis, this is a log scale. So this is actually an exponential expansion of the number of fixed points as a function of number of neurons in the network. Moreover, these fixed points have very large basins. So these are fixed points that are stable, so that even if you corrupt the state away from the fixed point, the dynamics of the network automatically correct the state to move it back to the correct fixed point. In this case, in this particular numerical implementation, up to 10% of all errors can be corrected for every one of those fixed points. So this is a very special structure, because with linearly many neurons, you have exponentially many stable states or fixed points, and the basins are large. So this is all I want to describe about the workings of this model, but I just want to give you one final thing, which is an application of this model. So it's possible to map. So those states that I talked about, exponentially many, are not user defined. So in order to do a task that's user defined, you want to take external inputs and map them to these fixed points or these stable states in such a way that, and this is how you do it. So this is our bipartite 2-layer memory network. And now these are the external inputs, for example, the image of Angelina Jolie. And there's Hebbian learning, one-shot Hebbian learning between these inputs and then the states in this memory network. So now these states become abstract label states for these inputs, for the input of Angelina Jolie's face. Now with this construction, so these are random and fixed. These are input driven and associated with one-shot learning. It turns out that this network is capable of one-shot recognition and robust recall. So given a noisy version of Angelina Jolie's face, it will correct back to the label corresponding to Angelina Jolie. So it leads to one-shot rapid learning. The mapping preserves distance relationships from the input space, so it's content addressable. This circuit in computer science terms is performing nearest neighbor search, which is a very high complexity problem in general and high dimensions. So that's the end. And so this is just showing you that some results showing that with this kind of associative memory, it's possible to generate and pull up correct labels from noisy input data after one-shot learning of the categories. And so, so this is it. So I'd just like to conclude by saying that what I've shown you is that it's possible to make a fast reuse of low dimensional recurrent structures to represent new variables. The traditional view of compositionality for representation is that you take fixed structures that factorize a problem, and that these same factors recombine to solve new problems that may have the same subfactors, right? So in other words, we factorize a problem, have fixed representations for the fixed factors, and then hopefully other problems that come along have the same factorizations so we can reuse those fixed factors. This is a new view of compositionality because through mixed modular coding, which is that the structures that are modular are fixed, but the representations or the way they factorize problems changes. You just have these random input projections to those fixed structures, and then you combine those random projections onto the modules to solve new problems using new factors. And the solutions are high efficiency and maybe a solution to the problems of the curse of dimensionality representing higher dimensional spaces, and also the problem of lifelong learning, which is appropriating the same structures to learn multiple new tasks. So I will stop here, except to acknowledge my really, truly wonderful and inspiring collaborators and students and postdocs who've done all of the work and come up with lots of these ideas, including Mirko Kluklas, Rishi Chaudhry on these works, Marcus Lewis, and many inspiring collaborations with Laura Colgan, David Tank, and Thibaut Taifunier. So thanks very much for your attention.
Thank you very much, Ila, for this wonderful talk. So just a very quick question. So what do you think modulate or regulate this fixed structure for, creating new representations? And, obviously some obvious candidates like the neuromodulatory system or whatnot. Any comments on this?
Yeah, Li Wei, I think that's a great question, right? So the point is that, right, so you've got these structures, and now the structures are representing certain inputs, right, in one domain, right? They're solving one kind of problem, and now the same structures, if they need to be reused to solve a different problem, the question is, what is the signal that switches? Which inputs are making it into the network? And that's a really good question. And so clearly they're going to be contextual inputs that come in and switch which inputs can be effective in driving these networks. But whether it's all on the level of neural dynamics, like for example, winner-take-all mechanisms can lead to switching, right? So you can have also Robert Yang, one of our new faculty members who's worked with Xiaojing Wang, has worked on some ideas about switching in networks based on just activity and winner-take-all dynamics to do switching. But of course, neuromodulatory influences, I think, are a very, very important factor as well. And it could be that contextual cues then drive certain neuromodulatory systems that then drive the switching of which inputs make it into these circuits. And that's definitely something I'm very interested in, and hopefully we can work on and make progress on. But yeah.
Fascinating. Thank you so much, Ila. And so with that, we will conclude the morning session. And I would like to thank all of the speakers in the morning for their wonderful talks. And now we will have a lunch break, and we will come back at 1:45. Thank you.
All right, welcome back, everyone. I'm Matt Wilson, and I'll be moderating this afternoon's first session. We have 3 speakers, and our first speaker is David Anderson from Caltech on hypothalamic control of social behavior and internal states. David, take it away.
Thank you. Can you see my slide, Matt? I appreciate the invitation. Good, thanks. I appreciate the invitation to this seminar series and this symposium. I think it's a great topic, and I want to thank the organizers, and in particular Stephen Flavell, for choosing this topic and inviting me. So similar actions in animal behavior can be used to express distinct internal states or intents. For example, chasing can be used to obtain food for consumption or a mate for mating behavior. And in this case, it's easy for us to distinguish the intents based on our anthropomorphic projections onto the behavior, but it's not always so clear. And what I'm interested in is whether we can understand and study the coding of the intent of behavioral actions independent from the encoding of the actions themselves. And that's what I want to talk about today. And I'm going to talk about this in the context of male-directed mounting behavior. Now, in contrast to female-directed mounting behavior, where the intent is pretty obvious that it's reproductive, the intent of male-directed mounting, which is widespread throughout the animal kingdom, is unclear, and it may differ for different species. In particular, it's not clear whether this represents an affiliative behavior or rather a dominance behavior, a type of aggressive behavior. And those of you who have male dogs may have seen them engage in mounting behavior and wondered what exactly this behavior means. And so this raises the question, how to compare its underlying circuitry to that of female-directed mounting behavior to see where and how they are similar and distinct. So we focused on this question in mice. And in mice, female-directed and male-directed mounting behaviors by males are motorically very similar. Very difficult for a human observer to tell them apart. And in an effort to see if we could tell them apart in a more objective way, Ann Kennedy in my lab and Christine Segelin and Pietro Perona's lab used a system that they developed called the Mouse Action Recognition System, or MARS, for automated classification of behavior based on 14 key points and features derived from them to see if they could train it to objectively distinguish male-directed from female-directed mounting. And although the decoder does perform significantly better than chance, it's not much better than chance, as shown here by the distribution of the predictions of the mounts from the frame. So that indicates that even using objective criteria based on machine vision and machine learning, it's difficult to distinguish female from male-directed mounting. So we want these two forms of mounting. And as many of you know, it's well known that ultrasonic vocalization is used by mice in various social contexts, including female-directed social behaviors. But we've never before examined the type or nature of ultrasonic vocalizations that occur during male-directed mounting behavior. So Tomomi Kirigo, a talented postdoc in my lab who's responsible in collaboration with Ann Kennedy for almost all of the work I'm going to show you today, which I should say is unpublished, decided to perform recordings of ultrasonic vocalizations in our standard resident intruder assays during interactions with female intruders or male intruders. And to our surprise, we found that in contrast to the case of female-directed mounting, in where 100% of the mounts were associated with ultrasonic vocalizations, essentially no ultrasonic vocalizations were detected during male-directed mounting. Now, we use this in conjunction with some other behavioral assays to try to understand more about the meaning of USV negative male-directed mounting. And so, for example, in the case of context, we find that male-directed mounts are much less likely to be followed by pelvic thrusting than are female-directed mounts. In contrast, male-directed mounts are much more likely to be followed by attack behavior. So that indicates that USV negative male-male mounting behavior typically precedes attack, or is more likely to precede attack than pelvic thrusting. In addition, it shows a different effective experience, social experience, than female-directed mounting. If we begin resident intruder assays with animals 1 week after they have been co-housed, we find that on the first day of testing, the male mice actually show more male-directed mounting than female-directed mounting. But over the ensuing 3 days of testing, mounting towards female intruders increases and towards male intruders decreases. And in particular, what we see is the fraction of attack behavior that is associated with mounting decreases, whereas attack behavior without mounting, accompanying mounting behavior increases. So what that indicates is that this USV negative male-male mounting becomes less frequent as mice become more aggressive. So together, these data on the influence of context and experience suggest to us that USV negative mounting represents a low intensity form of aggressive behavior, or what we refer to, or I'll refer to for the rest of the talk, as dominance mounting. So we're then interested in how these types of behaviors are encoded in the brain, and we're focused on the hypothalamus, which is well known to play a role in the control of social behaviors, including aggressive and sexual behaviors. Much of our work has been focused on the ventrum in aggressive behavior, and I'll be talking about that, as well as a related area, the medial preoptic area, MPO or MPOA, with which it is densely interconnected. And our initial question going into this is whether mating behavior and aggressive behavior shared a common neural substrate in the hypothalamus controlling mounting behavior, or whether there were distinct neural substrates for mounting associated with mating versus mounting associated with aggressive behavior. Now, we had shown back in 2014 that optogenetic stimulation of estrogen receptor neurons in VMHVL triggers attack at high intensities or frequencies of stimulation, whereas at lower intensities of stimulation, it promotes close investigation and mounting. But because we weren't using ultrasonic recordings at that time, it wasn't clear whether that mounting was reproductive or aggressive in intent. So we repeated the stimulation experiments with ultrasonic recordings, and we found that indeed stimulation of the estrogen receptor neurons in VMHVL promoted an increase in mounting, but it was USV negative mounting, not accompanied by ultrasonic vocalizations. And here you can see that the fraction of USV negative mounts is increased to almost 100% by the optogenetic stimulation of these neurons. But that's not just because these neurons promote mounting but fail to trigger ultrasonic vocalizations. Rather, they exert a dominant inhibitory effect on ultrasonic vocalizations. So if we put a male in a cage and trigger ultrasonic vocalizations with a bit of female urine on a piece of filter paper, then optogenetic stimulation of these estrogen receptor neurons in VMH strongly suppresses the ultrasonic vocalizations in a time-locked manner. So that suggests to us that the mounting behavior that is promoted by VMHVL stimulation is not reproductive in nature or intent, but it is aggressive in intent and is low intensity aggressive behavior. Nevertheless, it is the case that VMHVL estrogen receptor neurons do play a requisite role in female-directed mounting behavior, shown originally by Nirao Shah and the Ogawa laboratory, and extended here. Chemogenetic inhibition of the estrogen receptor neurons strongly suppresses mounting behavior towards female mice. This may seem counterintuitive, but we think that it reflects the fact that the estrogen receptor population is heterogeneous and consists of subsets that respond to male cues and subsets that respond to female cues. And that's based on data that we have both from microendoscopic imaging of these neurons that show distinct male and female responsive cells, and also from single-cell RNA-seq data in conjunction with immediate early gene labeling that shows that there are distinct subpopulations activated during mating and fighting. So we hypothesize that this loss of function phenotype is due to the silencing of that subset of estrogen receptor neurons that is normally activated by female cues, whereas the gain of function phenotypes of attacking and USV negative mounting reflect activation of the subset that normally responds to male cues, although we have not proven this yet. So if VMHVL neurons control dominance mounting, then what neurons control reproductive or sexual mounting? That behavior has long been associated with the medial preoptic area, which lies anterior to VMH, but is still part of the medial hypothalamic behavioral control column. That assignment of function has largely been based on neuroendocrinological studies and the effect of steroid hormone manipulations. And we spent a long time trying to evoke mounting optogenetically by stimulation of these neurons, and were spectacularly unsuccessful. So we were very surprised that this region, which was well known to be implicated in female-directed mounting, we were not able to stimulate mounting behavior. And we reasoned by analogy to some of our work in Drosophila that we might need more specific activation of these neurons than just of the estrogen receptor positive neurons. And we noted in gene spares that were activated during mounting behavior were expressing VGAP, they were GABAergic. And so we designed a genetic intersectional strategy to optogenetically activate MPO neurons that express both the estrogen receptor and VGAT using FLIP and Cre recombinases. And what you can see is that in contrast to activation just of the bulk estrogen receptor population, which produces very little influence on mounting, even weak stimulation of this estrogen receptor VGAT population produces mounting behavior in almost 100% of the mice tested and in close to 100% of all trials. Moreover, the mounting that is promoted by stimulation of this intersectional population is accompanied by ultrasonic vocalizations, USVs, and these ultrasonic vocalizations appear in a manner that is time-locked to the onset and offset of stimulation. And importantly, the vocalizations are not a secondary consequence of the mounting behavior that is promoted by stimulating these neurons, because solitary mice in a home cage can be triggered to produce ultrasonic vocalizations by stimulation of these neurons in the absence of any intruder mouse. And I should say that Richard Mooney's lab has a paper on bioRxiv demonstrating a similar effect to promote ultrasonic vocalization by stimulation of VGAT neurons in MPO, and these are presumably very similar populations. So not only can activation of these neurons trigger ultrasonic vocalization and mounting towards female mice and male mice, it can also trigger it towards an inanimate object. So here's an example showing what happens with a toy mouse when we start to stimulate these neurons. You can see the ultrasonic vocalizations here, and you can see attempts at the mouse to mount the toy mouse. Furthermore, stimulation of these neurons can dominantly interrupt ongoing attack behavior and convert it to mounting behavior with ultrasonic vocalizations. And I'll show that here. Keep your eye on the circle when the light appears in the circle we started, the optogenetic activation of these neurons. So here we have attack going on. This is using our automated classification system, and we trigger activation of these neurons, and the attack stops almost instantaneously and is converted to mounting, accompanied by the ultrasonic vocalizations. So for us, these were really the long sought after make love not war neurons. Now, we were curious as to the functional relationship between MPO and VMH in the control of these behaviors. These two nuclei are densely interconnected, as shown by classical and viral tracing studies. And we wondered what was the nature of these reciprocal connections, the functional influence. Since MPOA stimulation, as I just told you, could inhibit attack behavior, we asked whether optogenetic stimulation of MPOA terminals in VMH had a similar effect. And you can see here that it does. The red rasters are attack, and you can see there is a strong inhibition of attack behavior. Conversely, if we stimulate the terminals of VMH neurons in MPOA, then we strongly inhibit mounting towards females, as well as ultrasonic vocalization triggered by female urine. Now, the VMH neurons are GABAergic, the estrogen receptor neurons are glutamatergic, and so this presumably involves some intermediate population of GABAergic neurons that is targeted by these VMH neurons, and we don't know where they are. So these functional studies provide a picture in which causally dominant antagonistic nodes, MPOA and VMH, control reproductive versus aggressive behaviors, with activation of MPOA promoting a reproductive state and activation of VMH-VL promoting an aggressive state. And this looks very similar to models of the type proposed by Tinbergen, where there are distinct nodes that control mating and fighting behavior that are interconnected with by reciprocal inhibitory pathways. This leaves open the question of what is encoded by actons in these regions during social behavior using different methods. And as I'll show you next, the results produced a very different picture than we took away from initially based on the optogenetic experiments. So basically what we found initially by performing two-site simultaneous fiber photometry, bulk calcium measurements from MPOA and VMH-VL estrogen receptor neurons in the same mice is that both nuclei are active during both types of social, of mounting behaviors, but the relative level of activity between the two structures differs, with activity in MPOA being much higher during reproductive mounting in green than during dominance mounting in blue. In VMH, both areas are increased during both types of mounting, but there's a slightly higher, but not significantly higher level of activity during dominance mounting than during sexual mounting. Now, one sees this when comparing between the two areas within a given behavior, where you see that during USV positive mounting towards a female intruder, MPOA activity, scaled activity is higher than VMH activity. And in what I'm calling neurotypical male mice, the converse is true. So that you could say that the relative levels of activity in these two regions simply reflect a sensory representation of intruder sex. This represents a female intruder, and this pattern represents a male intruder. But we think that that's not the case based on some very rare but important animals that we encountered in the course of these studies. So one of these animals, mouse 629, was a socially experienced mouse that never showed attack towards male intruders, but only showed ultrasonic vocalization accompanied mounts. And in that mouse, the pattern of activity of MPOA relative to VMH was more similar to that typically seen with a female intruder than it was with a male intruder. That is, MPOA activity was higher in MPOA than VMH. We saw another example of a mouse like this. This was a naive mouse, again, where in the presence of a male intruder attack behavior and the relative levels of activity in these two nuclei are flipped relative to neurotypical, the case of neurotypical mice. So this indicates to us that the relative levels of activity in these two nuclei are not simply representations of the sensory features of a male or female intruder, but they are related to the underlying motivational state or intent of the mouse. And we would infer in the case of these rare mice that this type of male-directed mounting is affiliative rather than aggressive. Now, do these changes in activity represent just quantitative variation in the same neurons during these different behaviors, or are different populations of neurons activated during these different types of mounting? To investigate this, we carried out analysis at the single cell level using miniature microendoscopic imaging using the Inscopics. And we found that in MPOA was the dominance of responses to female intruders, relatively fewer responses to male intruders, which is the converse of the situation in VMHVL, as we previously reported in 2017, where responses to males predominate, but one can also detect responses to females. And importantly, in both of these cases, the female and male responses in the two nuclei are in distinct and largely non-overlapping subpopulations of neurons. Now, within these populations, by correlating activity with behavior using our Mars system, we found in each nucleus neurons that were activated in a time-locked manner during the initiation of both USV positive mounting and USV negative mounting in a specific manner. So you see that here in MPOA, and you see that down here in VMH. That is, each nucleus had cells activated at the onset of USV positive. Now, these mount-activated neurons are not simply responding to the sex of the intruder or to male or female-derived cues, because using choice probability analysis, we can show that there is a small but significant subpopulation of neurons in each of these nuclei that can predict mounting behavior rather than sniffing, which we use as a surrogate measure of detection of the sex of the intruder, and with a choice probability of greater than 0.7, and which is statistically significantly different. And again, these populations of USV negative mount-tuned neurons and USV positive mount-tuned neurons are largely non-overlapping in the two nuclei. So accordingly, one can train decoders, linear decoders, to predict the type of mounting that is occurring from the pattern of activity in either of these two regions, MPOA or VMHVL, either in a time evolving manner or in a frame-wise manner. In other words, the type of behavior, mounting behavior, can be decoded from activity in these two regions. Now, all that said, most of the strongly activated neurons in these two regions show mixed cell activity for both mounting and investigation. Those are the gray dots on these scatter plots. The colored dots down here are ones that are specific for investigation here or specific for mounting, one kind of mounting or the other. And this is reflected in how the population, the variance in population activity is explained in both of these nuclei, but particularly in VMH. A large fraction of the population variance in activity is explained by the sex of the intruder rather than by the encoding of specific behaviors. that we think that that represents the encoding of a state that is associated with intruder sex rather than simply a sensory representation of the intruder for the reasons that I mentioned earlier. So how do we reconcile our understanding of the function of these nuclei based on causal manipulations using optogenetics with the coding features that we've uncovered by measuring activity? So that causality reveals causally dominant functions for VMH in controlling aggressive behavior and MPOA in controlling reproductive behavior. And this fits with a conventional and prevailing view of social behaviors in the hypothalamus, such that one nucleus is assigned to control one behavioral state. But our neural coding analysis suggests that that's not the case. We find representations of both male and female directed behavior in both of these nuclei. There are even attack-tuned neurons in MPOA, arguing for a more distributed coding of social behavior across these hypothalamic structures. So given this, how can we account for the causal results that we see? Well, these neurons show mixed selectivity, not only for the type of behavior, but also for their preference for male and female intruder. And so you can see that in VMHVL, even cells that are tuned to recognize sexual mounting are more strongly represented among male preferring cells than among female preferring cells. Conversely, in MPOA, even cells that are preferentially tuned for dominance mounting are more strongly represented in female tuned than in male preferring cells. So one hypothesis to reconcile these data is that the causal dominance that we see in optogenetic stimulation experiments may arise from this asymmetry in sex-specific state representation, such that in MPOA there's a predominance of female state evoked cells, and in VMH of male state evoked cells with this strong reciprocal inhibition. Now, we don't yet know the precise function of these subsets of neurons that are tuned to the opposite state, whether it's positive or negative acting, although, as I said earlier, we hypothesize that the female responsive cells in VMHVL may be required for mating behavior. But the different cell types in these regions, and work from my lab on VMH and from Catherine Dulac's lab in the preoptic region has revealed that there are tens of different types of transcriptomically distinct neurons in these two nuclei. So it's going to require a combination of cell type specific functional perturbations together with quantitative modeling to understand how these functional interactions are implemented at the level of specific circuits. So the take-home messages from what I've shown you so far today are that male versus female directed mounting in mice are distinct social behaviors, with aggressive versus sexual intents in mice, although there are rare exceptions. That the genetically tagged neurons we've identified in MPOA and VM versus aggressive mounting, respectively, but that the neural coding of mount type is distributed across both MPOA and VMH, and that many, if not the majority, of the estrogen receptor positive neurons in these two nuclei appear to encode internal state rather than specific sensory representation or behavioral commands. And we think that the behavioral phenotypes that we're getting result from the triggering of the neurons that represent these aggressive states or reproductive states. So just to step back a little bit from this whole system, which takes a great deal of input from the pheromonal system, as initially described by Richard Axel and Catherine Dulac, transitions from sensory encoding into state encoding in the medial hypothalamus, and then to action encoding, likely beginning at the periaqueductal gray. And this recent bioRxiv paper from Richard Mooney shows very nicely that this transition for the case of ultrasonic vocalizations occurs at synapses between preoptic area neurons and periaqueductal gray neurons. We've been generally interested in how these internal state properties or features, some of which you've heard mentioned by Mala Murthy, are encoded by these nuclei, particularly including scalability, the ability to control behavior at different levels of activity, and persistence, the ability of the state to outlast its inciting stimulus. And interestingly, VMH not only contains in its ventrolateral region neurons that control these aggressive behaviors, but in other work that Mala referred to, there is a dorsomedial set of neurons marked by a different transcription factor that control defensive behaviors such as flight and freezing. And both of these populations control their respective behavioral types in a scalable manner. In the case of VMH, from dominance mounting to attack. In the case of the defensive neurons, from avoidance and freezing to undirected escape behavior. And they both exhibit persistent responses to the respective stimuli that activate them. In the case of these defensive neurons to predators, such as a rat. In the case of the VMH neurons, persistent responses to conspecific intruder males. And we think that what this may represent, and this is speculation, is that these neurons, which we know are largely glutamatergic and heavily recurrently connected, they function as some sort of a neural integrator that combines different sensory cues and converts them into some continuous variable that encodes the motivational or arousal state of the animal, and that according to the level of that state, different behaviors can be produced at different thresholds at downstream targets. And that's how the scalability would result from that. And in this view, the persistence is a property shown by neural integrators, as has been described in many cases by David Tang, Sebastian Sung, and others. And I'd just like to close by saying that these insights may help us try to reconcile what in my mind have always been two very different and conflicting views of the control of behavioral decisions by Konrad Lorenz and Nico Tinbergen, where Lorenz had this view of behaviors as being controlled in a sort of hydraulic manner by levels of drive in the animal and that he felt was controlling behaviors with progressively more specific decisions. And what I've shown you is that VMH and MPO likely correspond to these nodes predicted by Tinbergen for aggression and mating, and we provided evidence of this reciprocal inhibitory connections between these nodes. I've also shown in other work that VMH-DM corresponds to this defensive behavior node. But what's interesting is that in each of these regions, there is the scalable control of the type of behavior that emerges at the next level in the hierarchy: freezing versus flight, sniffing versus dominance, mounting versus attack. And perhaps it is the scalable control that incorporates some of the mechanisms in the form of neural integrators that Lorenz's hydraulic metaphor was referring to. Again, this is speculation, but it's one way of bringing these two views together. And with that, I'd like to thank you all for your attention. Again, to acknowledge the very talented people in my lab who made this work possible, particularly Tomomi Karigo, shown over here, and Ann Kennedy, our computational and theoretical neuroscientist who now has her own lab at Northwestern University. Thanks, I'd be happy to take any questions.
Great, thank you very much, David. We have a couple of questions in the Q&A. The first question is from Ernesto Cabezas-Bu, and he asks, can optogenetic inhibition of MPOA or VMH neurons during the onset of male to female reproductive mounting behavior trigger attack behavior or dominance mounting?
That's a great question. So we have some data from some animals indicating that it was actually chemogenetic inhibition of the MPOA neurons in a male-female encounter can actually switch mounting behavior to attack. But there were just not enough examples of that to get to statistical significance, but we haven't observed that. The converse we have not observed.
Second question from Andre Badesky asks, how distinct are the NPOA neurons that suppress attack toward adults from NPOA neurons involved in infanticide? And how specific is the inhibition of attack by stimulating NPOA neurons? Can they inhibit other non-aggressive behaviors?
So to answer the second question, we haven't seen inhibition of behaviors such as locomotion or defensive behaviors or feeding behaviors by stimulating these neurons. We've not looked at infanticide, so I can't answer your question. I will say that the neurons in NPOA, including the galanin positive GABAergic population studied by Catherine Dulac, are quite diverse. There are at least 7 or 8 different cell types within that population, just like there are 6 or 7 different subtypes of estrogen receptor positive neurons in VMHVL. So it's possible that the neurons that suppress attack towards adults are related to the neurons that suppress infanticide, but we don't have any evidence for that yet.
Great, thank you, David. I think we can move on to our next speaker, who is Amita Sehgal, who comes to us from the University of Pennsylvania, proof that good things happen in Philadelphia. And her talk is Metabolic State Determines the Need for Sleep in Memory Consolidation. Amita?
All right. Can you see my screen and hear me?
Yes.
Okay, great. So thank you, Matt. Good things do indeed happen in Philadelphia. So I'd like to start by thanking Li-Wei, Steve, and the other organizers for inviting me here. I've really been enjoying this symposium all day, and I'm really honored and delighted to be part of it. So I'm going to share with you today some of our recent work on sleep in the lab. Now, sleep is a behavioral state that's seen across species. It was thought to be restricted to mammals and some birds, but about 20 years ago, we developed a fly model for sleep, and it was followed by development of similar models in worms, in zebrafish, in aplysia, and most recently in jellyfish. So now work on these small model organisms is, rapidly leading to some novel insights into sleep. Now, despite the fact that we and most other animals that sleep spend a significant proportion of their lives sleeping, we still don't really know what the function of sleep is. But even a really long time ago, it was recognized that sleep might help with memory. Now, in this quote here, you will notice that sleep is not explicitly mentioned. The talk is of how a single night will increase the strength of memory. But I think the idea would be that it is sleep that contributes to that. Now, data for this came much later, although still about 100 years ago almost, where it was shown that people did better at memorizing syllables when they had slept rather than when they had been awake for a long period of time. So at this point, there's data from multiple organisms that sleep helps with memory. And exactly how it does that, we don't really know. It's probably a combination of replay, like the kind that Matt studies, together with some cellular processes. But there's quite a bit of evidence now that sleep helps with memory. So we've asked, are there links between sleep and memory in Drosophila, which is our favorite model? Now, in flies, the mushroom bodies, familiar to many of you, are a site of learning and memory. Oops. And they consist of these lobes shown here, alpha, beta, gamma, which I will come back to. Now, we quite fortuitously stumbled upon mushroom bodies as being relevant for sleep. And what that came from was that we had found that the cyclic AMP signaling pathway, protein kinase A in particular, affects sleep. And so we started mapping the site of protein kinase A action and found that PKA expression, so we're expressing a constitutively active PKA in the mushroom bodies, affects sleep amount. But we found that based on which part of the mushroom body we express it in, this PKA would either increase sleep or decrease sleep. And indeed, when we went and manipulated neural activity in these neurons identified by pKa expression in terms of their effects on sleep, we found that there were sleep promoting and sleep inhibiting neurons in the mushroom body. So shown here are two representative drivers. This one here, and again, both are pretty widely expressed. This one here, if you look at the sleep profile, so we're looking at sleep over several days during the daytime and the nighttime, and the darker or the bold line shows you the experimental. And you can see here that with this driver driving, activating these neurons, you get an increase in sleep here. And you get the opposite effect with this other driver. And then subsequently, Paul Shaw showed that sleep is required for learning in a mushroom body dependent task. And the task he used was an aversive phototaxis assay. And he basically found that flies learn this, but if they're deprived of sleep for 6 hours or 12 hours following the training, then they don't perform as well. 3 hours of sleep deprivation has no effect. So it seems that sleep is required for memory in flies and that mushroom bodies are important for this. But there are periods of time in all our lives where we have to survive without sleeping. And shown are a couple of examples of this. So dolphins, for instance, when they're taking care of their young, are sleeping very little. And when people or when animals are starved, they usually get very little sleep because what they're doing is spending their time foraging and looking for food rather than sleeping. So it's, trade-off between getting sleep and getting food. So we asked, how is memory consolidated when flies are starved and not really sleeping? It turns out that actually the assay for appetitive olfactory conditioning, a widely used assay for learning and memory in flies, is based upon starving the flies. So here is the typical paradigm. What you do is you starve the flies, then you train them, and the training consists of teaching them that this particular odor is associated with sucrose, with reward. And then you starve them again and you test. And this is taken from this paper by Michael Crashes and Scott Waddle. So we asked, how do you get learning in this case? And is there some sleep that's occurring that is required for this? And this work was done by, this is an unpublished story, and it was carried out by a very talented postdoc, Nitin Chauhan. Now, I should say that learning and memory is new to my lab. We haven't really done it. We don't know how to do it. But Nitin came with experience in this from Martin Heisensberg lab in Würzburg, Germany. So what he did was ask about the role of sleep in the typical paradigm. And also in this altered version of it, which Scott Waddle had shown, can also lead to consolidation of memory. And in this paradigm, what happens is that after training the flies, they're actually fed, but then they need to be starved again before they can be tested. So here is the protocol that Nitin used. So the appetitive conditioning consists of coupling a particular odor with sucrose, and the flies are taught to associate this. And then they're exposed to another odor that is not coupled to sucrose. And then they are transferred following this training into either vials that contain sucrose, or they are starved, which means that they are placed on agar without any sucrose. And what we did then was to see whether sleep is affected following this training using the Drosophila activity monitoring system. And what we found was that flies that are fed after conditioning show increased sleep. And that's shown here. So this is the number of hours after training. You see an increase in sleep if the flies are fed. On the other hand, if they're starved, there really is no increase in sleep, which was not surprising because, as I said, when flies are starved, they actually do reduce their sleep because they're looking for food. So Nitin then asked, what happens if you deprive these flies of this sleep that they have afterwards? So he trains them then and subjects them to sleep deprivation under either fed or starved conditions. And then he starves them before he tests them for memory. And what he found was that sleep is required for memory in flies that are fed, but not in those that are starved. And you can see that here, there's a pretty dramatic decrease in performance when you sleep deprive the flies post-training, only if they're fed, but not if they're starved. And consistent with the idea that you need sleep for memory if flies are fed, we found that one of our short sleeping mutants, a mutant we call red eye. So one of the things we do in the lab is do genetic screens for mutants that sleep less in an effort to identify regulators of sleep. So one of our mutants is called red eye because it has reduced sleep. And what we found was that these flies show deficits in memory if they are fed after training. So if they are starved after training, these flies actually do fine. It's only if they're fed. So what we have then is the situation where following training, if flies are fed, they show an increase in sleep and they need that sleep for memory. On the other hand, if they are starved, there's no change in sleep and they are capable of consolidating sleep-independent memory. So we asked then what it is about the food that's important. Is it the nutritive value or is it the taste? And to distinguish between these possibilities, we used arabinose, which has a sweet taste but is non-nutritive. And we use sorbitol, which to flies is not sweet and does have, though, nutritional value. And we found that flies that were kept on arabinose were using sleep-dependent memory. So in other words, they were regarding the arabinose as food. And so if they were sleep-deprived post-training, they showed a decrement in performance. On the other hand, those that were fed sorbitol were getting nutrients, but not sweet taste. And they were behaving like starved flies, where they had no dependence on sleep post-training. Now, the sweet taste can provide some level of satiety, at least on a short-term basis, to the flies. So we asked if they needed to be hungry in order to go to a sleep-independent memory mechanism. Now, neuropeptide F is the fly ortholog of neuropeptide Y, NPY, in mammals, and it is a hunger signal. So high NPF makes the flies hungry. So what we did was to knock down NPF or NPF receptor, as shown here. And when you do that, what you get is that flies that are starved, which normally would be consolidating memory in a sleep-independent fashion, now become dependent on sleep. So NPFR mutants, when sleep deprived, shown here, show a decrease in performance. And also they show an increase in sleep following training. So it seems that for sleep-independent memory, the flies need to be hungry, and this hunger is driven by NPF. So to summarize what I've told you so far, what we find is that you have sleep-dependent memory in fed flies, sleep-independent in starved flies. This is mediated by NPF. And in the case of the fed flies, you need the sweet taste gustatory neurons. And we've, in fact, confirmed a role for these neurons in sleep-dependent memory. Now, this idea that sleep, that there is a context to when sleep is required for memory is supported by some mammalian data. And this is actually from several years ago, where rats were put through a Morris water maze task and then deprived of REM sleep. And what was found was that when the task used a hidden platform, in which case the rats had to use spatial cues to figure out where the platform was, then they were affected by REM sleep deprivation, paradoxical sleep deprivation. So if REM sleep deprivation was carried out in the first 4 hours following training, then they took a longer time to find the platform. So for spatial learning, which is a hippocampus dependent task, they needed REM sleep. On the other hand, if it was a visible platform, then the REM sleep deprivation had no effect. So in other words, they could learn the cue and they could find the platform without any need for sleep. And this is a hippocampus independent task. Now, so this is a little bit different from our fly scenario, where this is 2 different types of learning. We're actually looking at the same type of learning, but under different conditions. But the fact that the circuits are different for these made us then think about what circuits might be used for sleep independent and sleep dependent memory. Now, I told you that the mushroom bodies are the relevant centers for learning and memory in the fly brain. And we know that for the appetitive memory consolidation, the relevant lobes of the mushroom body are the alpha prime shown here and the beta prime shown there. And in recent years, it's become clear that the alpha prime, beta prime can be divided into further subsets. So there are the alpha prime, beta prime AP or anterior posterior neurons. And then there are the alpha prime, beta prime medial neurons. And these are labeled with different colors here. I hope you can see that here. I'm just gonna play this here so it's pretty obvious that these are quite distinct from each other. And it turns out that these are functionally also distinct. So when you activate these neurons, and we're doing that here with the TRPA1 channel, what we find is that the AP neurons promote sleep. This is a driver for the AP neurons, and here are the data for that. The AP neurons promote sleep, whereas the M neurons shown here in the blue decrease sleep. So in other words, they promote wake. Now, consistent with the finding that the alpha prime, beta prime medial neurons promote wake, we find that they are activated under conditions where wake predominates. In other words, when they are starved after training. So here we're using Calexa, which is a reporter for calcium, to look at the medial lobes in flies that were fed after training or starved after training. So the lobes are marked in red here, and here's the calcium signal, and you see that it is higher in flies that are starved after training. So they're more active in flies that are starved after training, and they promote wake, and they are required for memory under these starved, more awake conditions. So here we're using Shabiri to silence these neurons, and using 2 different drivers that label the alpha prime, beta prime medial neurons, we find that sleep is, performance is reduced when these neurons are silenced. They are not required in flies that are fed post-training. The medial neurons are not required. Now, I'm not going to show you the data for this, but the AP neurons are actually functional under these conditions, but not under those conditions. So the AP neurons, which drive sleep, are required for memory in fed flies, which are dependent on sleep. And it turns out that if you sleep deprive flies, then, this is just showing that these neurons are required to promote sleep. If you sleep deprive flies, you reduce activity of these alpha prime, beta prime AP neurons, and that's indicated here. So you get this dramatic reduction in the calcium signals shown in green if the flies are sleep deprived. So the activity of these neurons is required for memory, because this is presumably why sleep deprivation impairs memory under these conditions. Now, in addition to the actual lobes, memory consolidation also requires the activity of dopaminergic neurons, specifically dopaminergic neurons in the PPL1 cluster, which connects to the mushroom bodies, and in the mushroom body output neurons, which are shown here. In fact, the mushroom body output neurons form a recurrent circuit with the dopaminergic neurons, which helps in the consolidation of memory. Now, I told you that the paradigm that's typically used to study appetitive memory in the fly is the starvation paradigm. So in fact, much of this circuitry has been mapped out for those conditions. So in starved flies, where we find that there's no change in sleep and memory is sleep independent, we know that the MP1 dopaminergic neurons in the PPL1 cluster and this particular gamma-1 peduncle mushroom body output neuron are required for memory, and they form a recurrent circuit. So we asked if these same neurons are required in fed flies, and the answer is they're not. So here in blue are the MP1 neurons silenced, and there's no effect on memory. On the other hand, if you silence a different dopaminergic neuron, the MV1, you see a decrease in performance. And likewise for the mushroom body output neurons, if you silence the gamma-1 peduncle output neuron, there's no effect on memory. It's a different output neuron. So we had this circuit here in starved flies. We now know that in fed flies, you have a different dopaminergic neurons and a different output neurons. And just putting it all together here, then here in fed flies, the sweet taste gustatory receptor neurons are signaling, providing a brain state, if you will, that leads to an increase in sleep and sleep-dependent memory. And the circuit used here is distinct from the one that's used in starved flies. So just to summarize, I've told you that sleep increases and is required for memory in flies that are fed after appetitive conditioning. It's not required in flies that are starved. These two types of memory are mediated by different circuits, so different subsets of alpha prime, beta prime lobes, different DANs, dopaminergic neurons, and different mushroom body output neurons. And the relevant subsets are activated under the different conditions. So the AP neurons are activated in sleep-dependent conditions and they promote sleep, and the medial neurons are activated in sleep-independent memory and they promote wake. And we believe that what this does is this plasticity in circuits will allow adaptation to different environments. So for instance, nutrient-rich or nutrient-poor environments. And these environments, of course, are altering the internal state of the brain. In this case, the metabolic state of the brain. And finally, I'd like to conclude by just acknowledging the people who did the work. The bulk of the work was carried out, as I mentioned before, by this very talented postdoc, Nitin Chauhan, who really brought these assays into our lab. And Paula Haynes, another postdoc, who actually, while as a graduate student in Leslie Griffith's lab, had started to dissect the roles of the alpha prime, beta prime, medial, and AP neurons in sleep, was a collaborator. And I think I'm actually a few minutes ahead of time, but I'll be happy to stop here and take questions.
Great, thank you very much, Amita. We do have a few questions. The first question comes from Augustin Amaril Porash, and Augustin asks, is there any apparent trade-off between the starved, hungry, sleep-deprived flies and the fed, rested flies?
Right, so you're asking basically if the memory that's formed is different in any way, right? I'm assuming that that's the question. So the answer is that so far we have not been able to detect a difference. In terms of how long the memory lasts, we've gone a few days out and we don't see a difference between the two. It's possible that on some subtle level, there's a difference, and we're trying to design assays to address what those differences might be.
Okay, great. The second question comes, Q&A window scrolling here, comes from Aguria Madan. And the question is, in your assays, is sleep deprivation exclusively induced by starvation? What about brief bouts of quiescence that may occur even during starvation? Was quiescence, or the lack of it, during starvation quantified?
Yes. So the sleep deprivation, okay, I should have clarified. So when we're actively sleep depriving the flies, we're doing it mechanically. Starvation does lead to a reduction in sleep. It doesn't completely eliminate sleep, but there is a reduction in sleep with starvation. And it is absolutely the case that there are brief bouts of quiescence that do occur during starvation, which is why when we wanted to test the role of sleep, we deprived the starved flies, because we wanted to make sure that that quiescence that occurred during starvation wasn't actually enough to give them the sleep that they might need. But then we found that, in fact, they didn't need it, and we found that by mechanically depriving them.
Okay, the next question comes from Troy McDiarmid, and I think you may have answered this in the first question, but I'll just repeat it here. And the question is, does sleep-independent memory last as long as sleep-dependent memory?
Right, so far it looks like it does, but maybe we need to go out further.
All right, and next question is from Lionel Muller-Igaz. Lionel says, great talk, Amita, which it was. Is activation of sleep-promoting neurons in alpha-beta mushroom bodies sufficient to increase memory performance in sleep-dependent memories in fed flies?
Okay. Yeah, that's a good question. Is if you activate them, can you enhance memory for... Okay, the answer, I'm going to cut out a lot of the complicated experiments we did, is probably not, because it turns out that what happens under fed conditions is not so much that you get an increase in activity in the AP neurons, you actually get a decrease in activity in the medial neurons. If that makes sense. And so it seems that the changes, that the AP neurons are not necessarily getting more active during the consolidation of memory. It's the other subset that's going down. So there are some inhibitory relationships here which we haven't quite sorted out.
Okay, thank you. Next question comes from Zhao Zhuan, and the question is, does mammalian NPY play a similar role as NPF in supporting sleep-independent memory in starved animals?
We would love to know the answer to that, but have not yet taken this work into mammals. So stay tuned.
Great. Next question from Hernan Lopez Shire. Is there any sexual dimorphism in sleep-dependent memory?
As far as we can tell, there isn't. So we're not testing males and females separately. We actually, all our flies are mixed, equal proportion of males and females. Every once in a while, there are more females that survive the assay than others, or males the other way around, and we haven't noticed a difference.
Great. And the last question I think it is here is from Wanhee Lee. And the question is, can you feed sleep drugs to the starved fly and enhance memory?
Great question. We have not done that. We probably should. Thanks for the suggestion.
All right. I think we've answered all of the questions. I want to thank Amita again for a great talk. And I think we can move on now to our next speaker, who's Bill Schaefer, comes to us from the MRC Laboratory of Molecular Biology in Cambridge, the other Cambridge, or as you might say, the real Cambridge. And the name of his talk is the Neuromodulatory Connectome, Wired and Wireless Networks. Bill?
Okay. Yeah, can you see my screen?
I see the full screen, not the presentation. I see your desktop, I should say.
Okay, is that?
I see your presenter mode, so I see the slides.
Oh, we've got the wrong.
Yep, other screen. Sorry, we have a few minutes, so we're still on time.
I think if you swap the displays, it might work for you, Bill.
Okay, how about that? Did that
Same,
work? Or?
Same.
Same. All right. Well, all right. I'll just forget the presenter mode. Sorry. Sorry, folks. Okay.
That looks good. Yep. All right, I'm going to suppress my my Mac comment. We do have a few minutes.
I'm gonna stop. Okay. Is that any better?
Yep.
Or is he?
Yep, that looks good. Yep. See the
Sorry,
slides, your cursor? I can hear you.
I can't hear anything.
But we can hear you. That's the important thing. Yep, I see your slides.
Is this working?
I see your cursor, and I hear you.
I hear you. Oh, okay. Sorry about that. Okay. Ay. Okay, I apologize for the Zoom catastrophe. I guess I'll get started. So I guess, as most of you know, a major push in modern neuroscience is connectomics, which is the idea that we can understand how brains work by mapping their connectivity at the single neuron level. But although the word connectome is relatively new, the concept of trying to map circuits by reconstructing them at the EM level is actually quite old, and many of you probably know that it was more than 30 years ago that White and colleagues published the complete worm connectome, a figure from which is shown in this slide. So in the entire C. elegans from adult hermaphrodite, there are 302 identified neurons, and we know all of the synapses that each of these make to one another, as well as all their gap junctions. And so this is just an example showing these neurons that each have an individual name, and we know more or less all of the single level connectivity in this entire organism. Now, perhaps for people who are fans of connectomics, this might be a little bit of a disturbing thought that we've had this complete connectome for an animal with only 300 neurons for over 30 years. And actually, we don't have this profound understanding of exactly how the C. elegans nervous system works that you might hope to get from mapping a whole connectome. And so the question is why, what information are we missing? So one thing, of course, we don't know is that even though we know which neurons make connections with which other neurons, we don't really know the functional nature of these connections. So for example, this neuron makes dopamine and we know it makes synapses, this neuron ABE. We don't know if these connections are excitatory or inhibitory. We don't really know anything about their functional properties. And perhaps a bigger problem, especially in terms of the theme of this symposium of behavioral states, is that we don't really know the complete structure of the interneuronal signaling networks of the animal, because extrasynaptic signaling, which we know is very important for neuromodulators like monoamines and neuropeptides, often occurs outside these mapped synaptic connections. And so what I'm going to talk about today is our attempts to address these two things. So first, I'm going to tell you about our attempts to start developing connectomic type maps for this sort of wireless extrasynaptic signaling. And then I'm going to tell you a little bit about some novel proteins that seem to function as synaptic in synaptic transmission involving neuromodulators. So before I get started, I should say that mapping the connectome is certainly not been irrelevant in terms of having a complete connectome for C. elegans. So one of the things that people have found, and again, this is quite an old paper, a classic paper from Watson-Strogatz, where they showed that the C. elegans neural connectome was one of the first biological examples of what they called a small world network. And so basically what that means is that that this network shows a high degree of clustering, that is a relatively high degree of interconnectedness of connected, well, basically that there's a lot of triangular patterns among the connectivity map, which indicates that neurons that are connected together tend to connect to common targets. But there also are a much shorter than average or shorter than expected path links in the network. And the reason that the network has these properties is because it's organized in the following way. So that it's both modular, where you have these communities with high internal connectivity, which gives you the high clustering. But then these modules are connected by these high degree nodes, which are referred to as hubs, but the hubs themselves are more interconnected with one another than you would expect by chance. And this sort of pattern of organization is what's sometimes referred to as a rich club. And so together, this modularity and this rich club organization gives you this small world properties. And so in C. elegans, it was shown that this rich club actually consists of 11 neurons, which are premotor interneurons that have been shown in subsequent work to be really important. For example, in the whole brain imaging studies of Manuel Zimmer and colleagues in driving global brain states in this animal. And it's also been shown that animals, including very complicated animals like ourselves, show a similar rich club type organization. But in this case, the nodes of the rich club are not individual neurons, but they're cortical areas composed of hundreds of thousands or maybe millions of neurons. And so this sort of global organization has been conserved across these very wide gulf of scale between this 300 neuron animal and this 100 billion neuron animal. Okay, so so we're of course also interested in understanding how neuromodulators fit into this circuit. And we actually can look at the pattern of expression of monoamine receptors and monoamine biosynthetic enzymes to see that, in fact, signaling that involves monoamine neurotransmitters is actually quite independent of this wired synaptic connectome. So in C. elegans, there are only 8 neurons that make dopamine, 4 pairs of neurons, 6 neurons that make serotonin, and a single pair of neurons that make tyramine and octopamine, respectively. Now, if we look at the expression pattern of known monoamine receptors and compare it to that synaptic wiring diagram, we find the following. So these are the two tyrosinergic neurons that are named RIM left and right. And these are the neurons that receive synapses from RIM. And you can see that some of them are colored yellow because they express known tyramine receptors. But there are many other neurons that are known to express tyramine receptors that clearly receive no synapses from the RIM neurons. So upwards of 95% of the known neurons that express tyramine receptors receive no synaptic input from these neurons. And we see a similar thing for octopamine. So in fact, in this case, none of the neurons that express known octopamine receptors actually receive synapses from the RICs, the only neurons that make octopamine. And similar things are seen for dopamine and serotonin as well. So what we can conclude from this is that the connectivity map of C. elegans, and probably for every animal, can't be really described as a single network, but rather as a multi-layer network where each network represents a different type of neuron-to-neuron signaling. And the edges in these networks represent the same neurons, but that are connected through these different modes of signaling. And of course, these synaptic and gap junction connections mediate communication that's wired and occurs on a rapid time scale, whereas these wireless networks of monoamine and neuropeptide signaling are mediating behavioral effects on a much slower time scale. Now, of course, we're interested in actually developing maps of these wireless signaling networks because in C. elegans, because we have a relatively small scale nervous system, we already know the pattern of synaptic and gap junction connectivity. We could, in principle, make a true multi-layer connectome where we actually relate these neuromodulatory layers to the same neurons in the synaptic and gap junction layers. So, and to make these wiring diagrams or these wireless signaling diagrams for neuromodulatory signaling is actually feasible in C. elegans more so than in many animals because of the structure of the nematode brain. So in C. elegans, almost all of the neurons, regardless of where their cell bodies are located, send their processes to one of two places, into this circumoral nerve ring that wraps around the pharynx, or into this ventral nerve cord, or to both. And so almost every neuron that sends a process into this nerve ring, which is a majority of the neurons, has a process that's within a micron of every other neuron that sends a process into the nerve ring. So to a first approximation, we can make a connectivity map for neuromodulatory signaling for monoamines and neuropeptides simply by knowing the expression patterns of the ligands and the receptors. And so Barry Bentley, a former graduate student in my lab, did this for monoamines using the known positions of monoamine biosynthetic enzymes within the C. elegans anatomy, and also the known or data that we obtained ourselves through reporters, expression patterns of the 5 known dopamine receptors, 5 serotonin receptors, 4 tyramine receptors, and 2 octopamine receptors. And so in doing so, he generated a network, the adjacency matrix of which is shown in this part of the slide. So if we look at the connectivity of monoamine signaling in the worm, we find something very different from what we see for synaptic signaling. So for all the different monoamine systems, the monoamines are released from a very small number of neurons, but they signal to a wide range of targets. And so what we generate is these highly disassortative star-like networks where there's a small number of releasing hubs, which are releasing various monoamines, and many peripheral receiving nodes. And if you think about it, this isn't so dissimilar from what we know about monoamine signaling in humans. So for example, we heard earlier today from Susan Temecki about serotonin signaling. Again, the serotonin neurons are very few in number, and they're found in these brainstem Raphe nuclei, but they project all over the brain and signal to all kinds of diverse neuronal targets. Okay, so this is all very interesting about monoamines, but of course the real holy grail for understanding neuromodulator signaling networks would be to understand neuropeptides, because this is a much more complex situation. So in contrast to monoamines, where there's only a small number of signaling molecules. In C. elegans, as well as in many other animals, there's a huge array of neuropeptides. So in C. elegans, there are at least 400 known neuropeptides that are produced from over 100 different precursor genes. And likewise, there's about 150 predicted neuropeptide activated GPCRs in the C. elegans genome. And many of these have significant homology to well-known mammalian neuropeptide systems, such as oxytocin, tachykinin, and so forth. And at least when we started thinking about this, most of the ligand receptor interactions were uncharacterized. The other thing that's worth noting is that the ligand receptor interaction patterns between peptides and receptors can be complex. So in some cases, there's a one-to-one relationship. A single ligand binds to a single receptor. But there also can be receptors that are highly promiscuous and bind a whole array of related neuropeptides. And you can get this many-to-many interaction. And finally, it's worth mentioning that most, if not all, neurons in C. elegans produce at least one neuropeptide and at least one neuropeptide precursor gene. So clearly, we're not going to see these highly disassociative star-like networks where only a small number of neurons are broadcasting signals to a wide range of receivers. The communication pattern is clearly going to be more complex than that. So we were interested in trying to see if we could generate a neuropeptide signaling map for C. elegans. And so a number of years ago, we got together with a couple of other labs, so with Oliver Hobert's lab at Columbia and the Bates and Timmerman labs from KU Leuven in Belgium, to try to see if we could generate a complete neuropeptide connectome for C. elegans. And so the general strategy is quite simple. So in one case, we would like to biochemically deorphanize all of the neuropeptide receptors in C. elegans and identify neuropeptide receptor couples, and then to do comprehensive gene expression analysis to find out where all these receptors and where all these peptides are expressed. And if we do so, we can generate this neuropeptide connectome. And once we have this connectome, we can ask all kinds of interesting things. We can define its basic topology. We can relate where these neuropeptide links are in relation to synaptic links, and hopefully generate functional hypotheses for how neuropeptide signaling works and maybe how it's affected by plasticity. So to begin with, we wanted to look at this question of expression. And in this case, we got a head start from the fact that Oliver Hobert is participant in the so-called SenGen project, where they attempted to generate, they've been attempting to define the complete gene expression patterns of all the neurons in the worm. And so basically the idea is twofold. So they do single cell RNA-seq to identify each neuronal class through this sort of dimensionality reduction clustering, and then to do deep bulk RNA sequencing of all these sorted neurons to generate a complete transcriptome for each cluster. And so in doing so, they've actually succeeded in matching up a single cluster to each known neuron type in C. elegans. In most cases, this refers to a single bilateral pair, or in some cases, a single neuron. So this gave us a starting point to this first part, or to this gene expression component of our connectome map. And then the second component was spearheaded by Isabel Bates in Leuven, and basically her idea was to express all of the C. elegans GPCR genes in this expression system where the cells express a promiscuous G protein that should couple to all the receptors and would couple it to this phospholipase C reaction that gives a calcium signal that you can detect in a chemoluminescent assay. And so in doing so, they screened this whole battery of GPCRs using a library of 402 C. elegans neuropeptides that can be detected by mass spec. And then they validated all these initial hits by dose response curves. And so in doing so, they were able to generate this matrix where about half of the receptors and about half the peptides were matched up in terms of ligand and receptors. So there were about 53 neuropeptide precursors, where at least one of the peptides produced by this precursor was a ligand for one of these 55 receptor genes. So once we had this data, we were able to generate our first version of a draft neuropeptide connectome. So for this molecular linkages, we decided to take a stringent cutoff and only use receptor ligand pairs where the in vitro EC50 was less than 500 nanomolar. So this is probably a pretty stringent cutoff, and we think that there are some peptides that do seem to interact with receptors in vivo, whose in vitro EC50 is lower than this. Likewise, we picked a fairly stringent threshold for analyzing our gene expression patterns, where we tried to minimize our false discovery rate at the expense of this true positive rate. And so in doing so, we generated this network adjacency matrix where we had basically directed edges between each peptide expressing gene with each neuron that expresses a receptor that can be activated by that peptide. So using this draft connectome, what can we learn? Well, we've actually only just got this in hand, so we're only beginning to dive into this data. But we've already learned some interesting things. So if we look at we compare the degree distribution of the synaptic connectome with this draft neuropeptide connectome, we see some very striking differences. So this is the degree distribution of the synaptic connectome. And you can see that there's a few neurons that have quite a high degree, and these are these members of the Rich Club. And so they have much more connectivity than the run-of-the-mill neurons in the C. elegans synaptic connectome. Now, in contrast, if we look at the neuropeptide connectome, we see something very different. So in this case, we see that almost half the neurons have actually a very high level of connectivity. If you look at this y-axis, you can see that this is itself about fivefold larger than this. So almost half of the neurons in the worm make either both in and out connections for more than half of the other neurons in the worm. So this is a much more democratic, if you will, form of organization that almost every neuron in the worm seems to have the ability to communicate directly with more than half of the other neurons in the worm. The other thing that we see is that there is this little spike here of neurons that are particularly well connected in the neuropeptide signaling, which suggests that they may be particularly important for organizing neuropeptide signaling. Now, if we actually look at the correlation between synaptic degree and neuropeptide degree, we find some interesting things as well. So first of all, we see that there's a slight positive correlation between neuropeptide degree and synaptic degree. And moreover, if we look at this synaptic rich club, we find that all of the neurons in the synaptic rich club, the most highly connected neurons in the synaptic network, all have a very high degree for neuropeptide signaling as well. And this makes sense because we already know these neurons are very important. We know that they have an important role in driving global brain states, and it makes sense that they should also be heavily involved with communicating between neuropeptide systems that also play a role in behavioral states. But there's also this other group of neurons that wasn't part of the synaptic rich club, but actually have more neuropeptide signaling links than the synaptic rich club. And there's 21 neurons that fall into this group. And if we break these down, we find that 5 of these are neurons that are known to be important for controlling sleep or arousal type behaviors. Another 5 are known to be important for oxygen sensing, which, if you remember Mario De Bono's talk this morning, we know is an important environmental cue controlling behavioral states. And finally, 6 of them are known to be important for navigation or motor behaviors. And then there are 5 that are completely uncharacterized. And so we actually know nothing about these neurons, but they seem to be really important for neuropeptide signaling. And of course, we're very interested to try to investigate their function using the typical tools of optogenetics and ablation and so forth. So just to, this is really sort of early days in this neuropeptide connectomics. So obviously there's many more things we'd like to do. First of all, we'd like to further characterize the network topologically, identify not only hubs, but also modules. We'd also like to do some functional analysis of these critical nodes by ablation and optogenetics, and also investigate the modes of interaction of this wireless connectome with the wired signaling using things like multi-layer motif analysis. We'd also like to do some refinement and functional characterization of this network. For example, determine whether the connections are excitatory or inhibitory. And we can do this by identifying the type of G-protein coupling that's mediated by each receptor. And we've got a pretty good pipeline going now in Xenopus oocytes to characterize these GPCRs in this manner. We'd also like to investigate the scope and dynamics of neuropeptide action in vivo. And so we're working on using GPCR sensors that are based on the principle of, say, the delight dopamine sensors to try to investigate this in vivo. And of course, we'd also like to de-orphanize the remaining receptors. And finally, we're also generating these integrated reporter lines that will allow us to visualize both receptor and peptide expression in real time. And this should allow us to investigate how the network structure changes during things like development, aging, and also in response to environmental cues. Because I think one thing that's clearly true is that it's much easier to change the structure of these wireless signaling networks because all you need to do is add a new receptor or take away a receptor, and you're actually adding or taking away an edge in the network. And so I think that this is going to really give us some interesting findings in terms of how various types of plasticity can affect the structure of neuromodulatory networks. Okay, now in the last part of the talk, I'm going to talk about something a bit different. And this is something that might have bothered you when I showed you this slide at the very beginning of the talk, which is it's all very well to say that there's extrasynaptic signaling that's responsible for communication between these tyrosine neurons and all these neurons. But what's going on in these white neurons that clearly receiving synapses from RIM, but don't have any receptors for tyramine, or these receptors, or these neurons that clearly are receiving synapses from octopaminergic neurons, but have no octopaminergic receptors? Maybe there are some missing receptors. And indeed, we wondered whether we might be able to find more monoamine receptors in the C. elegans genome that could explain these aminergic synapses. So as you probably know, there are two types of receptors that can mediate signaling from monoamines in all organisms. Most of the monoamine signaling is due to these metabotropic receptors that are coupled to G proteins, both in humans and worms. But it's also known that there are some ionotropic receptors, the 5-HT3 receptors in humans, and various receptors that have been found in C. elegans that are part of this pentameric cis-loop ligand-gated ion channel family. And it turns out that the cis-loop ligand-gated ion channel family is vastly expanded in C. elegans compared even to humans. So this is sort of a dendrogram of all of the cis-loop channels from various organisms, and the vertebrate channels are shown in red, and the nematode channels are shown in green, and you can see that there's quite a lot of green. And this includes some channels that are found in both humans and nematodes, like these GABA-A receptors, also some of these like glutamate-gated chloride channels that are specific to invertebrates. But also there are some worm-specific families, such as these ones that contain the known biogenic amine channels, and this one, which has no known ligands, which potentially could include new channels that could be activated by monoamines or other things. And so this is just showing a table from Wormbook showing that for these two families, most of the ligands had not been identified. So two postdocs in my lab, Iris Hartiga and Julia Morad-Lekholm, decided to use a very old technique of expressing these channels in Xenopus oocytes and performing two electrode voltage clamp on the oocytes that express these different C. elegans genes, and then just throw on all the monoamines and really anything else they could think of and see if they could find ligands for these orphan channels. And to summarize a lot of work, they found a lot of channels that have ligands either that are monoamines or other types of ligands. So they found one new serotonin receptor, 4 new receptors that are gated by dopamine and or tyramine in this monoamine group. They've also found 3 choline acetylcholine receptors, 2 receptors that are sort of novel polymodal, and also 2 new acetylcholine receptors, a new GABA receptor, and 2 new histamine receptors. So I'm going to focus for the rest of this talk just on the monoamine receptors. So this is just showing you the three homomeric dopamine tyramine gated channels. And you can see that, that if you look at their dose responses, in general, they're mostly low affinity receptors. In particular, this GGR3 receptor has EC50s in the 70 and 600 micromolar for tyramine and dopamine. And this is in contrast to most of the monoamine activated GPCRs, which have EC50s in the tens to hundreds of nanomolar ranges. So these may be receptors that would be well suited to synaptic as opposed to extrasynaptic signaling. We could also do ion substitution experiments to determine whether these channels are anion channels or cation channels, and thus whether they're excitatory or inhibitory. And most of these channels, it turns out, are chloride channels and thus inhibitory. And this is just showing that if you perform these ion substitution experiments, if you substitute chloride, you see this positive shift in the reversal potential when you substitute out chloride that's indicative of an anion channel. And this isn't necessarily surprising because all of these channels are related to GABA A receptors more so than to, say, nicotinic receptors. But there was an exception, and that was this serotonin receptor called LGC50. And so this is just showing that for LGC50, we see the opposite effect in the ion substitution experiment. We see this negative shift in reversal potential when we substitute sodium. And this is interesting because if we look at the evolutionary relationships in this family, the closest relative of LGC50 is this other serotonin receptor, MOD1, which is a chloride channel. And if you actually look at this sequence around this M2 transmembrane domain that's supposed to be important for ion selectivity, you can see that this is highly conserved among other members of the family, but some of these residues, in particular, this otherwise completely conserved proline, have been changed in this LGC50. So we don't know whether this particular amino acid change is sufficient to switch from an anion channel to a cation channel. But clearly we can use the evolutionary diversification of this family to identify how this has been able to switch in evolutionary time from an anion channel family to a cation channel. I mean, basically what we've done here is convergent evolution has evolved a 5-HT3 receptor from a GABA A-like receptor. So it's really sort of remarkable in a way. Now, we've also been looking at the expression patterns of all of these new ligand-gated ion channels. And many of them, as you can see in these pictures up here of transcriptional reporters, are expressed in relatively restricted groups of neurons. And if we look at where these neurons fit into the synaptic wiring diagram, in most cases, they're major postsynaptic targets of monoaminergic neurons. So these LGC51 and 52 and 54 are expressed in these postsynaptic targets of dopamine or tyramine releasing neurons. And interestingly, this co-expression data allowed us to identify a heteromeric channel composed of LGC51 and 52. You can see that LGC52 on its own is a dopamine selective channel. But if we co-express LGC52 with LGC51, which makes no channels on its own or no functional channels on its own, it shifts it to also being well activated by tyramine. Anyway, to get back to this expression though, one thing that we also found that was interesting, in particular about this serotonin receptor LGC50, is its expression in this neuron RIA. And the reason we found this interesting was because work initially by Yun Zhang when she was a postdoc with Corey Bargman had shown that this RIA neuron was important for one of the best defined forms of learning in C. elegans, which is called pathogen avoidance learning. So basically, if you culture C. elegans on a pathogenic bacteria like Pseudomonas, the naive worms actually like the odors of Pseudomonas, but if the worms have been grown on Pseudomonas and allowed to get infected and get ill, then they'll learn to avoid those odors. And work by Yun in her own lab using cell ablation experiments identified these synapses between this serotonergic ADF neuron and this RIA interneuron as the critical first step in the neural pathway for learned aversion. So we were, of course, interested to know whether LGC50 receptors could be involved with this learned avoidance of pathogen. So we sent our strains to Yun in the first instance, and she tested them in their behavioral assays. And interestingly enough, she found that these LGC50 mutants were very strongly defective in learned avoidance of Pseudomonas odors. Now we found something else interesting when Julia decided to tag the LGC50 receptor with GFP and look at the distribution of the protein in C. elegans. And so initially, there's very little LGC50 protein detected in the nerve ring where the synapses would normally be formed. But if we grow these worms under the conditions of this learned aversion assay in the presence of a pathogenic bacteria, we find the appearance of puncta of LGC50 in the nerve ring, and we see them with the same time course as the behavioral acquisition of pathogen avoidance. So this suggests the intriguing possibility that LGC50 receptors may actually be inserted at RIA synapses during learning, and that this may actually be a key mechanism for pathogen avoidance learning, is the insertion of these receptors. And this is also interesting because the in vitro properties of the LGC50 receptor are consistent with this sort of idea. So if we look at actually the currents that are produced by expressing LGC50 compared to the other serotonin receptor MOD1, we see that they're orders of magnitude smaller. And we think for various reasons that this is because the receptor is not trafficking efficiently to the surface in these oocytes. Now, it turns out that you can swap this large intracellular domain between MOD1 and LGC50 and confer these either this poor expression or this good expression, depending on whether it has the LGC50 cytoplasmic domain or it has the MOD1. So if we put the MOD1 domain on LGC50, we get good trafficking. If we put the LGC50 domain on MOD1, we get poor trafficking. And not only that, but we can actually delete this small region of this domain of only about 15 amino acids. And that's sufficient to actually give us a very efficiently trafficked receptor. And just in terms of why we think that this is actually trafficking that's giving us this expression, if you actually look at the protein and compare, for example, the levels of protein you see in wild type versus in the deletion mutant where we see good currents, we see that there's, if anything, more protein in this in these wild type oocytes that give us very poor currents. So we really think that what this domain is doing is conferring regulated trafficking on this receptor. And of course, one thing that we hope is true is that maybe this regulated trafficking process may be part of the learning mechanism. So this is, again, something that we're actively investigating at the moment. So finally, I just want to mention that some of these other channels that we found also have some interesting properties. And this is just sort of a funny one that we found in this diverse group, which is actually activated by both acetylcholine and also by monoamines. And of course, these are completely chemically different compounds. And preliminary data suggests that we can actually get mutations that selectively affect activation by either acetylcholine or by ectopamine or tyramine. And actually, the expression pattern of these also is consistent with them mediating both monoaminergic as well as cholinergic synaptic transmission. So finally, I just want to mention that this whole analysis of this C. elegans LGC family suggests that our view of fast synaptic transmission from thinking about vertebrates may be a little bit restrictive. So typically we think of fast synaptic transmission as involving three excitatory neurotransmitters, acetylcholine, glutamate, and serotonin, and two inhibitory ones. But if we look more broadly at all of the C. elegans ligand-gated ion channels, we find that there are both excitatory and inhibitory channels for all of these neurotransmitters, except glutamate, which nematodes don't seem to use. Moreover, they also seem to have ligand-gated channels for all kinds of other molecules that aren't even used as fast neurotransmitters in vertebrates, like dopamine, tyramine, octopamine, and so forth. And finally, we can also see that this diversification also can give us differences even with things that bind the same ligand in regulated trafficking that could be linked to things like learning and memory. And so basically, you know, we've sort of done things that maybe a synthetic biologist would want to do. We've basically seen that C. elegans through evolution has evolved ionotropic receptors with all these diverse sorts of properties that basically allow synaptic transmission to occur in very different ways than it occurs in vertebrates. And this may not be just specific for nematodes. So a new postdoc in the lab, Amy Courtney, has also looked at some other species, including this flatworm and octopus, which as far as I know, the ligand-gated channels haven't really been characterized that much. And you know, all of these different phyla contain nicotinic receptor-like channels and GABA-like channels. But most of these other phyla also contain these outlier groups that seem to have quite different sequences from these standard nicotinic and GABA-A-like channels. So these are the nematode monoamine channels. These are the mammalian 5-HT3 receptors, which aren't found in nematodes. But clearly there's these other flatworm channels that seem to be different from those found in other phyla, plus lots of novel things in octopus. So it could be that if we start looking at some other types of animals, we'll find other surprises in terms of how different molecules can act in fast neurotransmission. So to summarize what I've told you, we've seen that monoamines and neuropeptides act largely extrasynaptically in C. elegans, and these wireless neuromodulatory networks can be mapped to give you this multi-layer neural connectome. We've also found that monoamine-gated channels in C. elegans mediate synaptic neuromodulator signaling and may be particularly important for synaptic plasticity. And finally, that neurotransmission itself, both in C. elegans and possibly in other animal phyla, is evolutionarily plastic, and different ligand-gated ion channels can evolve diverse functions, both in terms of ligand binding, ion selectivity, and even regulation. So I would just stop there and just acknowledge the people that did the work, in particular, Lydia, who did the stuff with the neuropeptide connectome, Iris and Julia, who did the stuff with the ligand-gated ion channels, and of course, our very important collaborators. So all of the network stuff that I've done has been a collaboration with my colleague Petra Vertes at Cambridge, and of course, Oliver and Isabel have been the co-leaders in this neuropeptide connectome effort, and Yun for her help with the learning stuff. And I'll stop there.
All right, great, thanks. I think Bill can't actually hear me, but I'm cutting and pasting the questions from the Q&A into his chat. So I think that let's see if I can get an acknowledgement, or I just cut and paste the first question from Ruben Falkovich, who asks, is there a significance to the gradient of neuropeptide monoamine across the animal can be measured? For example, weigh each edge in each in connectome by the average concentration that receptor sees given its location.
I can't hear anything, actually.
Right. Okay, I'm just chatting.
Oh, yeah.
Right.
Okay, I'll try to answer the first question. I don't understand why my sound isn't working. I apologize for this Zoom issue. So the first question, is there a significance to the gradient of neuropeptide monoamine across the animal, and can it be measured? That is, can the weight in each connectome? Yeah, so this is a really important question, and it's definitely something that we want to address, because so for many of the neurons that we're talking about that have processes in the nerve ring, I don't think space is an issue, but there are clearly some receptors that are expressed in places like the tail or even the ventral cord where the neuromodulator would have to diffuse some distance. And I think it's really critical to understand whether these actions of neuromodulators at a distance occur, and if they do, whether their temporal dynamics are different from short range effects. And so this is why we're trying to develop these fluorescent sensors that are based on this principle of attaching a circular commuted GFP to a neuropeptide GPCR and then expressing them in target cells and seeing if we can visualize this signaling, because I think that's an extremely important question, and I think we need to find ways to address that. So that's a really good question. Next question is.
The next question is from Ellen Hill. It says, you talked about the functional roles of some of the neuropeptide hub neurons, how the synaptic rich
club. You talked about the functional roles of some of the neuropeptide hub neurons. How are the synaptic rich club and the neuropeptide hub neurons physically distributed throughout the animal? Are they near each other? Yeah, so that's another really good question. So one of the interesting things about the synaptic rich club is many of them are in the head, but some of them are in the tail. And actually, so in that original Rich Club paper, which was a collaboration with Emma Towson and Petra and some other people, one of the things that they looked at was the wiring costs of the Rich Club versus the typical neuron in C. elegans. And actually, the neurons in the Rich Club made up a disproportionate amount of the wiring cost of the overall animal, which was consistent with the idea that they were functionally very important, that the animal invests a lot of effort into wiring them up. In terms of the ones that have a high that are highly connected in neuropeptides, they also seem to be distributed throughout the animal. I mean, again, most of them are in the head, but most neurons in general are in the head, and there certainly are some of those neurons that are in the tail. And so I think that also is consistent with the idea that they could be doing long range as well as short range signaling.
Okay, the last question is from Ernesto.
Eve Marder says there's a long history of pharmacology in mollusks that have characterized many of the responses you're now starting to see. It is now gratifying to see them surface more generally. I completely agree with that comment. And in fact, I probably, before I mentioned the White et al. Connectome, I should have, of course, mentioned the work of Eve and other people in the labs of somatic gastric ganglion, which of course was really the original connectome, and also illustrates the point that merely knowing where the connections are is not enough to understand function, as Eve said in her particular lecture this morning. How is the regulation of electrical synapses considered into the wired connectome analysis, and how does it differ from chemical neuromodulation? So yeah, so that's another really interesting question. So it turns out that you can look at the correlation between where synaptic edges occur and where gap junction edges occur, and in comparison to, say, where neuromodulatory edges occur. And actually, the synaptic edges and the gap junction edges co-occur much more than any of these other type of edges co-occur. So, and that doesn't seem like functionally that would have to be the case, but it actually seems like most of the gap junctions are actually part of mixed synapses with chemical synapses. And so I think that's probably significant in terms of understanding the role of gap junctions in computation. And I think that obviously that would change the functional properties of the connection if you have a gap junction and a chemical synapse.
Okay, I think we're done. We're a little bit over time, but
okay, we're done.
Bill,
okay,
Bill can't hear me. Maybe he got my last message. We are scheduled to have a little break and reconvene at 4 o'clock, and that's in 22 minutes. That's 4 o'clock Eastern time. So I want to thank all of the speakers, including the one that can't hear me, but I'm going to text him. Thank you all, and we'll see everyone back here in 22 minutes.
God damn it. What is this
Welcome back, everyone. We'll go ahead and get started. I'm Troy Littleton. I'm a faculty member in the Pickower Institute, and I will be moderating our final session of the afternoon. And our first speaker of the afternoon is my colleague, Steve Flavell in the Pickower Institute. He'll be telling you about his work on a neural circuit for flexible control of persistent behavioral states. Steve?
All right. Troy, are my slides and voice coming through?
Yes, you're fine.
All right, great. Thanks. So as we've heard today, whether animals are mating, fighting, or foraging, they often organize their behaviors into long-lasting states. And even in the face of being able to generate these persistent behavioral states, animals obviously also retain a great deal of flexibility. For example, they can switch behavioral states rapidly when the sensory context changes. And so in the lab, we've been interested in trying to understand how neural circuits implement this balance between long timescale stability and flexibility. And we've been studying this in the context of a set of behavioral states that C. elegans animals generate while they're foraging for food. And you can see these states right here on this slide. So what I'm showing you is a data set from one wild type C. elegans animal that was recorded for 90 minutes as it freely explored a bacterial food source. C. elegans eat bacteria as their diet, so the animal was just placed on a fairly homogeneously seeded plate of E. coli. And you can appreciate that over these 90 minutes, there are these moments in time where the animal was traveling with high speed in green, low turning rates in blue, in these so-called roaming states. This is the animal actively exploring through its environment. And you can see that when the animal is roaming, these states can persist for 10 minutes, 20 minutes on end. And then every now and then the animal flips to a reciprocal state called dwelling, where it has very low speed and turns very frequently. And these dwelling states are also remarkably persistent, lasting for minutes up to an hour. And we think this reflects the animal foraging for food. Why is that not working? We think this reflects the animal foraging for food. And that's because when there's less food around, the animals spend more time roaming or exploring. And when there's more food around, animals spend more time dwelling or exploiting that food source. So basically the animals are evaluating the food environment and deciding, should I stay or should I go? And so a few years ago, we became interested in trying to understand the neural circuitry that generates these states. And in previous work, we found two opposing neuromodulatory systems in the worm critical for these states. So first of all, the serotonergic system of the worm is critical for them to display long-lasting dwelling states. And then the PDF, pigment dispersing factor neuropeptide system, is critical for animals to show long-lasting roaming states. And so we mapped out the neurons in the worm's connectome that make and detect these two neuromodulators in a manner that's required for these behavioral states to be regulated appropriately. And that gave rise to this kind of distributed neural circuit shown here. And so from top to bottom, this is kind of just a classic sensory motor hierarchy of the worm, a bunch of chemosensory neurons feeding into sensory processing neurons. And a few of the neurons down at the bottom here send projections down the ventral cord and can influence locomotion. And you can see that serotonin produced by these two neurons here on the left acts on multiple levels of this hierarchy. And when it acts on this circuit, we found that it drives animals into these stable dwelling states. PDF, this other neuromodulator, is produced further downstream in the circuit, and we saw that when it acted on the circuit, it drove animals into stable roaming states. Now, our identification of this circuit allowed us to start asking new questions about how these behavioral states, these foraging states, could really be flexibly regulated, such that animals made efficient foraging decisions in complex sensory environments. And so that's really what I want to talk about today. And there's really two things I'll go over. The first is the question of how that circuit really integrates and receives information about food availability, which is obviously this really important cue that allows animals to allocate their time between roaming and dwelling. And then secondly, ultimately, what's the functional architecture of that circuit that I just showed you? What are the elements that give rise to stable behavioral outputs? And how does this circuit enable the animal to flip states really rapidly and suddenly? And as you can see, these neurons are really densely connected. All of these gray arrows are synapses from the worm connectome. And so we can't just kind of gaze at these synapses and infer the functional architecture or logic of the circuit. And so we've been trying to deduce that through experiments. Okay, so first of all, how does that circuit receive information about food availability? It turns out that we were able to make progress on this problem just by studying this one neuron, NSM, in greater detail. And this was the work of Jeff Rhodes in the lab. And so this neuron NSM is a serotonin-producing neuron. And in previous work, we found that NSM has these big calcium peaks right at moments that animals switch from high-speed roaming to low-speed dwelling. This made some sense. It releases serotonin, which is the neuromodulator that promotes the dwelling state. And so we just wanted to know natively what drives the activity of this serotonergic neuron. And there were two important clues. The first is that this neuron NSM is really interesting morphology. So this is its soma here, and from its soma, there are these two major branches where it releases its serotonin. And actually, these serotonin release sites are opposed to the main neuropil of the worm's brain, which presumably allows this neuron to exert a strong influence on many neurons throughout the worm's nervous system. Then in addition, there's this third neurite that's almost hard to see in this picture because it's very thin. And in EM, this neurite has no synapses along it, but extends actually all the way to the alimentary canal, where it's exposed to the bacteria that the animal actually eats. In addition, NSM also has really dense synaptic inputs in the wiring diagram. And so we first of all tested whether these inputs onto NSM were driving these calcium peaks. And we did that by crossing our NSM calcium imaging line to a strong mutant background called UNC13, which has a strongly attenuated, nearly abolished synaptic transmission. And we were surprised to see that in these UNC13 mutants, NSM was still robustly active. Each of these lines is a calcium recording of NSM from a different animal. Suffice it to say, 30 minutes each, suffice it to say that there's at least as much NSM activity in these UNC13 mutants as there is in wild type. But somewhat bizarrely, these UNC13 animals can still eat. It's about all they do. They're otherwise paralyzed. So we removed the animals from food, and then we saw that the activity of NSM was abolished. And so this result really suggests that even in the near absence of synaptic transmission, information about food availability is making its way to this neuron NSM. Now, food is obviously a really complicated stimulus. It provides odors to the worm. There's mechanosensory cues as the animals move around on it. There's proprioceptive cues associated with eating, and then post-ingestive cues like satiety. And so we disentangled which of these features of food availability really matter for activation of this neuron. And it turns out that really we can only observe activity in this neuron NSM while animals are successfully ingesting the bacterial food in their environment. We can even play mean tricks on the animal, like we can optogenetically pinch their mouths shut. We do this by contracting their pharynx. And you can see here when we turn on red light to pinch their mouths shut, basically, that this neuron NSM, its activity falls off a cliff, and the neuron stays inactive as long as the animal isn't eating, even though things like food odors are obviously still present in the environment while we do this. And other experiments that we did, for example, we fed C. elegans animals beads the size of bacteria, and that totally failed to activate this neuron. These experiments, together with a few others that I won't go through, really led us to conclude that NSM is only active when animals are successfully ingesting bacterial food. This obviously reminded us of this neurite that NSM extends to the alimentary canal. And so we wanted to test whether this neurite could be involved in this feeding-dependent activity. And fortunately, developmental geneticists had identified a mutant, UNC101, that's defective in the outgrowth of that neurite, but not the other neurites made by NSM. And so we took a look at these UNC101 mutants. And to look at them, we used an assay that's pretty simple, where we just remove animals from food and then let them freely navigate their way back to a food patch. And kind of unsurprisingly, based on what I just told you, when they do this, right when they bounce into the food patch and start eating, NSM in wild type animals shows a big burst of calcium influx. But those UNC101 mutants lacking that neurite that goes to the alimentary canal, they have a really attenuated response. And so altogether, these types of experiments make us think that NSM is really an enteric sensory neuron, probably using this sensory dendrite to acutely detect the ingestion of bacterial food. And we thought this was interesting, so we wanted to identify the molecular machinery in NSM that was mediating this detection of bacteria or bacterial products. And the reason we wanted to go that far is because there's this interest in trying to understand how the microbiome or bacteria in the gut can ultimately signal to the brain. And here in this neuron NSM, we have a very simple case where this appears to be happening. And so to get a hold of that molecular machinery, we did molecular profiling of this neuron NSM, and then did a candidate screen where we looked at mutants lacking the different receptors or channels expressed by this neuron NSM. And the clear candidates that came out of that screen were these two genes called DEL3 and DEL7. And so DEL3 and DEL7 are close paralogs. They both encode A6 subfamily sodium channels, which is a class of sodium channels conserved all the way up to mammals. And I'll just show you two lines of evidence that really kind of support an important role for these channels in mediating this bacterial detection by NSM. First of all, their localization. And so DEL3 and DEL7 are more or less uniquely expressed in NSM in the worm. DEL3 is in just a few other neurons as well. And we asked where within the NSM neuron these channels were localized. We did that by tagging the native DEL7 gene with GFP using CRISPR, and then asking where those GFP tagged channels localize within NSM. And so first of all, here in red is the full morphology again of this neuron NSM. It's soma, those two major branches, and pretty hard to see here is this neurite that extends to the alimentary canal, but I'm zooming in right on the posterior tip of that neurite. And that's because it turns out that the only place in the animal that we can detect these GFP tag channels is right at the tip of that neurite. And this is really reliable across animals. And we thought this was notable because this neurite actually terminates right in the grinder of the pharynx, which is exactly what it sounds like. The grinder is the place that all the bacteria are being mechanically ground up while the animal is eating. And so this says that these channels are on the membrane in extremely good position to be detecting bacterial products. In addition, these channels have an important functional role. They're really necessary for this feeding-induced NSM activation. Again, we can measure that using this assay where we just remove animals from food, let them navigate their way back to a food patch. And when we do this, we see that wild-type animals show a big burst of NSM activity when they eat, and the animals switch their locomotion to these low-speed dwelling states more or less immediately. And mutant animals lacking these DEL3 and DEL7 channels have a strongly attenuated GCAMP response and an attenuated behavioral response. Moreover, we can rescue this phenotype by restoring expression of these channels just back into the NSM neuron. And so this really leads us to the conclusion that this serotonergic neuron NSM, really important for these dwelling states, is actually an enteric sensory neuron. It's acutely detecting the food ingestion for the animal. And these new ASIC genes, DEL7 and DEL3, are mediating that bacterial detection in NSM. The types of open questions here that still interest us that I won't go into any further today are questions like whether DEL3 and DEL7 are directly part of the sensory transduction pathway, mediating detection of bacteria. And we also really want to know what's the bacterial signal that activates NSM. And so these are things that we're actively pursuing in the lab. And I'll mention in passing, that this role of NSM in mediating this kind of gut to brain signaling is really reminiscent of the role that the serotonin producing enterochromaffin cells play in the mammalian gut. These are cells that basically line the alimentary canal. They can detect contents of the lumen, and they can signal to the to the CNS via serotonin release onto vagal sensory afferents. And so here in C. elegans, we have a serotonergic cell type seemingly doing a very similar thing. And so our continued studies of this kind of gut to brain signaling hopefully can reveal other molecular mechanisms involved here. Okay, but this kind of leads nicely into the second part of the talk and what I mostly want to mention today. So our understanding of that NSM neuron that I just told you about kind of nicely explains why animals dwell more in appetitive food environments, right? They eat more bacteria, the bacteria acts on those channels, activates NSM, and that drives serotonin release, which is this neuromodulator that drives the dwelling state. But when animals are in appetitive food environments, like this animal right here, they actually can switch to roaming states just fine and sustain these roaming states for tens of minutes. So one possible explanation for that could just be that animals don't eat while they're roaming. But we've carefully quantified this, and it turns out not to be the case. Animals have basically identical food consumption rates or pharyngeal pumping rates across these two states. So another possibility was that there might be ongoing circuit dynamics that relate to these behavioral dynamics. And perhaps other components of the circuit in which NSM is embedded, perhaps these other components of the circuit actively inhibit NSM to keep it off while the animals are roaming. And so we decided that we really wanted to know more about the broader neural circuit controlling these states and how the dynamics of that circuit really relate to these behavioral states, these stable states and abrupt transitions. And when people think about these kind of stable states or stable patterns of circuit activity, they commonly think about three types of mechanisms or motifs. The first is recurrent excitation, and this can obviously allow circuits to generate lasting responses to transient inputs. The second is cross inhibition, which can allow for alternative states and sharp state transitions. And then the third is neuromodulation, which can allow neurons to signal to one another over longer timescales than is typically possible with classical transmission. And so if these types of motifs within circuits really kind of promote stability, or long time scale persistence, an important question is how dynamic incoming sensory inputs act on these types of motifs to really lead to flexible state transitions. And so we wanted to look at this in the roaming and dwelling circuit. Where are these motifs within that circuit, and how are incoming sensory inputs in more complex foraging environments acting on these motifs to regulate these states? And this was really the work of a fabulous postdoc, Ni Ji, in the lab, who was joined for part of the project by a grad student, a fabulous student, Gurin Maran. And so Ni figured that if we wanted to understand this, that we would need to record the activity of neurons throughout the circuit as animals were freely foraging and switching between these states. And so she constructed a GCamp line expressing GCamp in most of the neurons in this circuit. And because neurons in C. elegans are always in the same place in every animal, in her recordings, she could know which fluorescent spots in the head corresponded to exactly which ground truth neuron in the worm's connectome. And she recorded these animals under a microscope that she constructed, inspired by the work of Andy Leifer and Aravi Samuel. And this microscope basically has two light paths. The light path below here allows us just to do fast volumetric imaging of the worm's head while it's moving around. And that allows us to see the GCamp signals of all of these different neurons. And then the light path above the animal records the animal's behavior and controls a motorized stage, resulting in a closed loop tracking system. Okay, so we did this while the animals were roaming and dwelling. What does the data look like? So this is what the data looks like off the scope. First of all, just one neuron, the NSM neuron, the serotonergic cell I keep talking about. The calcium trace of this neuron in this example is in black, and the colors in the background indicate the ongoing behavior of the animal. So when the color is green, the animal is going forwards quickly. When the color is red, the animal is going backwards and making a reversal. And when the color is a really pale color, the animal tends to be going slowly. And so you can see, right, this neuron NSM tends to be active while the animals are going slowly in these dwelling states. But now we have data not just from NSM, but from the other neurons simultaneously recorded in the circuit. And different neurons in the circuit have very different relationships to behavior. Some neurons tend to be most active when the animal is going forwards in green, like this AVB neuron. Other neurons are really only active when the animals make turns, like this AVA neuron. Every time the color goes red to indicate an animal is making a reversal, AVA turns on. And so we can kind of quantify these relationships. What we were really interested in, though, is whether there were modes of dynamics across this circuit that were really associated with these stable states and state transitions. And so to get at that, we performed principal component analysis on these data sets to find the dominant modes of dynamics in the circuit. And when we do this, the first couple principal components actually have very intuitive correlates. And I'm showing that here. So this is just basically a scatter plot showing lots of data from lots of animals projected into this principal component space. And here I'm false coloring the data points by the ongoing velocity of the animal. And so when the color is green, the animal's going forwards quickly. When it's red, the animal's going backwards. And you can immediately appreciate that this PC1 is just directly correlated with the animal's velocity. When PC1's high, the animal goes forwards. When it's low, they're going backwards. And this just says that there's a set of coordinated changes across neurons in this circuit that are associated with these forward-backward locomotion dynamics. The second principal component, it turns out, is really most closely associated with the foraging state of the animal. When PC2 is high, the animal tends to be dwelling. And more particularly, the serotonergic neuron NSM tends to be highly active. And you can see that here. And I should say that we observe this principal component even if we withhold the NSM neuron from our analyses. And so this result just kind of says that there's a set of coordinated changes across the circuit associated with the dwelling state and with the activity of the serotonergic cell. And so it should be kind of immediately obvious that it's actually pretty easy for us to tell you the behavior of the animal from these circuit recordings, right? Even these two axes shown here can more or less predict what the animal's behavior is based on activity. But we wanted to know how the functional components of the circuit, how the neurons were interacting to give rise to these kind of coherent activity changes across the circuit. And we decided to focus on two features of activity across the circuit. The first is persistent activity. And the reason for that is that neurons in this circuit, like the NSM neuron, tend to be highly active, not just for a second or two, but for long stretches of time. When NSM becomes active during a dwelling state, its activity is sustained for many, many minutes, closely paralleling the duration of that state. So where does that persistence come from? And then secondly, we wanted to know how mutual exclusivity arises in the circuit. And that's because it turns out that the neuron NSM that makes serotonin, its activity is mutually exclusive with the activity of another neuron called AVB, which makes that PDF1 neuropeptide, kind of a nice neural correlate of the antagonism between these two modulatory systems. So where does that mutual exclusivity come from in the circuit? And so it turns out that both of these features of the circuit's activity come from the neuromodulators themselves. And the way that we were able to get at that was by recording the activity of the circuit in mutant animals lacking these different neuromodulatory systems. So let me just quickly show you what I mean by that. So first of all, if we look at the animals lacking serotonin, these TPH1 mutants, which can't synthesize serotonin, we saw that under the microscope, first of all, these animals had shorter dwelling states, something that we fully expected to see. But then in addition, we saw that the duration of time that NSM stays active during its activity bouts were greatly truncated. And this wasn't something we were necessarily expecting. And because we observed this phenotype, not just in the TPH1 mutants, but also in mutants lacking this serotonin receptor MOD1, which is Bill just mentioned in the last session, is a serotonin-gated chloride channel and inhibitory receptor expressed on downstream neurons. This really suggests this type of general functional architecture, where NSM inhibits neurons expressing this MOD1 receptor, and these neurons must somehow, perhaps indirectly, feed back to inhibit NSM. This would allow the release of serotonin to stabilize NSM's own activity, which is what's suggested by this result here. Okay, what do we see in the mutants lacking the other neuromodulatory system, the PDF system? Well, we see in those animals a loss of this mutual exclusivity. Again, in wild-type animals, NSM and AVB, these neurons that make these opposing neuromodulators, have mutually exclusive activities. Here's a scatter plot. This is just kind of an example trace from an animal showing that NSM turns on after these forward active neurons like AVB and RIB turn off. And in mutants lacking PDF signaling, lacking the only PDF receptor in the worm called PDFR1, this mutual exclusivity is kind of gone. And instead, actually, it's more common to see NSM co-active with AVB than it is to see NSM active on its own. And when we look at the raw data, we just see these really weird bouts of activity in these animals, lasting for tens of minutes sometimes, where NSM is co-active with the entire network that drives forward locomotion, or at least the parts of the network that we recorded in these experiments, like AVB and RIB. And because AVB is a neuron that releases this PDF peptide, we interpret this to likely mean that the AVB's release of PDF during roaming likely keeps the NSM neuron off typically during that state in wild-type animals. And consistent with that interpretation, we saw that when we looked at PDF gain-of-function animals, that NSM was basically always off in those animals. And those animals also more or less constantly roam. And so adding that to what I already told you from the serotonin null experiments, these other experiments with the PDF mutant suggests that somehow PDF signaling is functionally required to keep the NSM neuron off during the roaming state. Now we've asked whether these two feedback branches are in fact kind of one and the same. We did that through epistasis experiments and through experiments like where we activate one group of cells while inhibiting the other. And the upshot of that's all shown here. And so what we're seeing is that there's this kind of core mutual inhibitory loop here, where the serotonergic cell NSM inhibits these MOD1 expressing neurons. Again, this neurons expressing this inhibitory serotonin receptor. Those neurons seem to have functionally excitatory action towards the neurons making the PDF receptor. And somehow, we don't yet know how, these neurons feed back to inhibit the NSM neuron. And we see that these neurons, NSM and AVB, which have mutually exclusive activities, they don't appear to directly interact with one another. We've looked for that, but haven't yet gotten any evidence that it happens. Instead, their interaction seems to be mediated by them both interacting with this network of neurons in the middle here. And this is just a cohort of neurons that express the neuromodulatory receptors and are connected through synapses. And many of these neurons are well known to orthogonally receive dense feed-forward inputs from the chemosensory system. And so this kind of overall architecture really raised the obvious possibility that incoming chemosensory inputs could change the activity of these neurons and drive behavioral state transitions. And so we decided to start looking at that. And we began by looking at the neurons that Nhi had recorded in her GCamp line that were part of the chemosensory system. And so one neuron that Nhi recorded really caught our eyes. And so this is the last thing that I'll mention. This neuron that caught our eyes is called AIA. It's a second order neuron in the olfactory system of the worm. What we saw was that AIA is active during both roaming and dwelling, but it switches which network it's coupled to in the animal when the animal switch states. And so I'll show that here just in these example traces. So when the animals are roaming, AIA is active primarily when the animal is going forwards. Again, the green color here in this example is the animal moving forwards. Red means the animal is going backwards and making a turn. And AIA is co-active with the rest of the forward network, neurons like RIB, during the roaming state, and it shuts off when the animals make a reversal. At that moment, a bunch of reversal active neurons like AVA turn on. So that's what AIA does during the roaming state. Now, when animals switch to dwelling, most of the neurons that drive locomotion, like RIB and AVA, actually really reduce their activity. But this neuron AIA stays reliably active. And during the dwelling state, then it becomes correlated to the NSM neuron. So in other words, AIA is active in both states, but it becomes coupled to different neurons depending on which state the animal's in. And we thought this was kind of interesting. We did experiments, like we tried to ask whether AIA could drive behaviors typical of either of these states by optogenetically activating it just in freely foraging animals. And when we did this, we got these really weird results where in some trials, AIA would activate the roaming state, and in other trials, AIA would drive the dwelling state. And so optogenetics are obviously a fairly artificial way to activate this neuron. And so that got us thinking about what's really the native way that this neuron is activated in the animal. And really the native thing that drives the activity of the cell AIA are the vast majority of its synaptic inputs, which come from chemosensory neurons. And a lot of these upstream chemosensory neurons are well known to be involved in detecting food odors, an obviously important cue during foraging. And actually really nice work from the Bargman lab last year showed that this neuron AIA generates reliable responses to appetitive food odors, including bacterial odor mixtures. And so this really raised the possibility that this neuron AIA might couple the olfactory environment, food odors, to these behavioral states. And so we decided to test that, and we tested it by comparing the behavior of wild-type animals to animals in which AIA was silenced. And we compared these animals in the context of a behavioral assay where animals had to use their olfactory systems to guide these behavioral states. And this is really a pretty simple assay. All that we do is we take animals and we position them on a sparse bacterial food plate, but we just seed a really dense patch of food nearby with a very abrupt border. And that means that these animals, while they're foraging, can come into proximity of an odor gradient and navigate that gradient to get to a better food source. And so it turns out wild-type animals are great at doing this. They make it to the better food source. And when we carefully quantify their behavior as they do so, it turns out that they really use two strategies to do that. First of all, they increase their roaming when they come into proximity of that odor gradient. That's this dark bar versus the gray bar right next to it. And this says that the mere presence of this odor gradient arouses the animals to roam more. And then while the animals are roaming, they perform efficient chemotaxis and navigate up that gradient. And we saw that animals in which that neuron AIA was silenced, those animals fail to do this. They aren't aroused to roam more in the odor gradient. And when they do kind of randomly switch to roaming, they don't efficiently chemotax up the gradient either. And so this says that in the sensory context, that AIA is primarily promoting the roaming state and chemotaxis during that state. But consistent with this kind of dual function of AIA, we see very different roles for AIA in different sensory contexts. Like if we just expose animals directly to food, either a sparse food only plate or a dense food only plate, then animals really like to dwell in these environments. There's no alternative. In particular, they love to dwell in these dense food environments. That's this bar right here. And when we silence AIA in these environments, there's a reduction in dwelling and a net increase, therefore, in roaming. And so this says that in these sensory environments, upon direct food exposure, AIA is actually promoting the opposite state, the dwelling state. Now, these results actually start to make some sense to us, actually, in light of these behavioral assays. And the way that we're thinking about this is that when animals are foraging, these food odors that they detect are actually a very ambiguous sensory cue. For example, here in this case on the left, when animals are navigating this food odor gradient from a nearby food source, the increased concentration of food odors that they detect signal a need to the animal to roam more and to chemotax up that food gradient to get to the better food source. But over here on the right, when animals actually encounter a better food source, they also detect an increased concentration of food odors. That's detected coincident with increased ingestion, and that actually signals the need for the animals to dwell more. And so in other words, the mere detection of an increased concentration of food odors isn't sufficient to specify the appropriate behavioral state response. Instead, this needs to be contextualized with other cues, like whether those increased food odors are being detected coincident to increased ingestion. And so we think that AI's ability to detect these food odors and to be able to potentially transmit this information to different downstream circuits might allow animals to make efficient use of information about odors in the environment, but to contextualize this information appropriately. And obviously, based on what I told you earlier in the talk, our knowledge of this NSM neuron, the fact that we know that it actually detects food ingested inside the animal, provides a pretty plausible mechanism for how these cues could be coincidentally detected by the circuit. Okay, so just to wrap up, I told you here in this part of the talk about our efforts to characterize the functional architecture of the circuit. And I told you that the neurons that make these opposing neuromodulators have rivaling activities, and that the function of the neuromodulators that they release are to promote the stability and mutual exclusivity of the circuit states that correspond to dwelling and roaming. Right, the loss of serotonin led to an imbalance in this winner-take-all system where the NSM neurons shut off more quickly. The loss of the PDF peptide led to odd correlations in the network where the roaming and dwelling circuits were coactive, as opposed to in wild-type animals where they have mutually exclusive activities. And the rivalry between these neurons, or these networks, is not mediated by NSM and AVB directly interacting. Instead, it seems to be mediated by them both interacting with this network of neurons here in the middle, a cohort of neurons that actually make the modulatory receptors and receive orthogonal feed-forward chemosensory inputs. And so I mentioned that these feed-forward inputs aren't all of a flavor that's really easy to understand, just promote more roaming or promote more dwelling. Some of these inputs, like the ones that come in through AIA, provide bifurcating outputs to both parts of the circuit and require additional contextualization for the animals to really generate efficient foraging responses. But we think this overall architecture really allows the animals to make flexible responses to sensory cues and potentially to make efficient foraging decisions in complex sensory environments. So I just want to thank the people who actually did this work. I tried to highlight them throughout the talk. Jeff Rhodes was the real driver of the work on those ASIC channels in NSM and NSM's detection of food ingestion. And Niji was the real driver of this project on the functional architecture of the circuit, again, with really important contributions by Gurun Madan. Other folks in the lab also made important contributions. And I also want to thank our collaborators. Daniel Colin Ramos was a collaborator for part of the DEL7, DEL3 project. These folks for strains and these folks for funding. Thanks.
Thanks, Steve. We have a time for a couple of questions. Since there's no questions in the chat yet, I'll ask a couple. Do you think the DEL3 and DEL7 channels themselves are responsible for directly depolarizing the neuron?
Yeah, so we're really interested in trying to understand this better. Based on their localization, we think that they could definitely be involved in mediating directly that food sensation, given their localization. But we haven't yet done the heterologous expression experiments necessary to draw that conclusion. And the holdup there is that we need to identify the bacterial components that are actually activating NSM natively before we can deliver them to a heterologous system. And so that's what we're actively doing. But I should mention one control that we did, for example, these channels aren't just kind of generically involved in the calcium handling or generic excitability of NSM. So Jeff did an experiment where he recorded the activity of NSM while directly activating it with crimson. And he saw across a wide range of light intensities that wild-type animals and the DEL mutants had the same responses. So they're generically involved in excitability, but they are involved in the feeding-dependent activation.
Okay, we do have a question from the chat. Does pre-starvation change the way that NSM and AIA respond to food and odor?
Yeah, that's a great question. So starvation does make a difference, but we haven't quite figured out, it makes a huge difference, first of all, for these behaviors. Animals dwell more when they're starved. And what we observed for NSM activation is that if you consider the case where the animals are off food, navigating their way back to a food patch, starved animals, the second they encounter that food patch, reliably, almost 100% of the animals slow down and start dwelling. When animals are roaming, when animals are well fed and they encounter a food patch, sometimes they slow down, but sometimes they persist roaming through the food patch a bit before they transition to dwelling states. And so we think that starvation certainly primes the circuit to be kind of more dominated by the NSM neuron. And we're trying to understand where that really arises in the circuit. The other thing that we know is that, yeah, the inputs coming into AIA and generally the chemosensory inputs to this system, work being done by Ian McLaughlin in the lab, we're seeing that there is a lot of change in the olfactory drive when animals are starved versus well fed. And so we think that that's probably playing an important role as well.
We have a question from David Anderson. Are NSM neurons truly persistently active in the sense of continuing to fire independently of their initial stimulus? Could it be that while they're dwelling, they're eating, and as long as they're eating, the NSM neurons remain active? Yeah,
that's a great question. And so, yeah, I think the latter might actually be true. And so like so I think that basically, and the reason for that is basically when the animals are off of food or not eating, we never observe activity of this neuron. And so the way that we're thinking about that to kind of put these things together is when the animals are foraging on food, they're basically constantly sampling the food. And so that should be a tonic stimulus to the NSM neuron, basically constantly depolarizing it. And we think that its persistence comes from periods of inactivity of the roaming circuit. And so we think that the real dynamics, and I should have made this a little bit clearer, are kind of from the downstream components of the circuit and this mutual inhibitory loop that basically the rest of the circuit can shut off. NSM dominates it through that inhibitory receptor and kind of keeps it off for a while until the winner-take-all system switches, the roaming circuit becomes active, and then that can keep NSM off despite the fact that the animals are still eating and that there's excitatory drive onto the NSM neuron. So the roaming circuit is kind of the part of the circuit that we think probably has these inherent dynamics. Yeah, and the NSM probably is just getting tonic input.
Great. There are a few more questions in the chat that you might want to respond to, Steve, by typing, but given the time, we'll move on to our next speaker. Thank you very much. Great talk.
Thanks.
Our next talk for the session will be from Mario Penza, who is at the National Institutes of Mental Health. He'll tell us about the paraventricular nucleus of the thalamus, biases to selection of passive and active fear. Mario?
Can you hear me now?
Yes, we can hear you.
Can you see my slides?
Yes, you look good, thanks.
So first of all, I wanted to thank Steve and the scientists at MIT Picard Institute for giving me the opportunity to share some of the work from my lab at the symposium today. It has been a great honor to participate in this event. So like many of you today in the audience and part of the panelists, my lab is interested in understanding how the brain orchestrates adaptive behavior. And in particular, something that my laboratory is really interested in studying is the neurobiology of emotional states such as fear. And as has been nicely proposed by David Anderson and Ralph Adolph, we share this view of emotions and fear as central states that are elicited by some type of stimuli and that drive these parallel responses that we can study in the laboratory. So in the context of fear, which is what interests my lab, we can measure this type of responses all the way from somatic responses to observed behavior, with the exception of course, of subjective reports, which is a category that currently is exclusively for humans. And as you guys know, a lot of what we know about fear in the literature mainly comes from studies of Pavlovian fear conditioning, and of course its characteristic freezing behavior in the rodent literature. And there is no argument that studies of Pavlovian fear conditioning have impacted or have generated a great deal of knowledge about the networks of the brain that participate in the processing of fear and this fear state that we've been talking about. In particular, these studies have led to this amygdala-centric view. Let me see if I can turn my pointer here. To this amygdala-centric view of fear, in which other structures like the medial prefrontal cortex and the hippocampus also play a critical role. But it is important also to realize that when considering the type of behaviors that are elicited by a fearful state, freezing, is only one metric of fear, and in that regard, studies of Pavlovian fear conditioning have not generated an equally rich literature or data in another type of fear response, which is active avoidance. Similarly, Pavlovian fear conditioning has also not generated sufficient knowledge about mechanisms in the brain that drive the selection of different types of defensive behaviors. I think what I'm saying is better illustrated here in this chart, which shows a number of publications by year, and you can see that unlike studies on fear conditioning, which have seen this massive increase in number of publications per year in the last 30 years, studies on active avoidance have stalled since the late 80s, and this is adapted from a nice review article published from Christopher Kane, published last year. So today I would like to shift attention from the fear conditioning and move into the topic of active avoidance. And specifically, I would like to discuss unpublished data from my group, in which we have identified that a structure of the brain which we and others previously identify as being critical for fear conditioning and freezing behavior also is similarly involved in guiding active avoidance. In addition, which we have evidence which I will present today that suggests that this area of the brain, which is the paraventricular nucleus of the thalamus, seems to play an important role in biasing the selection of these two types of fear behaviors. Okay, so what do we know currently about active avoidance? So classical models of active avoidance view it as a two-stage process in which there's a first stage that is dominated by Pavlovian conditioning, and this is an entirely amygdala-dependent process that depends largely on freezing behavior that's supported by the central nucleus of the amygdala. Of course, with additional training, there's a shift in the behavioral strategy of the animal in which this Pavlovian freezing is attenuated, and then goal-directed avoidance behavior emerges. And this involves a recruitment of different circuitry, particularly the nucleus accumbens, the ventral striatum, and diminish activity of the central nucleus of the amygdala and its accompanying freezing behavior. And of course, with additional training, there's a further remodeling of this circuitry and the appearance of more habitual-like behavior. Now, when we look at this two-stage model of active avoidance, we quickly realize that there's two structures within the brain, within this circuit, that are seemingly very important for this process. There's the central amygdala, which is important for freezing behaviors, and studies have shown that manipulations of the brain that result in diminished activity of the central nucleus of the amygdala, whether it's pharmacological inactivation or lesions, decrease freezing behavior, but actually potentiate avoidance behavior. In contrast, manipulations of the ventral striatum, particularly the nucleus accumbens, have been shown to attenuate active avoidance behavior and to promote freezing behavior instead. So these results suggest that these two regions of the brain, the central nucleus of the amygdala and the nucleus accumbens, are in some kind of antagonistic interaction for the selection of defensive behavior, which leads to the idea that perhaps identifying regions of the brain that directly interact with these two regions could be helpful for our understanding of the neurobiology of this selection of defensive behaviors. Of course, a logical candidate for this is the basolateral amygdala, which is a structure of the brain that innervates densely both the central nucleus of the amygdala and the nucleus accumbens. And studies have shown that the basolateral amygdala and its interaction with these two structures is important for both freezing behavior as well as active avoidance behavior. But there are some studies that have come to suggest that the necessity of the basolateral amygdala for the expression of both freezing and avoidance seems to decrease over time, suggesting that there's some kind of system consolidation process and the engagement of other regions of the brain for example, for remote memories. So this suggested to us that perhaps there are other regions of the brain that may be implicated in guiding the selection of passive and active fear. So this led us to focus our attention in the paraventricular nucleus of the thalamus, an area that we have been studying for some time now, because we know that the paraventricular thalamus densely innervates both the central nucleus of the amygdala and the nucleus accumbens. In addition, work from Fabrizio De Monte and Gregory Quirk, and also from my time as a postdoc at Cold Spring Harbor with Bo Lee, we've shown that PVT communication with the central nucleus of the amygdala is important for fear conditioning and the expression of freezing behavior. However, today there are no studies which have addressed the question as to whether PVT projections to the nucleus accumbens are important for avoidance behavior. And this is a question that a very talented postdoc in my lab, Jun Ma, was interested in addressing when he joined the lab a few years ago. So upon addressing this question, the first thing that Jun wanted to explore was whether neurons of the PVT that project to the nucleus accumbens and the central amygdala belong to a common population of PVT neurons. So in order to do this, he used a retrograde tracer, chlorotoxin B, which is conjugated to different fluorescent dyes, in this case, a red fluorescent dye, and in this case, a green fluorescent dye. And he injected these retrograde tracers in either the nucleus accumbens, specifically one color, and in the central nucleus of the amygdala, a different color. Then he looked in the paraventricular nucleus of the thalamus, and consistent with data from the laboratory of Gilbert Kerouac, Jun found that neurons of the PVT that project to either the central nucleus of the amygdala, here in green, and those that project to the nucleus accumbens, here in red, largely belong to a non-overlapping population of neurons. In addition, Jun found that these long-range projecting neurons of the posterior PVT, particularly, we're interested in the posterior PVT because of our previous studies on the importance of this part of the PVT in aversive processing. And Jun found that most of these posterior PVT projection belong to a proportion of dopamine D2 positive cells of the PVT. And this was important because in a recent publication, we showed that neurons of the PVT that express D2 receptor belong to an aversive modulated population of PVT neurons. So next, Jun wanted to address whether these D2 positive cells of the PVT actually participate or signal or become engaged during active avoidance. So for this, he injected the genetically encoded, the virus expressing the genetically encoded calcium indicator GCAM6 in the posterior PVT of D2 Cree animals. And then he implanted an optical fiber over the PVT to use a technique that you're familiar with and has been mentioned today, fiber photometry, to monitor changes in the activity of these neurons in their calcium fluorescence. And then he took these animals and he trained them with active avoidance. Essentially, the task, animals are placed in this shuttle box in which there's two compartments that are separated by a hurdle. And animals learn that presentation of an auditory cue precedes the subsequent presentation of a food shock, and they learn that if they shuttle to the adjacent compartment during presentation of the CS, the presentation of the US or the food shock is completely prevented. So they gain instrumental behavior that allows them to avoid the presentation of this food shock every time the tone is played. And consistent with previous reports, you can see that initially the behavior is largely dominated by Pavlovian freezing behavior during the presentation of the CS. But with training, there's this decrease in freezing behavior and the emergence of active avoidance. So Jun took these animals and he imaged in the last couple of sessions of active avoidance. And then he separated trials that he recorded into either avoidance trials or failure trials. Because remember that in some cases, the animal will avoid presentation of the food shock by shuttling to the next compartment, but in other cases, they wouldn't. So what he observed, and you can see here in the summary plot here in the heat map, is that here presentation of the tone was associated with a rapid increase in calcium signal in these D2 expressing cells of the PBT. Importantly, you can see that the rise here in the signal coincided with the termination of the tone, meaning that the animal has shuttled. This can be visualized much better when looking at individual trials. You can see here in these trials that the shaded green area behind the traces actually depict the entire expected duration of a tone, which is 15 seconds, but the black line shows the actual duration. And what you can see here is that every time that the tone terminates, this coincides with a previous rise in calcium signal that continues after the termination of the CS. So this suggested to us that D2 expressing neurons of the PBT were activated during the shuttling behavior. Importantly, when we're looking at failure trials, we observe that during failures, we actually observe mainly a reduction in GCAM signal during these failure trials and a subsequent activation of these neurons during presentation of the US, which is consistent with previous findings from our lab, that these neurons are sensitive to adversive stimuli. Of course, a rationale was that this decrease in fluorescence was perhaps associated with freezing behavior occurring during these failure trials. Indeed, we observed that when we look at the behavior that animals show higher freezing behavior during failure trials here in red than during avoidance trials shown here in blue. Importantly, when we look at time lock events about the behavior to gain some more information about what these neurons were doing, we found that indeed when animals reach maximal velocity during shuttling or shock elicited escapes, we noticed that such increase in such maximal velocity coincided with the increase in GCAM fluorescence with a maximal increase in GCAM fluorescence in these neurons of the PBT, suggesting that these neurons were somewhat signaling the vigor of the behavior. In addition, we observed that when animals were actually engaging in freezing behavior, this was associated with a decrease in calcium fluorescence. So collectively, these findings suggest that while D2 expressing neurons of the PBT are activated during active avoidance, they are attenuated during freezing behavior. Next, to investigate whether these neurons were actually important or required for the expression of active avoidance behavior, Jun took animals that were injected, these are D2 Cree animals that were injected with Cree-dependent halorhodopsin in the posterior PBT, and then he trained them with active avoidance, and then he performed three tests, first of which was considered as a pre-test to assess avoidance rate pre-manipulation of the circuit. And you can see here that animals typically display this high avoidance rate. However, upon silencing of D2 expressing PBT neurons during the following day on the next test, you can see that the active avoidance behavior was largely attenuated. And this was consistent with also accompanied by an increase in the latency to avoid. a small but non-significant increase in freezing behavior, specifically in halo expressing animals. So this was to some extent a bit surprising to us because previous findings from our group and from others have shown that the paraventricular nucleus of the thalamus is important for driving freezing behavior. So now we find that these neurons of the PVT are actually attenuated during freezing behavior and are signaling active avoidance. So what's going on? So one of the first experiments that June did to address this was to actually look at the activities of this neuron, these D2 expressing neurons of the PVT, during a retrieval test following classical fear conditioning. So what you can see here is that when June assessed the activity of these neurons during during the retrieval test, we didn't see much modulation of this signal. However, June then clustered the different trials into either low freezing trials, moderate freezing trials, or high freezing trials. And what he observed is that in contrast to the low freezing trials, high freezing trials were associated with an attenuation of calcium fluorescence from these D2 neurons, consistent with our previous finding that these D2 expressing neurons of the PVT are actually attenuated during freezing behavior. Interestingly, June also compared whether there was any type of correlation between locomotion during the fear retrieval test and the calcium fluorescence. He observed that whereas during prior to fear conditioning, during habituation, there was not much of a correlation between the animal's movement and signaling these two neurons. During the freezing trials, he could observe a very nice, significant correlation between movement and fluorescence, consistent with our previous finding in active avoidance. So when we've had this result, we of course we had to take a step back and wonder what this will mean in the context of PVT's role in freezing behavior and fear conditioning. But of course, one thing that is important to keep in mind is that fire photometry is a population-based approach. So when using fire photometry, the activity of the neurons that we're measuring in the posterior PVT here represent the collective activity of the D2 expressing cells that have GCAMP. And as I showed previously, these neurons can actually be segregated into those that project to the nucleus accumbens and those that project to the central nucleus of the amygdala. So the next thing that we wanted to explore was whether active avoidance led to a specific engagement of accumbens projecting neurons. So for this, June again used fire photometry, but in this case he expressed GCAM7 in the D2 expressing neurons of the posterior PVT, and then he implanted an optical fiber over the nucleus accumbens to image the activity at these terminals in the nucleus accumbens. He again trained these animals with active avoidance and did imaging during the later portion of the training, as I described previously. So consistent with the results that I showed you before, we observed that during early presentation of the tone, when animals mainly displays some freezing behavior, there was attenuation in this terminal activity, but upon shuttling to the next compartment, there was an increase in calcium fluorescence. Also consistent with our previous observation from the cell body imaging, you can see here that during the failure trials, there was also an accompanying decrease in GCAM fluorescence, presumably because animals during the failure trials are largely freezing. So to explore this further, we again conducted similar analysis as I described previously. And you can see here that indeed these PVT to accumbens projections are largely signaling the maximal velocity of the animal, again consistent with the idea that they are related to the vigor of the movement of the animal. And instead, during moments of freezing, either during avoidance trials or failure trials, these moments of freezing were associated with attenuation in GCAM fluorescence in these terminals. Interestingly, this didn't translate to the projection to the central nucleus of the amygdala. June did not observe that the activity of these neurons was associated with maximal velocity. Interestingly, he did observe that failure to avoid was associated with CS-mediated activation of PVT to central amygdala projections, perhaps because these neurons are largely engaged during failure trials to sustain freezing behavior. So the next thing that June wanted to investigate was whether this PVT to accumbens communication was actually important for the expression of active avoidance. So he used a similar manipulation as previously described. In this case, he expressed halorhodopsin in the PVT, but he implanted optical fibers right above the nucleus accumbens to attenuate these projections. And after training animals with active avoidance, he performed 3 tests. One pre-test here, where you can see that these animals show robust avoidance behavior on day 1. But on day 2, when this PVT to accumbens connection is silenced, you can see that this leads to a significant impairment in avoidance behavior. You can also see here the accompanying increase in the latency to avoid. Even in trials in which animals successfully avoid, the latency is increased. Importantly, we also observed that this manipulation was accompanied by an increase in freezing behavior, demonstrating that silencing a PVT projection to the nucleus accumbens not only attenuate active avoidance behavior, but also potentiate freezing behavior in these subjects. Now, upon a closer look at this finding, we were quite intrigued by the fact that on day 3, when June performed a post-test in which the circuit was intact, it appeared as if active avoidance was still suppressed. These results were very intriguing and interesting to us because of previous results from Fabrizio Domonte and Gregory Quirk, in which they show that PVT projection to the central nucleus of the amygdala are important for freezing behavior. Indeed, attenuating this connection suppresses freezing behavior. But importantly, these behavioral impairments persist on day 8. Now, the additional conclusion from these studies were that PVT likely participate in driving the maintenance of fear memory, and that upon attenuation of this connectivity during fear memory retrieval, the memory is essentially permanently damaged, if you will. But there's a finding, an observation in our findings that seems to contradict that conclusion to some extent, because as you can see here, whereas active avoidance was permanently impaired after silencing of PVT to accumbens projection, freezing behavior was potentiated, and this potentiation in freezing behavior persisted. These results suggested to us that PVT's role in fear memory may not necessarily be in CS-US association or modulating maintenance of this association, but instead in time fear association to specific behavioral outcomes, whether that is an active instrumental response like active avoidance or a passive behavior like freezing. So to summarize some of what I've shown you so far, we know from previous studies that PVT to the central nucleus of the amygdala is important for supporting freezing behavior and the retrieval of fear memory. But here we show that PVT projections to the nucleus accumbens not only attenuating this connection decreases avoidance behavior, but it also potentiates freezing behavior. So we wonder whether silencing of PVT to central amygdala communication not only attenuated freezing behavior, but could also potentiate active avoidance behavior. And this is something that has not been explored. However, there is some precedents in the literature that serve as an inspiration for us to pursue this avenue. And that is the following. When animals are trained, and particularly this is a study from the Ledoux lab in rats, both in rats and mice, there's typically a subset of subjects that do not learn or do not seemingly learn to avoid well enough. And they never seem to break this 20% rate of avoidance towards the end of training. These rats, and in our case mice, are typically deemed as low avoiders. And so an interesting observation by Joe Ledoux, and this has also been observed by Justin Moscarello, is that the central nucleus of the amygdala seems to be actually important for this low avoider behavior, because if you lesion the central nucleus of the amygdala, you see this unmasking of active avoidance behavior in low avoider rats. This suggests that because of the central amygdala supporting freezing behavior, overactivity of the central nucleus of the amygdala in these low avoider rats likely explains why they don't engage in avoidance behavior. Whereas after lesioning, you can see the rapid emergence of active avoidance. So we wonder whether PBT to CA projections could be involved in this process. So June screened for a subset of mice that behave in this way. We term these mice low avoiders. And then he took these mice and he expressed halorhodopsin in D2 expressing neurons of the PBT, and then he implanted optical fibers bilaterally over the central nucleus of the amygdala. And as you can see here, similar to what previously shown in rats with manipulations directly of the central nucleus of the amygdala, attenuating PBT to central amygdala communication unmasked avoidance behavior in these mice, but not in control subjects. And this is accompanied by a reduction in freezing behavior towards the end of training. And as you can see here, we observe the emergence of a correlation between freezing behavior, between the decrease in freezing behavior and the increase in avoidance rate in this subject. So to summarize what I've shown you today, I've shown you that the paraventricular nucleus of the thalamus, which innervates both the central nucleus of the amygdala and the nucleus accumbens, seems to play an important role in biasing the selection of these two types of fear-related behaviors. Now, we wanted to test this model further, and if you remember from earlier in my talk, I showed you that attenuating PBT to accumbens communication led to a significant impairment in active avoidance behavior that persisted a day later. So we wonder whether this persistence was due to the PBT to central amygdala pathway dominating the behavior. So in order to address this question, June did this complicated experiment in which he actually implanted optical fibers bilaterally over the nucleus accumbens, as well as over the central nucleus of the amygdala. He then repeated this experiment and he showed that attenuating PBT projection to the nucleus accumbens indeed suppressed avoidance behavior. But interestingly, a day later, when he actually attenuates next the PBT to central amygdala communication, avoidance behaviors was restored. So with that, I would like to summarize what I've shown you today. So first, I show you that posterior PBT neurons that express D2 receptor signal active avoidance via their projection to the nucleus accumbens. These PBT accumbens neurons are also inhibited during freezing behavior, and optogenetically silencing of PBT to accumbens communication seems to not only attenuate avoidance behavior in a lasting way, but drive a concomitant and lasting increase in freezing behavior. In addition, I show you that optogenetically silencing PBT to central amygdala communication promotes avoidance behavior and also attenuates freezing behavior in low avoiders. Finally, I show you that we have this evidence suggests that the CEA and accumbens projections of the paraventricular nucleus of the thalamus dynamically bias the selection of passive and active fear, respectively. So I think I'm out of time. So I'll show you in this last slide a little bit of what we're doing to further explore these observations. So because we see these differences in PVT central amygdala projections and PVT to accumbens projection, we wonder whether these two type of PVT neurons receive different inputs. So we are doing rabies, monosynaptic rabies tracing that's depending on the target. In this case, tracing the inputs onto accumbens projector or central amygdala projectors. And this is work done by a very talented postdoc in the lab and Sophia Beas, who's still a postdoc in our group. And you can see here that in preliminary findings that we have, looking at retrograde labeling and neurons of in the brain that project to these two type of PVT neurons, we observe that whereas some of these regions of the brain here shown in red preferentially innervate accumbens projector cells in the PVT, there are some regions of the brain that seem to preferentially innervate central amygdala projecting PVT neurons. So with that, I will go to my thank you slide, and I would like to acknowledge the people that did the work, specifically June, who's the postdoc that led this work and did a fantastic job in the 2 years that he has been in the laboratory. Of course, I'd like to acknowledge the contributions of some of our collaborators, Johan Du Hoffman and Yogita Shudosama from the Rodent Behavior Core, who have helped us in the development of the active avoidance task, and also in some of the data analysis that I've shown you today. Lastly, I would like to thank Morgan Kindle and Sophia Beas, who have been working in the rabies tracing experiment that I've discussed, with support from the group of Ted Austin at the System Neuroscience Imaging Research of the NIMH. So with that, I would like to thank you all for your attention, and I'll take questions.
Thank you, Mayo. We'll wait for a few questions. So while we're waiting, can I just sort of ask you a broad question about what kind of situations might drive an animal to freeze versus actively avoid a threat, and sort of what type of sensory inputs might drive one of those responses versus the other?
Yeah, so that's a very interesting question, and we haven't explored much of that. I would like to say that in the active avoidance task, one of the reasons why we like this task is because during the task, the animal fluctuates back and forth between freezing and active avoidance behavior. So, you know, I think what's interesting about what we've observed is that it seems like the midline thalamus seems to play a role in favoring the selection of one behavior over the other. But the mechanisms that actually drive this favoring of these behavioral outputs, we don't really understand them, and that is part of the research focus in the lab at the moment.
Okay, a question from the chat is, how does PVT talk with BLA? What's the transition like when PVT takes over projections to CEA and NAC during fear learning?
Yeah, so the PVT sends mainly projections to the nucleus accumbens and the central nucleus of the amygdala. There are also some projections to the basolateral nucleus of the amygdala. And an interesting observation from the group of Gregory Quirk with Fabrizio De Monte is that they show that during remote fear memories, the basal amygdala becomes less important for the retrieval of fear memory, and this is overtaken by the midline thalamus. I think there's also some data from the lab of Li Chun Luo that sort of supports this finding, because they found that neurons in the prefrontal cortex that are important for the retrieval of remote memories are found in the deeper layers of the prefrontal cortex, which are the thalamic projectors. So there seems to be some kind of consolidation of the circuitry that's involved in the retrieval of remote fear, and it seems like the particular nucleus of the thalamus plays an increasing role with the passage of time.
Great, thank you, Mayor. I appreciate your talk. We'll move on to our final talk of the symposium today. This will be given by Richard Axel from Columbia University. He'll tell us about his work on order from disorder, internal representations of the olfactory world. Richard.
Thank you, Troy. Let me also thank Steve and the members and staff of the Picard Institute for their organization of this symposium and for providing an oasis of thought during these deeply troubling times that we are all experiencing. Now, Pat Kuhl, a neurobiologist of language, attempted several years ago to teach young American children Mandarin phonemes, and she succeeded. And then she attempted to teach them using solely audiovisual tools, and she failed. They learned nothing. And so if you learn nothing from my talk today, don't blame me. Can you see this image?
Yes, looks fine.
Good. This beautiful painting by the pre-Raphaelite artist Dante Gabriel Rossetti depicts Paolo and Francesca, two illicit lovers who find themselves in the second circle of hell. 100 years later, the master of the Zip, Barnett Newman, painted exactly the same image. Newman's Paolo and Francesca consists of lines, forms, colors that bear no resemblance to the physical world. This is an abstraction, and meaning must be imposed by an individual's experience. This is precisely what the brain must do. It must represent the perception of the external world by the firing of neurons. Meaning must then be imposed upon this abstract representation of neural activity, either by evolution or by experience and learning. This afternoon, I'd like to consider how meaning and value are imposed upon an abstract representation of odors in two brain structures, prefrontal cortex and hippocampus. Olfactory perception is initiated by the binding of odorants to sensory neurons that express a single receptor in the olfactory epithelium. Neurons expressing a single receptor are broadly distributed in the epithelium, but converge on a fixed locus in the olfactory bulb. It immediately follows, given the presence of 1,500 receptors, that a given odor will activate a combination of receptors, which in turn will activate a combination of loci or glomeruli in the olfactory bulb. And this is indeed what is observed in this very pretty image of two-photon imaging of the olfactory bulb by Tom Boza and his colleagues. And indeed, every odor elicits a distinct spatial pattern of neural activity so that an experienced observer can look down upon the olfactory bulb through a two-photon microscope and tell you what odor the mouse has encountered based upon his experience. Now, this is not, however, the question, because the question is, how does the mouse know what odor it has encountered? And a mouse doesn't have a two-photon microscope in his brain. And so we followed the information downstream to ask how meaning is imposed upon spatial patterns of neural activity that define different odors. And so Doris Sasolsky and Bob Dada in the lab did this in a simple way by simply injecting a dye into one of the 1,500 glomeruli in the bulb and following the information from the 10 projection neurons that innervate this glomerulus And you see this rich pattern of innervation. But I would like to focus on the pattern of innervation in piriform cortex. What we see in piriform cortex is that the information from just one glomerulus, and this is mirrored by the innervation of all glomeruli, is diffuse. And several lines of evidence suggest that this diffuse representation results in the unstructured innervation of neurons within this primary olfactory cortex, such that piriform neurons receive input from a random collection of perhaps 100 glomeruli. And this is consistent with early imaging studies by Dan Stetler in the laboratory, which demonstrate that the precise spatial patterning of the olfactory bulb is discarded in piriform cortex, and every odor activates a distributive representation of neurons that are where neurons are interleaved with other neurons responding to different odors with no apparent topographic segregation. And so if indeed neurons in piriform cortex receive input from a random collection of glomeruli, then it follows that the patterns of neural activity in piriform cortex will be different in every individual, and the patterns of neural activity can have absolutely no value, no significance. And significance or value needs to be imposed downstream. This argument is supported by experiments, early experiments by Gloria Choi in the laboratory, now at the Picower, who demonstrated that she could create a random ensemble of neurons in piriform expressing channel rhodopsin and generate a diverse array of behaviors from a single random ensemble dependent upon the nature of experience. And so we asked, how is value imposed upon this distributive, unstructured representation? When I say we, I mean Christian Baboila and Peter Wang in collaboration with Larry Abbott, and everything I will tell you today is in collaboration with my friend and colleague, Larry Abbott. So what Peter and Christian did, since I do nothing, is to develop a very simple appetitive learning task in which four odors are administered to the mouse, and after a delay, 2 of the odors are rewarded with water. And what we observe, as in the past, is that one sees in the delay anticipatory licking only in those animals that receive water reward. They then simply performed 2-photon imaging and asked whether they could discern any alterations in the representation of the CS plus and CS minus odors. So as we've shown before, if you look at the representation of odors, you find that every odor activates a distinct ensemble of neurons. And if you compare the ensemble of neurons for both C and CS minus neurons before and after learning, one sees no apparent difference. there are small differences in discriminatory power that we observe, but these differences are unrelated to learning. So it seems that piriform neurons exhibit no alteration in the representation after learning, and this may afford piriform the flexibility to elicit multiple different types of behavior. And so piriform projects to many brain areas, but exhibits a very rich projection to prefrontal cortex. And so with fear and trepidation, Christian and Peter went into prefrontal cortex. And to me, going into prefrontal cortex is akin to Virgil, who could be Larry Abbott, leading me through the gates of hell, where above which it is written, abandon hope, all ye who enter here. For the prefrontal cortex affords a level of complexity beyond that which a molecular geneticist wishes to encounter. And what they observed when they imaged prefrontal cortex was a very clear alteration in the representation. Before learning, what we observe is that both the CS plus and CS minus odors activate a small number of neurons in a non-selective and variable way. And after learning, what we observe is that there is a dramatic change in the representation of the CS plus odors. Both CS plus odors activate about 30% of the neurons in orbitofrontal cortex. Importantly, we observe that over 70% of the neurons responsive to one CS plus odor are responsive to the second CS plus odor. The implication is that odor identity has been discarded in orbitofrontal cortex. The odor identity of piriform is no longer observed. Rather, we have transformed identity into value. In accord with this argument, we can create what we call a Gloria Choi ensemble in piriform, where we create a random ensemble, pair that ensemble with a water reward upon light activation. And what we observe is that the neurons in orbitofrontal cortex responsive to laser activation are the very same neurons responsive to CS plus odors. And so indeed, orbitofrontal cortex is representing what we believe is value. In accord with this, we can test this more directly. Our animals are water deprived, thirsty, and what we observe with CS plus odors here in 5 neurons in orbitofrontal cortex is a response to the 2 CS plus odors. If we perform the exact same behavior with CS minus odors, in the vast majority of the neurons, we lose the orbitofrontal response. And so to sated animals, the emergence of a water reward is of no value, and there is no orbitofrontal response. Similarly, the orbitofrontal response is context dependent. The animals learn that water is delivered through a water port, and if we take thirsty animals and remove the water port such that odor does not any longer predict reward, we observe that the CS plus odors fail to elicit a response in orbitofrontal cortex. We can demonstrate that the piriform to orbitofrontal cortex projection is capable of eliciting an orbitofrontal response if we inhibit the piriform to orbitofrontal cortex with halo. As you see here, we lose the anticipatory behavioral response. So it looks like piriform cortex, that orbitofrontal cortex is encoding value and is responsible for the behavior. Moreover, we can directly inhibit orbitofrontal cortex and demonstrate that we lose the anticipatory licking behavior. This led us to suggest a very simple circuit in which piriform, which encodes identity, projects to orbitofrontal cortex, which encodes value, and value is imposed upon the premotor region ALM involved in licking behavior. This all seemed reasonable, and until we observed that if we follow orbitofrontal cortex as learning proceeds, what we observe is the saga of the disappearing representation, whereas the animal continues to respond to CS plus odors for several weeks after learning. Within about a week after learning, we begin to lose the robust response to the CS plus in orbitofrontal cortex. And this posed an interesting enigma, perhaps not unlike the transfer of information for contextual memory in the hippocampus to medial prefrontal cortex, where consolidation and long-term memory occurs. And so we investigated this, but to investigate this, we had to set up an assay in which we could look at the early events that orbitofrontal cortex might participate in. And so we turned to a more classical appetitive assay in which an animal is given a single CS plus odor. This is known, which is paired with water. And that's defined as pre-training. And after the animal learns that odor means water, we then perform discrimination with 2 CS plus and CS minus odors. And what we observe, again, is that orbitofrontal cortex shows robust representation of value during pre-training, but that representation is significantly diminished during later discrimination. So orbitofrontal cortex is necessary for appetitive learning, but it disappears. And this immediately suggests that orbitofrontal cortex may be performing a teaching function on yet a second higher order brain region. And so we looked at the neural responses in medial prefrontal cortex during learning. And what we observe is medial prefrontal cortex shows little or no activity early in the training process, but we see a representation of the CS plus during discrimination learning. And interestingly, we see a distinct representation of the CS minus. And this representation, which follows the early representation in orbitofrontal, appears to be stable for as long as we can hold the 2-photon imaging paradigm, which is about 10 days. Now, the presence of a CS plus representation is perhaps not surprising. Gareth Stuber observed this for auditory learning in MPFC, But the presence of a CS minus representation is interesting because not only does a CS minus representation provide a fail-safe for the behavior, but it provides a mechanism whereby an animal can update and unlearn information. And so we could then go on and demonstrate, and I won't belabor this, using silencing experiments, that orbital frontal cortex is required for pre-training, but not for discrimination learning of the appetitive behavior. And finally, medial prefrontal cortex is not required for pre-training, but rather is plays a significant role in discriminatory learning. So this leads us to a more complex and more interesting model, which posits the disappearing representation and consolidation. Early in training, we believe that a representation of odor identity in piriform, where piriform represents that which is visible in the world, projects to OFC, and upon learning, OFC represents that which is not visible in the world. That is value, learning, experience, context, and internal state. But that representation, capable of eliciting behavior early, disappears, and OFC performs a teaching function on the piriform projections to medial prefrontal cortex, which then results in a stable representation of CS plus and CS minus in MPFC, which then drives the behavior. So these studies demonstrate the imposition of value on an unstructured representation in piriform, transiently in OFC and stably in medial prefrontal cortex. Order has been created from disorder, from the lack of structure. In experiments performed by Walter Fischler in collaboration with David Clark, Mark Schnitzer, and Larry Abbott, we've also shown that odor participates in the generation of a different abstraction in the hippocampus, the generation of a cognitive spatial map. Now, these early studies demonstrate that hippocampus transforms space in the world into an abstraction of brain space. Spatial representations result from the convergence of internal path integration and external sensory landmarks in the world. We have examined neural activity in the hippocampus of mice performing a simple navigational task that relies solely on path integration and sparse fixed olfactory cues to ask how odor combines with space to create a cognitive map. We've developed a simple one-dimensional spatial landscape, a mouse on a ball with no sensory features and 1 degree of freedom, creating a virtual track. This allows us to locally administer odors at various distances from the start to ask how odor combines with space to create a cognitive map. The convergence of idiothetic signals, path integration, with sensory information to create this spatial map allows us to test 2 important predictions. First, the same odor will be represented differently in the hippocampus at different positions in space. And second, spatial information encoding the same location will be represented differently in different sensory contexts or upon exposure to different odors. In initial experiments, we allow a mouse to run 4 meters on a virtual track and receive a water reward in the absence of other sensory landmarks and odor cues. We observe that mice initiate licking and decrease their running speed after about 2 meters along a 4-meter track. Thus, path integration alone cannot accurately measure distances greater than 2 meters. This is in accord with the sparse representation of place cells. Only 6% of the image neurons can be classified as place cells, and spatial density was maximum at the start and decayed exponentially with distance. These data imply that path integration alone cannot support viable place cell activity beyond 2 meters, and beyond 2 meters, the mice begin to lick. They don't seem to know where they are. We next examined whether spatially localized odor cues enhance both navigational behavior and place cell representation. Mice were exposed to a virtual track in which one of the 2 odors, limonene or pinene, were placed at 1 meter and 3 meters along the track. Interleaved trials were performed with the 2 odors. And after 4 days of training with these odor cues, all mice, as you can see, suppressed licking and maintained high running speeds for at least 3.5 meters of travel, licking only a half meter before the reward. This demonstrates that the mice recognize odor as a spatial landmark and use these landmarks to improve navigation. In accord with this, we observe the density of place cells increases from 6 to 36%. More, the density of place cells, as you can see, increases at all locations on the track, but is far more pronounced at 1 meter and 3 meters, the sites of odor exposure. Importantly, what we observe, if you look at these 2 loci of administration of the same odor, is that the same odor at 2 locations generates different uncorrelated sets of place cells, demonstrating that the same sensory stimulus is represented differently at different positions in space. Again, we are discarding odor identity in the formation of this convergent spatial map. Now, we next examined whether different odors that convey distinct sensory contexts elicit different spatial maps by interleaving pinene and limonene trials. And indeed, the 2 odors generate different place cell representations, a demonstration of remapping. If we sort place cells against limonene trials and examine place cells in the pinene trials, we observe that the majority of place cells between the start and the first odor cue are shared in both limonene and pinene trials. However, beyond the first odor cue, the place cells generated in the 2 sensory contexts are completely different. So these data confirm the second prediction for the formation of a convergent map. Different odors at the same location in identical tasks appear to serve as different landmarks generating distinct cognitive maps. These observations suggest that spatial information encoding distinct locations are represented differently in different sensory contexts. Finally, and lastly, we examine the evolution of place cell representation. On the first day of training, what we observe with odor cues is that the spatial density of place cells increases locally at the 1 meter cue, but not at the 3 meter cue. Over subsequent days of training, the density of place cells progressively increased to a value 5-fold greater than we observe on the first day. Additionally, the emergence of a peak in density, between 1 and 3 meters, and as training proceeds, the emergence of a peak at 3 meters appears. The emergence of peaks in place cell density at both 1 and 3 meters is gradual and sequential. So these results suggest an iterative process for spatial map extension, in which increasingly distal sensory cues are recognized as landmarks. First, the odor at the start is recognized as a landmark, resulting in a local peak of place cell activity. This leads to a gradual increase in place cell density following the first cue, allowing the mice to recognize the second odor cue as a distinct landmark. In turn, this leads to an increase in place cell density beyond the second cue, and this iterative process may reflect what we believe is the basic mechanism for extending spatial maps into unknown territory. In this manner, the convergence of path integration and olfactory landmarks in the hippocampus may allow the mice to construct spatial maps that support navigation over distances far greater than would be possible with path integration alone. So I've provided 2 examples, the prefrontal cortex and the hippocampus, which take information from unstructured representations in piriform and through learning and experience, transform this information into representations of either value or convergent space in the hippocampus. And so what these regions are doing is apparent, maybe analogized to what we are doing when we observe this abstraction of Leda in the Swan, which is visible in the now open MoMA in New York by Cy Twombly. And only upon learning and experience can you look at this abstraction and know that it represents the attempted seduction of the beautiful Leda by Zeus, who has been transformed into a swan. Thank you.
Thank you, Richard. We have one question in the chat asking about the anterior olfactory nucleus. It receives information apparently from the hippocampus. What is your idea of how this nucleus is playing a role in the process?
Yeah, the function of the anterior olfactory nucleus is not well characterized. We know that it is important, as minimum low is shown, for identifying the sidedness of an olfactory stimulus, and other studies have suggested that it may be important in representing socially relevant olfactory cues. It receives information from a number of brain regions, including hippocampus and amygdala, but the function of those projections not known to me.
When you were discussing your sort of transfer of memory from the OPC to the prefrontal cortex, do you think this is reflecting a connectivity change in the OPC cells onto these prefrontal cortex neurons, or did those neurons have a different input of odors outside of OPC cells?
Now, you're asking whether there's a change in connectivity in the orbitofrontal cortex upon disappearance?
Yes.
Yeah, so this is a very interesting question. We believe, with no evidence, that it may reflect, this disappearing representation may reflect a fast learning function for orbitofrontal cortex and subsequent overwriting of reinforced synapses. So yes, I believe that you're weakening reinforced synapses by overwriting.
And do you think there's a similar process, habituation, has sort of been looked at in a number of systems where it's sort of learning some new rules? These things where you'd remove the water port, for example, is that learning delayed, or the loss of that learning delayed when the OPC has already transferred the memory, or?
So I think that is done at a time where the representation is present. And I think that that is the consequence of a regulatory inhibitory input to orbitofrontal cortex. And so thirst would disinhibit that inhibitory context, as would context.
And the last question in the chat is, how do you think changes in the number of olfactory genes between mice and humans change representations and meaning imposed on OPC cells?
Could you repeat that? Sorry, how do
changes in the number of genes between mice and humans, how would that alter this biology you're talking about?
I'm not sure that it would alter my thinking about these processes. Of course, it's hard to introduce a 2-photon microscope into human prefrontal cortex, but I would imagine that we would see the same phenomena, despite the fact that there may be a third to a quarter the number of receptors.
Okay, we'll have one last question here. What happens to place cell representations in mouse behavior if a different odor is presented at 3 meters?
We haven't done that experiment. That's a very good question. I've been trying to convince Walter to do that.
Great. Well, thank you very much, Richard, for a great talk. And I'd like to thank all of our speakers today for some wonderful talks. I'd also like to thank Steve Flavell for putting together a great program for us. And on behalf of the Picower Institute, thank you for joining the symposium. We look forward to seeing you again next year. Take care.